Well, welcome everybody to the American Lobster in a Changing Gulf Causes and Consequences of Environmental Change session. My name is Amalia Harrington. I'll be facilitating our session this morning. I am a Marine Extension Associate with Maine Sea Grant at the University of Maine, but I'm also the Program Coordinator for the Northeast Regional Lobster Extension Project, which is part of the Sea Grant American Lobster Initiative. So I just wanted to provide a little bit of context for those of you not familiar with the initiative, as it really sparked the idea for today's session. So the initiative started in 2019 and is funded through NOAA's National Sea Grant Office program. And it includes both the extension project, which I coordinate, as well as a competitive American lobster research program. So across both of these branches, we're working to advance research on how the American lobster is responding to a changing environment. And we're an extremely diverse group of individuals. So we have members from the 7 Northeast Sea Grant programs on the extension side, and we have researchers, industry members, students, and nonprofits, as well as state and federal agency members from over 30 institutions across the Northeast and beyond. So it's a really diverse group of individuals, and we're working on a wide variety of projects, but we are all united under this broad goal of increasing the American lobster industry's resilience to the various impacts of ecosystem change across the region. So like this goal suggests in today's title, this was really the inspiration for today's session, and we really wanted to highlight the work of the researchers that are part of this program. So you'll hear from some of those researchers and students today, but to find out more about the initiative as well as the extension program, please take a look at our website listed down below. And we also received quite a bit of interest from folks outside of the initiative. So we have, again, a really diverse group of individuals that we're going to highlight today. And because of the interest, we have a really jam-packed agenda. And you'll notice both online and this quick agenda here that we are running until 1 p.m. with our session today. So we'll start things off with a keynote presentation by Dr. Malin Pinsky from Rutgers University. And then we'll dive into some traditional oral presentations. We have 5 of those before we have 4 5-minute speed talks. And then around 12:10, we'll dive into our poster session, which we've divided into 2 groups of 4 presenters. So like the rest of the sessions that we've had so far in our meeting, please make really great use of the Q&A function to ask our speakers questions. And I'd ask that you identify the particular speaker that you're directing your question to, as I imagine we're not going to be able to get to all of the great questions today given our lineup. So presenters, please take a look at the Q&A box when you're done, so that way we can address as many questions as we can today in this virtual format. And I'll also do my best to keep you all on track for today. So I will turn my video screen on when you have a couple of minutes left in your presentation. And if I have to, I will interrupt you, unfortunately, so that way we can keep to this agenda and let everybody share this really great body of work that we've put together. So with that, I will stop sharing my screen and turn things over to Dr. Malin Pinsky to start things for us with his presentation, Fishing and Planning for Species on the Move. So take it away, Malin, when you're ready.
Thanks so much. Can you see that?
Yep, great.
And you can, and I guess you can hear me. So I've managed to unmute. So thanks so much for that introduction and the invitation to kick off the session today, Amalia. As you heard, I'm Malin Pinsky, Associate Professor at Rutgers University. And as I get started, I just want to acknowledge with respect the Lenape peoples on whose traditional territory I sit today. So my goal today is to introduce what I see as a few key ideas on how marine species, fisheries, and the marine spatial planning efforts respond in the context of climate change. I won't talk a whole lot about lobster, but I do hope to provide really useful context and food for thought for all of the amazing talks that are coming next. So to set the stage, we know this is a time of unprecedented change in the ocean environment, and the Gulf of Maine in particular is an epicenter for ocean warming, in part because changes in the Gulf Stream are amplifying the direct effects of radiative heating. Given these rapid environmental changes, how do living systems respond to changing climate? So my goal is a mechanistic understanding across scales of biological organization, from individuals to populations, communities, and human societies. We need theory and data to understand how changes at one level of biological organization scale up to affect other levels, including society. As we do this, it's also important to recognize that organisms have an impressive diversity of ways to cope with environmental change. So for an individual, a certain amount is just tolerable from a physiological point of view. Behavior allows individuals to avoid stressful conditions, while acclimation allows for changes in tolerance over slightly longer timescales. If we scale up to populations, evolution can rescue a population otherwise faced with extirpation, or populations can expand into new territory with more suitable conditions. If these individual and population level mechanisms are insufficient, though, extinction is the result. And it's these population responses that then feed up to restructure entire communities and subsequently affect human society. So today I want to focus on 3 questions. How sensitive are marine animals to warming? How do marine populations respond to warming? And finally, how do we as a society adapt to these changes? On this first question, we can examine something called the thermal safety margin, which is the distance between the hottest body temperature an organism reaches and the hottest temperature it can tolerate. So colleagues and I compiled data from experiments around the world that measured the upper temperature that ectotherms could tolerate, both for species on land and in the ocean. We then ran microclimate models to calculate the highest body temperature organisms were likely to reach at each location that the species had been collected from, accounting for both their environment and their behavior. And these 2 pieces together allowed us to calculate thermal safety margins. I'm showing you that here on the y-axis across latitudes on the x-axis. We have terrestrial in green, marine in blue. Each dot is a species, and the average across latitudes are shown with the 2 lines. And I think the main finding here is that we see less thermal safety across all latitudes in the ocean than we see on land. And I think that's especially striking because only 10 years ago, there'd been very little attention to the impacts of climate change on ocean life. And now, as Amalia introduced, there are major research efforts focused on these questions. The take-home message from this individual level is that marine organisms are more sensitive to warming than are animals to land. And this difference has a lot to do with the greater opportunities for behavioral thermoregulation on land. The next question is, though, is how do those effects at an individual level scale up to populations? So if marine organisms are more likely to exceed their thermal tolerance, one expectation is that populations at that warm range edge of a species will be extirpated as temperatures increase, and that range edge will move towards the poles. If we look at lobster, we can see some of this playing out. The map is made from Northeast Fisheries Science Center bottom trawl data and shows the continental shelf from North Carolina to Canada. Yellow and orange show where there were high densities of lobster in the early 1970s. And we can see them all the way down to Virginia, at least offshore at that time. By 2019, however, populations across much of southern New England had largely collapsed, and the population had contracted up towards Georges Bank. And we were interested in understanding how general these patterns were across species and across different realms. So we pulled data from the literature on resurveys of warm range edges in ectothermic animals. And we focused in particular on multi-species papers to reduce publication bias. So for the 100 or so marine species in the data set, about half had contracted to higher latitudes, whereas on land, only a quarter had, suggesting that extirpations from warming are substantially more frequent in the ocean. Whether or not that leads to species extinctions, though, depends on what happens at the other range edge, and whether that cold range edge at high latitudes expands fast enough. So here I'll show you a comparison across 43 species of fishes in the Northeast US with rate of shift on the y-axis, either for the warm trailing edge or the cold leading edge. And this is work that was led by Alexa Fredston, a current postdoc in my group. And what we found is that those cold leading edges are expanding almost twice as fast as the warm trailing edges are contracting. And it suggests that for many of these species, environmental change actually can be an opportunity, not just a threat. So, by extension, it's good news for lobster in places like Newfoundland, though it's not especially helpful for the Gulf of Maine, where the question really is one of persistence despite environmental change. So while these long-term changes that I'm talking about over decades are, I think, really interesting and important, I often hear from fishers and managers that they need to make decisions over much shorter timescales. So it's a big reason we're now working on methods to forecast shifts in species distributions over sort of 1 to 10 year timeframes. And this is a project we're working on in collaboration with the Mid-Atlantic Fisheries Management Council. So to make these kinds of shorter term forecasts, we need to think much more mechanistically about spatial population dynamics, like dispersal and extirpation and population growth, and how these rates are related to temperature and other aspects of environmental change. So as an initial test, we have a Bayesian model running for summer flounder, in which the population has been divided into 1 degree latitude bands, shown here on the left of your screen. And recruitment is temperature dependent. So the model does a pretty reasonable job recreating past fluctuations in the location of the summer flounder population, shown here on this graph in terms of the centroid, so just the mean latitude of biomass. The red dots are observations from the bottom trawl survey, and the blue line is the hindcast prediction. We're now working on testing the forecast skill, which is what really matters. So the take-home message, though, I think, is that marine species both contract and expand their distributions rapidly in response to changing temperatures. And that's actually quite different from the way that terrestrial species respond to environmental change, at least in terms of dominant mode of response. So I think some of the important questions: how do we adapt? So one finding from research I've been doing with a number of social scientists, including Kevin St. Martin here at Rutgers, is that there's a lot of bottom-up adaptation in fisheries, even if regulations are not changing. So just to show you one example, this map shows with a red square where large fishing trawlers from Beaufort, North Carolina were fishing in the mid-1990s. And by 2014, though, the boats were going all the way up to New Jersey, about 1,000 kilometers round trip, largely to catch summer flounder that had shifted north. And this shift has also started to drive quite a conflict among states over allocations and who should get to catch those fish. While striking, though, these changes in fishing location are actually pretty rare when we've looked at fishing trip reports across the Northeast US. Instead, we found that switching to new species is much more common in this region. As one example, southern New England lobster fishers, in many cases, switched to catching Jonah crab after the lobster decline around the around 2000, the year 2000. Jonah crab wasn't much of a fishery before that, but fishers could continue using traps, and Jonah crab was an open access fishery, so easy to move into. It's now a substantially larger fishery and a real market has developed. I think an important question in the Gulf of Maine is, what options would there be if lobster were to collapse? Another question from a different scale is, how do we adapt our ocean regulations, sort of top down, especially because many ocean uses, including wind energy that's growing rapidly in the Northeast, cannot be adjusted anywhere near as fast as fishing behavior can, and as we've seen, fishing behavior has been. So can we adapt ocean planning to the impacts of climate change? So we've tested this by basically putting together a hypothetical ocean plan. So we set goals that included fishing areas, areas for conservation, areas for wind or wave energy development. And then we let the computer optimize a plan to meet those goals based on where species and wind energy and wave energy are available now. And what we see is that a plan built around the present conditions will be less likely to meet planning goals in the future as species shift their distributions. This graph shows that decline. We've got year on the x-axis, fraction of fishing and conservation goals met on the y-axis. We've done this not just for the Northeast US, but 9 regions around North America, and we see very congruent patterns in each region. But what if we step back and intentionally plan for the future? So we can do that by including projections of where species may be by the end of the century in that plan optimization process. And I will say we're using one set of projections for planning and a different set of projections for testing, so to avoid circularity. And what we see is across all regions, these proactive plans shown here in purple are consistently better at meeting ocean planning goals through time. And that's largely because it leads to plans that spread fishing and conservation areas out as networks so that species can hopscotch between them as they shift their distributions. We're now working to design tools for doing this throughout the blue economy, in part in collaboration with the Nature Conservancy and the University of Connecticut and National Marine Fisheries Service and NOAA, as part of a National Science Foundation Convergence Accelerator project we're calling Future Blue, but that's just started now. So just in summary, I showed you some physiological data suggesting that marine animals are more sensitive to warming than our animals on land, that shifting to new locations is a key mechanism that populations use to cope with changing climate, even more so actually than on land. And finally, that planning ahead for species on the move is important for maintaining fisheries and ocean ecosystems. So I've got many people to thank, including a wonderful group of collaborators on the projects I presented today, the students and postdocs in my group, and the organizations that have supported our work over the years. Thanks so much.
Great, thank you very much, Malin. That was fantastic. As a reminder, everybody, please feel free to type your questions into the Q&A box for our presenters. So we don't have any yet, Malin, but I do have a question for you. So over the past couple of days, this question has been coming up. Is the Gulf of Maine changing too rapidly for us to study that? And I would like to take that a step further with what you presented when it comes to ocean planning. Do you think even taking a proactive approach with the rate of change that we're seeing, if we can still get there and understand the system before it changes so much?
So I don't think it's changing too fast to study. I mean, I think that's maybe you're asking the wrong person, but the change actually provides, I think, an amazing opportunity and a really important opportunity for us to learn how these systems work, right? You know, we can't think in terms of equilibria, but really in terms of these transient dynamics and how do species respond to this change. It's a natural experiment, even if it's one that we wish wasn't happening. And in terms of adaptation, I think that's in part why it's so important for us to think ahead. You know, as humans, we base our decisions largely on our experience. A lot of that is historical experience. But I think, you know, we're at the point where we have a wonderful set of tools for looking forward with uncertainty, yes, but despite that uncertainty, we're quite confident in the direction that things are going. And I think that actually provides a lot of ability to make decisions based on the conditions that are coming. You know, in many cases, it's not so much a question of what's the magnitude of what's coming, but when is it going to happen? And designing our systems to be resilient to those changes going forward. I think that's really important.
Great.
Looks like I do see some questions coming in too.
Yeah, so we have a question from Susie. Hey, Malin, great talk. Can you please share the Rutgers link that was up on the slide showing the changing distribution of American lobsters, or show that slide again? So I'll let you kind of do that. But we do have another question. So that wasn't really a question, but Ben Gutzler does have a question about how much of the leading trailing edge of distribution shifts is driven by individual movement versus larval dispersal?
Yeah. Yes, that is a really interesting question. And so I don't have strong data differentiating between the two. However, based on the species that we're looking at and the speed at which this is happening, so I think it's largely individual movement. Because of adults and possibly juveniles. You know, if it's larval dispersal, it takes a few years, in effect, a generation to disperse and then grow. But if it's adult movement, then these responses can happen even within a year or across a couple of years. You know, I'm thinking, you know, Kathy, for example, and others have talked about black sea bass showing up in the Gulf of Maine at the same time that the marine heat wave in 2012 was happening. And I think that's a good example of the really rapid responses that can happen with individual movement. On the other hand, for some species, kelps being one, it has to be a propagule dispersal process. So that will be slower. And I think that those differences across species are going to be really interesting to tease apart. And that's part of why we're moving towards these mechanistic models that include these demographic processes explicitly.
Great.
That's a great question.
Well, thank you very much. I'm sorry, we're going to switch gears.
Fine.
That was an excellent opening. So now we're going to turn to a joint presentation from Andrew Good, Josh Carloni, and Dr. Burton Shank. So they're going to be talking about the consequences of climate-driven shifts in phenology for American lobsters, links between lobster biology, zooplankton dynamics, and oceanographic processes in the Gulf of Maine. So Andy, take it away.
I'm Andrew Good with the University of Maine, and I'm going to start our series of talks that all have a common thread, and that's how changes in larval dynamics affect spawn and recruit demography of the American lobster. And my part of this story investigates climate impacts to egg hatch phenology. Is it going to the next slide?
No, we don't see it advancing.
All right. There it is. All right. So we collaborated with Maine Department of Marine Resources, Massachusetts Division of Marine Fisheries, and Rhode Island Department of Environmental Management to evaluate the timing of egg hatching using their ventless trap surveys. By using the egg stage codes of female lobsters caught in these ventless traps, we were able to reconstruct annual time series of the proportion of females with hatching eggs, an example of which you can see below. We then fit Gaussian probability distribution curves to these data to determine 1, the date of peak egg hatch, 2, the duration of hatch, and 3, the onset of hatch, which you can identify as the red line. These patterns across statistical areas were then used to determine how patterns of hatch phenology correspond to regional oceanographic conditions. We found that patterns of hatch phenology are tightly coupled and correspond well with patterns of seafloor temperature, whereby warmer seafloor temperatures correspond to earlier hatch over shorter durations. And to contextualize these patterns, a 1 degree Celsius increase in seafloor temperature during the entire embryonic development period, which goes from roughly October to July, corresponds to a 47-day earlier hatch over a 16-day shorter period. We then wanted to take this concept of thermal regulation of hatch behavior more in depth. To do so, we used modeled seafloor temperatures to determine the cumulative degree days necessary to reach the onset of hatch. We were able to identify a degree day threshold to initiate hatching that corresponds well with other derived thresholds from more controlled experiments. And the high correspondence between these thresholds suggests that thermal control on embryonic development of lobster is robust and is a metric that can be used to evaluate climate influence over long timescales. Based on that, we used modeled ocean conditions to estimate the onset of hatch over the Northwest Atlantic continental shelf and used generalized additive models to investigate the processes driving how these patterns change over time. We chose to do this over 2 major time periods, the one being the entire model time series, which goes from 1978 to 2016, and then the most recently modeled decade, so from 2007 to 2016. And we can easily identify some interesting patterns from the results from this experiment. Over long time scales, there was a shift in hatch towards later in the year, which is identified by the red colors in the left-hand figure. And that was due to cooling benthic conditions along the Scotian shelf that was being transported within the Gulf of Maine. However, changing ocean circulation patterns have increased the intrusion of warm Gulf Stream water within the Gulf of Maine and weakened the southward transport of the cold Labrador Current. Over the last decade, this has translated into a ubiquitous shift in hatch phenology earlier in the year. And additionally, this rapidly warming environment has translated into rapid changes in hatch timing, which ranges somewhere between 2 and 4 weeks earlier over the last decade, depending on location. So which processes are driving changes in hatch phenology? Well, our optimal generalized additive models were able to explain somewhere between 48 and 61% of the observed patterns and identify the seasonal contributions to the timing of onset of hatch. During both time periods, change in fall bottom temperature explained most of the observed patterns, but something interesting popped up. Changing winter temperatures over the last decade became nearly as important as the fall in explaining changes in hatch timing. So why is that? We believe that the increased significance of winter temperatures is driven by thermal influence on lobster embryogenesis. Below 4 degrees Celsius, embryonic development of lobster stops, and this period can last somewhere between 3 and 4 months during the winter. But rapidly warming benthic conditions, especially during the winter, change the availability of winter habitat above this critical developmental thermal threshold. By increasing the area and time spent above 4 degrees Celsius in the Gulf of Maine and Scotian Shelf, there was a rapid increase in the importance of winter temperatures to the overall embryonic development of lobster, contributing to earlier hatching. These patterns highlight the need to consider environmental change relative to critical biological thresholds that regulate biology and behavior. And it also suggests that small changes can translate into large biological responses. In combination with climate-induced shifts in biomass, these changes in hatch phenology have significant implications to the connectivity, dispersal, and phenology of lobster larvae. And that's going to tie in well with the next talk, which is going to be by Josh Carlone. I'll transfer that over to Josh.
Thanks, Andy. Go ahead, Josh.
Thanks, Andy. Share screen, go. Presentation. Can you guys see that?
Yep, you're good to go.
Okay, thank you. So yeah, I'm Josh Carlone with New Hampshire Fish and Game. And today I'm going to talk to you about changing phenology of lobster larvae, as well as Calanus phenologicus along the coast of New Hampshire. And is there any evidence of mismatch of these two species? This builds off a hypothesis that came out of a paper we published in 2018 that showed a strong correlation between Calanus phenologicus and lobster post larvae, as well as young of the year recruitment throughout the Western Gulf of Maine. So this kind of builds on, or is in agreement with what Andrew saw, but this is much simpler. So what this is from New Hampshire sea sampling, and it shows that the proportion of eggs hatching in the month of June has increased over this 20 some odd year period. So later in the year, you can see the proportion has increased. There's a significant upward trend there. And if you go to the next figure down, this is first appearance of stage 1 lobster larvae from the Norman doe Newstone toes over a 30-year time period. And you can see there's a significant trend downward. And so this makes sense. Eggs are hatching sooner and stage 1 are showing up earlier. And both of these correlate well with bottom water temperatures, April through June in the Gulf of Maine, or in this area off the coast of New Hampshire. A little more on stage 1. This is last appearance. You can see there's an upward trend here. And then I'm going to bring your eye down to season length. And you can see there's a significant upward trend here. So not only are they showing up earlier, they appear to be spending more time in the water column as time goes on. So now looking at Calanus phenologicus, the beginning of the season here was determined as the 25th percentile. So when abundance reaches the 25th percentile, and you can see over that 30-year time series, there's no trend. That varies without trend over that time period. Moving down to the end of the season here, there's a significant downward trend. And I'll bring your eye to this time period here, which is really from 2010 forward, where the season is ending earlier. And keep this figure in mind, because I'm going to bring that up in a couple slides from here. And then here's season duration, which is showing the same thing, but really you see this truncation of the season, Calanus finmarchicus at the end. And I should mention the end of the season was determined as the 75th percentile. But what does this mean? So you have changing phenology of both lobster larvae as well as Calanus finmarchicus. So what does this mean for what time periods might be important for lobster larvae? So what you have here is it's building off the correlation that we found in our 2018 paper, but in the 2018, we looked at all these months combined and found a correlation: high years of abundance of Calanus, high years of abundance of post-larval lobster. Here it's broken down by month. So you can see what comes back as the significant months are July and August. So if you take that information and we go back to this end of the season that I had already showed you of Calanus finmarchicus, and you draw this line, these are time periods. So these are every 2 weeks. And you draw this line at the beginning of July, you see that earlier in this time series, the season was ending around July. And in this period where we had high recruitment in the Gulf of Maine, well after July in some cases. But again, bringing us to this 2010 period forward, there appears to be the season is ending in some cases well before July hits. But I wanted to look at this, how these two kind of coincide in the water column at the same time. So what I did is I took a mismatch index from Berth et al. 2012 paper and modified it to fit lobster larvae. And so what this is it's the difference between peak stage 1 abundance and the end of the Calanus finmarchicus season. And we use peak stage 1 because that's really the time period where these guys are going to need a lot of food to metamorphose to stage 2 and so on and so forth, and finally get to the post-larval stage. So I'm going to bring your eye right here. This is 0. This is the mismatch index. These are positive numbers and these are negative numbers. So what this is it's these positive numbers would say that the peak stage 1, occurred before the end of the Calanus finmarchicus season. So peak stage 1 occurs and then Calanus finmarchicus season ends at some time period later. So again, this period in the mid-2000s, you see it's the Calanus season is ending after the peak stage 1. And then you see this red circle here. In some of these years, really, you have this negative, which means the Calanus finmarchicus season ended, and then peak stage 1 came some time period after that, suggesting that really in this end of the year here is more of a mismatch between these two species. And so this could be a case of both abundance and a mismatch between when these two species are overlapping in the water column. And so I think I've used up most of my time here, but here's just a summary that folks can read through. And I just wanted to mention that this was funded through the Maine Lobster Research Collaborative.
Thank you very much, Josh. That was great. Burton, you all ready to go? Burton, are you there? Okay. Burton, it is your turn to present. Andy and Josh, I have his slides. I don't know if for some reason there's a technical issue on his end, but
He did email us saying that sometimes his email his internet.
I think I'm back.
okay. Yeah, there you are. Go ahead,
Boy,
Burton.
that was well timed, wasn't it?
Thanks, Burton.
All right. Yes, I wanted to sort of continue talking about the connection between copepods and lobster settlement, but looking at sort of local versus basin scale correlations and how this connects to how this potentially connects to oceanographic processes. Okay, so this is sort of launching off of work that Josh did, where he was looking at relationships between Finmarchicus copepods, specifically to coastal New Hampshire, but young of year settlement in the Gulf of Maine, and finding stronger relationships for mid-coast Maine to the southwest than for down east Maine to the northeast. But this is based off sort of a locally observed process, being the plankton community, but linking to broad scale settlement dynamics. So we were curious how this Finmarchicus or the copepod trajectory compared to basin scale zooplankton dynamics. So we sort of pursued three questions with here. This connection between local and basin scale dynamics for copepods, whether the basin scale copepods dynamics match up with lobster settlement, and whether both of these processes potentially connect to a basin scale physical oceanographic process. Data sets going into this, the American Lobster Settlement Indice, or ALSI, which includes the study areas for the Gulf of Maine, the nymphs, ecomon, zooplankton sampling, in this case constrained to summer and fall, the Finmarchicus index for coastal New Hampshire from Normando, another Finmarchicus index for the Bay of Fundy, and the source water composition entering the Gulf of Maine through the Northeast Channel. And in general, I constructed annual time series for these. A lot of this data is sort of data poor and potentially noisy, so I then smoothed them in many cases with a Kalman filter to see if I could pull out a process underlying some noise, and then compared the raw and the smooth time series across the data sets. So looking at local versus basin scale zooplankton time series, the top time series is for New Hampshire from Normando Associates. The middle one is the basin scale ecomon survey for the entire Gulf of Maine. The bottom time series is the Bay of Fundy time series. And we have both the raw and this smooth time series, which is the black line running through this. And we see that the coastal New Hampshire time series compares very well with the basin scale index, particularly once you smooth them and pull out the underlying process. Both these indices climb to a time series high around 2005 and are dropping to or at or near time series low now. In contrast, the Bay of Fundy time series ramps up steeply and more recently, followed by a similar but less precipitous decline. For our second question of basin scale copepod dynamics matching up with lobster settlement, in this case, we took the smooth copepod index for the Gulf of Maine and correlated it against the raw trajectories for each individual study area. The top map is showing unlagged correlations, where the bottom map is the 1 year lag. So this is where copepods are matched up with the lobster settlement for the following year. And one of the things that we see is that these correlations, there's sort of a spatial pattern that falls out again here. Sort of mid-coast Maine to the southwest correlates very well with what's going on in the basin scale. But to the northeast, there's a weaker correlation there. So based off this, we sort of split the ALSI data into two sets, a northern set of study areas and southern, and produced separate time series for those. And that's what's shown here in the bottom right. And we can see a real difference between the trajectory for the northern study areas and what we're seeing for southern. Looking at these a little bit more in depth. So across the top is a comparison between the southern study areas and lobster settlement with the basin scale copepod index. So on the left is both of the time series plotted on top of each other. On the right is the time series plotted against each other, with the numbers representing the years. And what we see is there's sort of a common relationship between the southern areas and the copepod densities at the basin scale that's consistent through the time period. What's interesting is looking at the center set of plots, which is the northern study areas against the basin scale plankton dynamics. And we see that it's generally uncorrelated up through about 2004. And then it sort of shifts dramatically and sort of snaps into a relationship that remains strong through the remaining years. We do not see this sort of dynamic when we compare the northern study areas with the Bay of Fundy columnist series, which is the bottom set of plots there. So it fits marginally better, and it doesn't show this sort of shift in relationship between the two. So for our last question is, can we connect this to sort of like a basin scale physical oceanographic process? And here we were really interested in sort of a question of what's the water that's feeding the Gulf of Maine, and is that driving things, basically everything downstream. What we found was convenient was a Labrador slope water index. And so this is the proportion of water entering into the Northeast Channel, there north of Georges Bank, that can be attributed to Labrador slope water based on chemistry and physical properties. And what we get is a trajectory that we see sort of here in the top right. And if we look at the correlations that we show here across the bottom, we see that the slope water index correlates well with the southern areas for lobster settlement, with the Ecomon copepod index, the New Hampshire copepod index, but less so for the Bay of Fundy copepod index. So our conclusions, we did find a good correlation between New Hampshire local scale and the basin scale copepod time series. So this suggests that we can do some coastal sampling and understand potentially what's going on more at a basin scale. These basin scale copepod dynamics are closely correlated with lobster settlement, particularly for mid-coast Maine to the southwest, less so for down east Maine, Bay of Fundy, and western Nova Scotia. And both lobster settlement and the copepod trajectories seem to be tracking well the water composition entering the Gulf of Maine through the Northeast Channel. Again, less so for the Northeastern Gulf of Maine regions and lobster settlement. And I will stop there and open things up for questions.
Thanks, Burton, and thanks, Alex and Josh, or Andy, sorry. Unfortunately, we don't have time for any questions, but please, everybody, put your questions in the Q&A box or the chat for our presenters. So now we're going to switch gears a little bit and have a joint presentation from Alex Asher, Emily Patrick, and Molly Spencer talking about lobster larval feeding rates, prey preferences, and potential food limitation mediated by a changing North Atlantic food web. So when you all are ready, please take it away.
Does that look, everyone see that okay?
Looks great.
All right, go ahead, Emily.
All right, good morning, everyone. I'm Emily Patrick, and today Alex Asher, Molly Spencer, and I will be presenting on larval lobster prey preferences, feeding rates, and potential food limitation. Next slide. So over the past couple of decades, we've been seeing a significant increase in lobster landings, which is shown in the figure on the left, and the quantity of egg-bearing females. However, you can see in the time series on the right that during the same time frame, there's been a decrease in larval recruitment, suggesting a breakdown in the normal stock recruit relationship. And this is referred to as the Great Disconnect. Understanding the cause of this disconnect is really important for the prediction of future lobster stock. And one potential cause is suggested by a recent paper by Carloni et al. 2018. Next slide. Although there's a strong relationship between the abundance of egg-bearing females and early-stage lobster larvae, there's no significant correlation to late-stage post-larvae. And however, post-larval abundance did correlate significantly with the abundance of Calanus binmarchicus, which is a foundational copepod species in the Gulf of Maine. Due to its high energy density, Calanus may serve as an important food source for lobster larvae or as an indicator of ecosystem health due to its importance in the Gulf of Maine ecosystem. And in order to determine this distinction, we can examine whether lobster larvae actually show preference for Calanus over other potential food sources in a lab environment. Next slide. So to do this, I completed controlled grazing experiments by isolating one lobster larvae in a 1 liter jar with equal amounts of copepod species that are found in the Gulf of Maine. So Acartia, Calanus binmarchicus, and Temera. Next slide. So after the 24-hour feeding period, I found that all larval lobster stages consumed more Calanus than Acartia and Temera, and in particular, the stage 4 larvae consumed the most Calanus and copepods in total. Next slide. Also, just to know, I had 4 control jars without lobster larvae in them, just to determine how much natural die-off occurred over the course of 24 hours. Next slide. And finally, I ran a 2-way ANOVA test that modeled prey consumed as a function of prey species, larval lobster stage, and the interaction between these 2 main effects. And as you can see from the indicated p-values, the prey species, larval stage, and the interaction between these 2 had a significant impact on the total prey consumed by larval lobsters across all trials. Next slide.
And so for results from that experiment, we were able to check in on the actual ingestion rates of this preferred prey species. And so for our experimental grazing methodologies, we investigated the rates of both early stage lobster larvae, stages 1 through 3, and then stage 4 post larvae on Calanus phenologicus to draw some inference on the capabilities of each stage that readily consume and search for this prey item, and then relate it back to their natural environment. And so the concentration of Calanus we used in this experiment were prepared in altering volumes of water, seen on this slide here as our 1 liter mason jar all the way up to our 20 liter bucket, to create various dense environments to introduce to the larvae. And then each experimental density had controls administered as well to standardize our mortality rate of Calanus. Next slide. And so for results, we have prey concentrations for the number of Calanus per liter on the x-axis here, and then the number of Calanus ingested per hour on the y-axis. And so what we can see in this feeding rate graph is stage 4 post larvae, depicted in the yellow data points, eat roughly between 2.5 to 4.5 Calanus per hour, reaching a max ingestion rate at around 5 prey per liter. And then for stages 1 through 3, the ingestion rates per hour was consistently less than 1 prey item consumed. And could you go into the next slide? And so this is just showing a close-up view of those stages 1 through 3 ingestion rates on the same graph to show the not really significant difference between their ingestion rates here. And so the key takeaways from this ingestion rate experiment was that all larval stages were capable and readily ingested Calanus, despite being 3/4 of the size of our stage 1 larvae. There also seems to be an important transition from stage 3 to post larvae, as stages 1 through 3 have roughly similar ingestion rates compared to the large increase in prey ingestion observed in post larvae. And then finally, post larvae searching capabilities compared to the stages 1 through 3 seem to never be limited. And even our most spray parse concentrations, it didn't seem like they were limited in terms of being able to be satiated from this prey item. And so this potentially exhibits how much more capable this stage of larvae are at exploiting their environmental resources for survival. And so this draws a sharp contrast to stages 1 through 3, which display much lower ingestion rates, even at double the prey concentration we see in nature. And so the earlier stages of lobster larvae are thus possibly in a higher state of vulnerability concerning food limitation compared to their post larvae. And then next slide.
So my work focuses a little bit more broadly on understanding how important food limitation is in general to mediate lobster larval survivorship. And that starts with understanding what the diet of lobster larvae are, which just represents a fundamental knowledge gap for us. It helps us to assess risk of food limitation, and can also help to get at that question of how important Calanus is in the field. The next part of that is to understand how sensitive lobster larvae are to starvation. This helps us understand what their food requirements are in the field to progress their larval development. So more sensitivity means likely they have higher food requirements, versus lower sensitivity would mean lesser food requirements. And then additionally, we can wonder a little bit more about which stage in particular is more or less sensitive to starvation. We also did fatty acid composition analysis, comparing field caught post larvae to lab reared, fed, and starved larvae in order to produce a benchmark. And that gives us a bit of a direct test of nutritional status of post larvae in the field. So there we go. this is some of my diet data. This is procured through microscopy and visual identification of gut contents. This is just for stage 1 lobster larvae right now. The y-axis is how many larval guts a particular food item appeared in. And what you can see is basically the right side of this graph isn't super filled out right now. And that's because the stage 1 lobster larvae are small, they're difficult to work with, and it's difficult to visually identify down to a finer taxonomic scale with those larvae. And so we're also conducting a metabarcoding eDNA analysis at the same time, which we should have results from pretty soon, which will hopefully fill out this right side. We can see that copepods feature prominently in their diet, though we haven't yet seen evidence of calanus predation in the field by stage 1s. Here's my data for the diet of post-larval lobsters. You can see this is a bit more filled in. That is one, because there are a couple more larvae represented here, but also they're a little bit easier to dissect. And the main features of this graph and their diet, what we can see is that their diet features prominently many different crustaceans and largely copepods. So these are 2 copepods found in the guts of lobster post-larvae, and malacostracans, particularly decapod larvae. So over here on the right side, that is the telson of a crab, zoea, and then in the bottom right is the leg of a crab, megalopa. Here we see our first instance of calanus found in the gut of a field-caught post-larval lobster. There is only one time that we've seen that out of it, 65 different guts so far, and that could be interpreted as they're not in fact that important calanus to the diet of field-caught post-larvae, or it could reflect a difficulty of abundance of calanus in the field. So they're just less likely to be able to predate upon them. Next, we move on to the question of starvation sensitivity. So how sensitive are they actually to starvation? We did this by taking 90 larvae of each stage and separating them into starvation chambers and doing daily surveys to see how many were surviving. So the y-axis is proportion still surviving of different stages of lobster larvae. The x-axis is day. And what we see, what the important thing to notice is that the stage 1s and stage 2s, those early larvae, have the same sort of precipitous downwards trajectory, indicating larger food requirements proportionally and greater sensitivity to starvation when you compare them to the stage 3s in this blue line, and especially the stage 4s, which seem to be able to last quite a long time, even under starvation conditions, with one of them lasting over a month while being starved. So finally, we have our fatty acid analysis. So what this graph is showing is for post-larval lobsters only. We have earlier stages sort of in the works right now to get this data out. The y-axis is showing % of their dry weight, which could be attributed to lipids, so energy stores. And the x-axis, we have 3 different treatments. So the fed and the starved are our benchmarks. So those refer to lab-reared post-larvae, either that were well-fed or starved. And then we can compare the lipid content of field-caught post-larvae in the center to those benchmarks and see how favorably they compare. And what we see is they compare very favorably to the lipid content of well-fed larvae from our lab benchmark test, meaning that the post-larvae that we find in the field, we expect them not to be food limited, which kind of follows from the previous talks where we saw that the post-larvae were much more capable at handling and feeding upon calanus, and also that they're not so sensitive to starvation. So the conclusion here is that first we saw that correlative analysis from earlier and also the earlier talk that suggests that calanus and marchicus may be important either as a food source or potentially just as a indicator of ecosystem health or available zooplankton abundance for larvae to ingest. And that laboratory studies found that given a choice of 3 different copepods, lobster larvae at each stage ate a higher abundance of calanus than these other 2 copepod choices, and that the stage 4 post larvae are in fact much more adept at handling and consuming those calanus than the other stages. And what follows from that is that we didn't see evidence of calanus predation in the early stage 1 larvae, but we do see evidence that post larvae in the field are eating calanus. Albeit only a little bit right now, but hopefully more data will flesh that out. And also that we see that those stage 1s and 2s, those early larvae, are very sensitive to starvation when you compare them to the later stage, especially the post-larvae, which we see from our field test of nutritional status, that indeed the post-larvae are not food limited. And so that food limitation is most likely to occur in the early stages, particularly stage 1 and stage 2. And with that, I'd like to acknowledge our home institutions, Bigelow and Maine, and our funding sources. And if we have time, we'll take any questions.
Great, thank you all. That was really fantastic. And I'll also make a plug for an additional component of your guys' research. Evie Leyland is going to have a poster later today too. So we do have one question in the chat that we'll do really quickly. When stage 1 or 2 are starved to half dead and then allowed to develop, does it have an effect on stage 3 and stage 4 development and or the ability to survive themselves?
So there's actually, I think this is, there is a point of no return for the larvae. So after the initial larval stages starve to a certain degree, no matter how much you feed them, they are not going to bounce back and develop. So that graph I showed, the stage 1 through stage 4 portion surviving while starving, those were not the same larvae. We had to raise larvae up through each of the stages and then take stage 1s as they developed and then stage 2s, 3s, and 4s. So I do not know whether starvation has a later effect on development. I do know that past a certain point, if they have been starved to a certain degree, they won't develop at all.
Great, thank you. And thank you all again. So now we're gonna switch gears a little bit and have a presentation from Jess Waller discussing decreasing size at maturity in female lobsters from coastal Maine waters over recent decades. So go ahead, Jess, when you're ready.
Okay, good morning, everyone. So I'm gonna take things back to kind of earlier in the life history of lobster and talk about mature females and egg development. So I'm going to be giving a very brief update from our research in the DMR Lobster Lab. I'm a lobster research biologist in our West Boothbay Harbor office to quantify the carapace length at which females become capable of sexual maturity. So while I'm speaking today, I want to acknowledge that everything I'm going to show you is the result of a team of hardworking people, including my fellow lobster biologist, Kathleen Reardon, everyone else on our DMR team here, our collaborators at Dalhousie University, as well as our research partners in the Maine lobster industry who allowed us to sample lobsters from their vessels and also provided valuable observations that really informed some of the methods of our research. I also want to thank our funding sources, of course, the Maine Lobster Research Education and Development Fund, as well as the Sea Grant American Lobster Research Program. So we brought together this big team of people that you see listed here to begin to study the size at maturity of female lobsters in coastal Maine waters. Size at maturity is an important life history parameter in the Atlantic States Marine Fisheries Commission American Lobster Stock Assessment and the supporting analyses for that model. Size at maturity can be used to calculate spawning stock biomass, which tells us about the reproductive potential of a stock, of the females in a stock rather, as well as the growth patterns of female lobsters. It's assumed that females will molt annually, pretty consistently before maturity, and then at maturity, they switch into a biennial cycle of reproduction and growth. So it's important for us to understand at what size that transition and growth patterns occurs. As of the 2015 stock assessment and that report, the best available data sets to study female size at maturity in coastal Gulf of Maine waters came from a DMR study conducted in the 1990s that focused on female size at maturity, collecting females in Boothbay Harbor, Maine, and Sorrento, Maine, which you can see highlighted on the map from that study on your right. There was also a study done in the 1960s looking at size at maturity just in Boothbay Harbor. So those were the two relatively small existing data sets. So the lack of more recent or broad scale data was noted in the 2015 assessment report and elsewhere, since there's been a well-documented inverse relationship between size at maturity and temperature, with females in warmer areas reaching maturity at relatively small sizes compared to females from other areas. And as we've heard a lot about, even just this week and this morning, the Gulf of Maine has changed a lot since the 90s and over the scale of recent decades, including significant rates of warming. So that means that a data set from the mid-90s is no longer an accurate representation, or is likely no longer an accurate representation, of the reproductive development of female lobsters in the Gulf of Maine today. So for all of those reasons, size at maturity was listed as a high research priority and a data gap in the 2015 assessment report. And so we decided to try to address that data gap and update these data sets. And we came at this work really from 3 different questions. So first, just to orient us, the map at the bottom of your slide is of the Maine coast and the 3 federal statistical areas that break up the coast. So we really wanted to start in our own backyard because there's been 2 size and maturity studies done in that location in the past. So we wanted to start by looking at how things have changed in Booth Bay Harbor in terms of female size at maturity to get a sense of how things have changed over time. And then we wanted to expand our work looking at female lobsters across the Maine coast in each statistical area. And now that we've completed that work, we are starting to think about other ways that we can do this kind of research in the future. So to begin to answer all 3 of these questions, we had to collect a lot of female lobsters from different ports along the coast. So that's the same map that we just looked at, and the 2 sampling locations from DMR's historical work, Booth Bay Harbor and Sorrento, are noted there. And then each shape represents an average waypoint from our lobster collection trips. And the shape also denotes the year that those collections took place. So our samplers would go out on commercial lobster boats, and they would get as many female lobsters, non-ovigerous female lobsters, as they could for us. Sampling is a very important consideration in this kind of research. And so we looked at temperature measurements, we looked at our sea sampling measurements, we also talked to our lobstermen for very local up-to-date observations, trying to determine when the best time period would be to sample, ideally to be collecting females either right before egg extrusion would occur or right before molting would occur. So we catch them at very distinctive points in their reproductive development. So for each area, that ended up being in points in May and June, and our samplers would be collecting females from 50 to 120 millimeters in carapace length. To really best represent the whole population of the females in each area, we collected both V-notched and non-notched females. And in total, over that 3-year period, from across the coast, we collected and analyzed 1,237 female lobsters. So after each trip, our samplers would come back to our office in Booth Bay Harbor, where we would start the lab component of these analyses. We would start by assigning an ID number, which would follow it through the process. And then we took a series of measurements that would try to tell us about their reproductive development as well as their growth, since those processes are so interconnected. We wanted to learn as much as we could about both for each female. So we took a series of external measurements first, and then we also took a hemolymph sample, which we preserved. After that, we'd remove the carapace to begin to characterize the condition of the ovaries. And I apologize if this is not what you wanted to see before lunch. I'm going to go quickly. On the left, that's a female with immature, underdeveloped ovaries. And on the right, just to give you a sense of the kind of things we're looking for, that's a female with mature, well-developed ovaries. You can begin to see the individual oocytes in the lobes of the ovaries as well. So we would photograph them, make some other notes, weigh the ovary, and then we would remove oocytes from the center of the ovary to be photographed under the microscope. So that's what we have on our left here. We would photograph the oocytes to record them, but also look at their diameter as well as some other characteristics. We would also remove a pleopod from the female and look at that under the microscope. So the pleopods, sometimes called the swimmerettes, the appendages that hang under the tail, because we can actually get a lot of valuable information from the pleopod as to whether the female is more likely to extrude eggs or more likely to molt in the coming weeks if either of those processes are imminent. So in the center image there, all the gray dots that you see, those are cement glands. Those appear just before egg extrusion. So that's an indication that that female was preparing to spawn. And then on the right, that's really the zoomed in kind of top third of a pleopod. That orange band you see there is newly developed setae within the pleopod. So if we see that, that's an indication that the female is preparing to molt. So we put all of that information together for each female to be able to perform ovary staging. This is the maturity determination method that's been widely used across the American lobster range, but also was the one recommended by the ASMFC American Lobster Technical Committee. So this method lets you make a determination of immature or mature based on the criteria of all the measurements that you see here. The color of the ovary, the average oocyte diameter, the relative weight, as well as indications of previous spawning events. So we did all of that for each female, and we were also able to kind of validate those determinations based on the information we just discussed from the pleopod, cement gland stage or cetogenic stage. So we did all of that, and then we were able, in each statistical area, to calculate the proportion of mature females by carapace length. So now that we're going to look at our results, let's go back to our first question about what was happening in Boothbay Harbor. So on your right, that plot is proportion mature by carapace length, and each line represents the maturity ogive, or the predicted proportion mature based on our observations and maturity determinations for each DMR sampling period in the Boothbay Harbor area. And the dashed line is our most recent effort. So as you can see there, in our analyses confirmed, females sampled in 2018 were reaching maturity at smaller sizes compared to females in the 1990s and the 1960s. So another way we can look at changes over time is looking at the length at 50% maturity, the L50 value. So that's what we have for each of the studies there. So we can see over a 50-year period, there has been a 10 millimeter downward shift in L50 in females in this area, with roughly 5 millimeters in between each DMR sampling period. And while there are likely many factors that work in concert to influence size at maturity, since there is a well-established relationship with temperature, we looked at the DMR environmental monitoring program and the sea surface temperatures recorded there, since we have a record going back to 1905, to look at sea surface temperature trends from 1960 to 2018. And as we might expect, we saw significant warming in the Boothbay Harbor area over that period. So now let's look at what's happened in each statistical area in a bit more detail. So these are the same kind of plots with the proportion mature by carapace length. The solid line is still the ogive for each statistical area, moving west to east across your screen. The dashed lines are the confidence intervals. The open circles are the predicted, observed proportion mature at each 5 millimeter size bin, so grouping females of similar sizes. The tick marks running along the bottom of the plot are individual females that we deemed immature, and at the top of the plot, individual females that we determined were mature, just so you get a sense of things across the size range of females that we collected. We also have our calculated L50 value for each statistical area. So we see this latitudinal gradient in size at maturity, with the smallest females smallest L50 in 513, and then that size increasing as we moved eastward across the coast. So now let's look at how things might have changed in each of these areas over the last 25 years. So we have our sampling map, again, to just kind of reorient ourselves. And then in this table, L50 values from the DMR study done in the 90s, and then our work that we're talking about today. And I'll note that there's an asterisk next to the 511 L50 from the 90s work, and that's because there was technically there were no females sampled in 511 during this period of time. But looking at our map, we see that the sampling site in Sorrento is in quite an eastern area of 512. So we're using that 512 value there as a point of comparison. So looking over the past 25 years or so, we see that there's been a downward shift in L50 in each statistical area on the order of 4.8 to 6.7 millimeters carapace length. So really seeing that consistent decrease across our statistical areas. And I don't have time to talk about it today, I don't think. But I will note in our paper discussing this work earlier this year, we also pulled out some of our individual metrics of growth and maturity and looked at those at the level of the statistical area, evaluated some changes over time. And we also looked at warming trends in each statistical area as a function of degree days above 5 degrees Celsius, since that's a threshold temperature for ovary development. So now that brings us to our present day efforts, which is thinking about some new and hopefully easy and cost-effective ways to do this kind of work moving forward. So as conditions in the Gulf of Maine continue to change, and as the Maine lobster fishery continues to change, we want to be able to update these life history parameters easily to be able to quantify potential shifts. And the current method, the way we have been doing things is quite time and resource intensive. It involves learning and executing many different techniques, as well as collecting a large number of lobsters. So in 2020, we put in a proposal to the Sea Grant American Lobster Research Program that would allow us to leverage the biological samples we've collected over the last 3 years, as well as the many thousands of images that we have, to try to develop and test newer, faster, and hopefully easier ways to get these kind of results, including image-based guides for some of the processes that we've talked about here. And I'm hoping that I want to make these very publicly available, very understandable. So I'm hoping to put together an advisory committee to help me develop some of those products next year. So if you're interested in joining me in that work, please send me an email, or I'll be reaching out to many of you, I'm sure. We're also working with Dalhousie University to look at the hemolymph samples that we've collected over the last 3 years in the hopes that we can quantify the concentration of vitellogenin in each sample. So vitellogenin being the egg yolk precursor protein. So we're hoping to link those concentrations to the ovary stage, the stage of development that we just assigned each female, and develop a method that would let us, hopefully on a lobster boat someday, go take a hemolymph sample, bring that back to the lab, and instantly learn something about the ovary stage or reproductive development of that female. So just really to wrap things up, we saw consistent decreases in the size and maturity in Booth Bay Harbor. We saw a latitudinal gradient moving west to east across the coast. There was also decreases over the last 25 years. We've provided all of these data to the American Lobster Technical Committee, and aspects of it were incorporated into the 2020 analyses as part of the stock assessment. These data are also now available for any considerations around management and conservation measures. And we hope to develop some instructional materials and new ways to do this work moving forward. So with that, I'd like to thank some others who helped us along the way, and thanks to RARM for having us today.
Great, thanks so much, Jess. You do have one question in the Q&A, but unfortunately we don't have time to do that live, so please take a look over there. All right, so switching gears again, we're going to be her presentation is assessing climate resilience in Maine's lobster fishery. So Kathy, you want to give your slides a try?
Okay, are you?
I see your slides, but not in the presentation mode. There you go.
How's that?
Perfect.
Okay. Well, thank you all for inviting me to come and present today, and for the great number of participants that have shown up for this session. I am going to be switching gears quite a bit in this talk. So we're going to go from thinking about lobster biology and lobsters in the ecosystem to really thinking about the lobster fishery in Maine as a social ecological system. And I'm going to be sharing some work that we are doing around trying to understand climate resilience in Maine's lobster fishery. I want to acknowledge Kat Maltby, who's a postdoc with me and a co-author on this work. So as many of you are familiar with, American lobster is an incredibly important fishery in the country, and particularly here in Maine. It's been the most valuable single species fishery in the US since 2004. And in Maine, it represents more than 80% of the landed value from our marine fisheries. And in many of our communities, there are just very strong social and cultural ties to the lobster fishery. We also know that the Gulf of Maine is changing rapidly, and that these changes have implications for the fishery. So some of the changes that we are experiencing are related to warming and patterns, temperature patterns over the course of the year. So I think you're all familiar with the fact that the Gulf of Maine has been warming incredibly rapidly relative to other parts of the world's oceans. Over the observed time period since we've been collecting satellite sea surface temperature data, the rate of warming here in the Gulf of Maine has been about 3 times faster than the global average. And that's been exacerbated in the more recent decade to 15-year time period. We're also experiencing marine heat waves in this region. So I'm going to be referencing back to the 2012 heat wave for a portion of my talk. And during 2012, we saw a heat wave occur across the Northwest Atlantic that extended from Cape Hatteras down in North Carolina over to Iceland and all the way north into the Labrador Sea, with temperatures here in the Gulf of Maine being exceptionally warm. And this was actually the first event that we had applied the term heat wave to in an ocean context. But since that time, we've experienced subsequent heat waves here in the Gulf of Maine quite frequently in 2016, 2018, 2020, and again this year in 2021. We've been in what we would consider marine heat wave status for most of this year. So I'm going to talk a little bit about what we have seen in the Maine lobster fishery as we've experienced warming in this region, and then also some of the experiences associated with the 2012 heat wave as context for thinking about climate resilience. So as ocean temperatures have warmed here in the Northeast US, we've really seen quite different experiences across that whole region in terms of how lobster populations have been affected by that warming. In contrasting the southern New England experience versus the Gulf of Maine, we can see different temperature responses. So what I'm showing here in the figure on the right is some work that we've done developing a population model for American lobster and trying to use it to evaluate the influence of different environmental conditions and management measures on population outcomes. So what I'm really showing here in the gray dots are is the population estimate from the stock assessments. And we can see that there was substantial decline in the southern New England lobster population starting in the late 1990s, but steady increase in the Gulf of Maine population over that time. And I'm not going to go into great details around the modeling work, but basically what we did with this was to ask the question if temperatures in those regions had held at the long-term historical average versus changing as temperatures have warmed in these areas, what would the experience of the population have been? And what we found is that in the, sorry, I need to get rid of my Zoom box. In the Gulf of Maine, if we'd held at the average long-term temperature, we would have projected a lower population size. Whereas the actual temperatures we've experienced have supported enhanced productivity of the Gulf of Maine lobster population. In contrast, the opposite is true in southern New England, where the warming in recent years has contributed to the declines of that population as those temperatures have reached beyond sort of critical thresholds for lobsters. And the 2012 heat wave, it provided a really insightful experience for short-term impacts on the lobster fishery, as well as a really interesting contrast to some subsequent responses. So during 2012, when we were experiencing a marine heat wave, temperatures warmed up, or we came into 2012 with very warm ocean temperatures. And essentially, as we looked at the annual cycle play out over the course of the year, what that meant is that it felt like temperatures were warming up 3 to 4 weeks ahead of the typical schedule in the spring. And that spring warming is really important for lobsters because it causes them to move into the nearshore waters where they're accessible to the coastal fishing boats. They just generally become more active, so move into and out of traps more readily. And the really important thing is that it triggers molting. And molting into a harvestable size class is a very strong connection between the biology of the species and the fishery itself. About 80% of the harvest is from lobsters that have just molted into a legally harvestable size class. So as this warming occurred early in 2012, it also led to a very early increase in the landings that year. So if we look at the months of June and July in 2011 in blue versus 2012 in red, we see that in 2012, the fishery landed more than 15 million more pounds of lobster just in Maine in those 2 months than in the prior year. And the timing of these landings really set off a series of events in the supply chain where it just wasn't possible to move product as quickly as needed from the dock out to processing facilities. And the processing facilities at the time and still are mostly in Canada. So that also created an overlap with the Canadian fishing season. So we saw this disruption in the supply chain during 2012 that led ultimately to a price collapse, with prices bottoming out at some of the lowest levels ever seen during the early part of that summer. So my question is, what can we learn from these experiences about resilience in Maine's lobster fishery? And how can we use these experiences to better prepare for some of the changes that might be coming in the future, particularly those related to climate change? So I'm working with a SNAP working group, which stands for Science for Nature and People Partnership, on climate resilient fisheries. And in this working group, we define resilience quite broadly. So we are considering the capacity to prepare for, to resist, to cope with, recover from, or adapt to a given stressor. And the purpose of resilience is to ensure the sustainability of the marine ecosystem, fishery resources within that ecosystem, and the human benefits derived from fisheries. So with this definition of resilience in mind, I also want to highlight that we're really thinking about resilience in fisheries from a social ecological system perspective. So recognizing that fisheries are influenced by the ecological condition of the ecosystem within which they operate and the stocks that they pursue, and the social system where individual, community, and management context, as well as the broader economic context, all shape the capacity to adjust and to respond to change. So we're thinking about resilience across these 4 dimensions: ecological, economic, social, cultural, and governance and management. So with this group, we have developed a set of resilience attributes that we think are relevant to climate resilient fisheries. This work was done with a group of 23 scientists and practitioners who are part of the core working group. And those members come from 7 different countries. But we also have consulted with a broader set of advisors that increase the input quite substantially. So with this working group, we've identified attributes of resilience, both from expert consultation as well as from the literature, to think about resilience attributes that are relevant to fisheries, both or across the ecological, socioeconomic, governance dimensions. We identified 38 attributes, defined those with definitions we thought were relevant to fisheries, identified mechanisms by how they might confer climate resilience, and pulled examples of their application or relevance within the context of fisheries. So then we also organized those into domains that follow from Josh Sinner's work. This is published in Center 2018, and are now using those attributes to look at how they emerge in a set of case studies of fisheries from around the world. And I'm working on a case study for this effort that centers on Maine's lobster fishery. So this just shows you quickly, I'm not going to talk through this slide, but gives you a sense of what the attributes look like and how they fall out across the ecological, socioeconomic, and governance dimensions, and in different domains of those dimensions. This is coming out in Fish and Fisheries sometime in the next couple of weeks. So I'd be happy to come back to that and dig in further. But I want to really jump right into relating some of these to what we are seeing in Maine's lobster fishery. So in Maine's lobster fishery, one of the other things we used the model I spoke to earlier that focused on temperature, we also used it to look at the management measure that's been in place in Maine for decades and even a century now to protect spawning female lobsters and to protect large lobsters. So these measures we found contributed to an enhanced growth of the Gulf of Maine lobster population. So those industry-led conservation measures really protected or provided a buffer in the population and conferred resilience attributes associated with population abundance and age structure. We also have seen economic resilience in the Maine lobster fishery following the 2012 heat wave. There were adjustments made that indicated that the supply chain was better able to accommodate early influxes of landings that had been experienced in 2012. A very similar pattern played out in 2016, and the industry was able to avoid the price collapse that had occurred in 2012. And contributing to this were flexibility in the infrastructure, adjusting transportation plans and processing capacity, And also it's demonstrated a learning capacity to learn from past experiences and make changes that would provide resilience in the future. From a governance perspective, Maine's lobster fishery has a really unique co-management system where local lobster management councils provide input and work with the state agency, DMR, to address issues in the fishery, and also there's strong state coordination through the Atlantic States Marine Fisheries Commission. So this governance arrangement brings forward attributes that support resilience like participation, transparency, and cross-scale integration. And finally, social resilience emerges in the fishery through certain attributes associated with individuals having a resilience mindset and feeling capable of adjusting to change can be really important. And we hear this from a number of fishermen that they feel like they've always adjusted to changes in the system. And as long as they have the flexibility to continue making those adjustments, that they have the capacity to figure out how to respond to change. They also have access to knowledge, given many of the robust scientific programs we have in place in this region, as well as from a diversity of perspectives that come from participants in the industry themselves. And as we think about climate resilience, looking ahead, we do expect to see changes in the productivity and distribution of the Gulf of Maine lobster population. So many of the current resilience attributes that I just highlighted will remain important in the future, but also it will be important to maintain and strengthen conservation commitments to support the stock, to provide flexibility and adaptive capacity within the harvest sector as well as throughout the supply chain, and to have the ability to make forward-looking decisions or the adoption of a resilience mindset to be thinking about and planning for change. And as part of that, leadership and initiative to take on some of those forward-looking challenges will be really important. And as we think about shifts in species and changes in productivity, there will likely be a different set of species that will become potentially utilizable by commercial fisheries in the state of Maine. But we also have to acknowledge that climate change, the direct impacts of climate change are not the only changes that are being experienced. So indirectly, there are other changes associated with climate change and just broader cultural change occurring in Maine that affect the lobster fishery. So changes in ocean use patterns, we've seen growth of aquaculture in our state. And then now the development of wind energy is becoming really important throughout the region. And also we heard, Carla Gunther's talk yesterday, just talking about community change and some of the shifting demographics. These are all contributing to some of the considerations that need to come into perspective in terms of being resilient to future change, and requires thinking beyond just the fishery and thinking about integration across sectors. So I want to quickly wrap up by acknowledging the importance of the SNAP working group more broadly to this work and some key collaborators. And I look forward to continuing this work. As next steps, we will be putting similar case studies together and trying to derive lessons from fisheries around the world, and then also thinking about resilience assessment frameworks and tools that can be applied in practical sort of field settings to extend some of the lessons that we are drawing out into other uses. And so I'd love to have further conversation and input from anyone who's interested in thinking about these topics further, and want to acknowledge Sea Grant through the American Lobster Initiative for funding to support this work, as well as SNAP, and then some of the funding from NSF, NOAA, and Maine DMR that have really set the context for understanding some of the changes that are occurring in the fishery. So with that, thank you. I would be happy to address questions now or later.
Great. Thank you very much, Kathy. Unfortunately, we're right at the end of time. So thank you very much, and take a look at the Q&A, and there might be some questions in there. So continuing on the theme of socioeconomics, our final sort of longer presentation will be by Teresa Burnham, who's going to present Building Sentinel Indicators of Social Economic Resilience in the American Lobster Fishery. So go ahead, Teresa.
Thanks, Amelia. I'm going to share and then go to presenter view. Does that work?
We see your presenter view, not the actual slides.
Okay. Sorry. How about now?
Great, you're all set.
Okay. Problem is I'm not seeing my presenter view. Sorry, we did the check this morning to nope. I'm gonna try one more time.
If you want me to share your slides for you, or
That is okay. Okay, so I really appreciate you all having me. Sorry about that technical difficulty. I'm going to be sharing our work on building sentinel indicators of socioeconomic resilience in Maine's American lobster fishery. And all of the wonderful talks today have really highlighted for us the impacts on the biological component of the American lobster fishery. And we know that climate and fishing and life history all have important impacts on the biological status, health, and resilience of the fishery. However, the link to the socioeconomic component of the fishery is unclear. Or it's not direct, I should say. And the beginning of the COVID-19 pandemic and the 2012 heat wave that Dr. Mills just covered for us is a really good example of that, basically showcasing that even in times when the lobster population biologically is healthy, socioeconomically, it's sort of a different story. And so our project is interested in understanding similarly to how we understand the impacts, the indicators of health for the lobster population. We'd like to know what those indicators of health are for the socioeconomic component of the fishery. And this is really important from a policy perspective because this figure comes from Atlantic States Marine Fisheries Commission and shows how the reference abundance of lobsters has changed over time in the increasing black line. And the horizontal lines represent different abundance thresholds and limits and targets. And if we focus on this bright purple line here, which is a proposed abundance limit, this is basically saying that there have to be 3 years, an average of 3 years of the reference abundance going below this limit in order to trigger a management action. And while this may be appropriate for maintaining sustainability of the biological stock, it would clearly be devastating from a socioeconomic perspective for the lobster population to go so low. And so it's really important that we develop these indicators and then are able to incorporate them into management actions like this so that we can also maintain the health of the socioeconomic component of this important fishery. And so that leads to the main question of our project, which is what social and economic indicators can describe the status of the lobster fishery at a given time. And at the top of the screen here, you'll see a preview of the steps of the approach that we're going to take. And we'll start with the collaborative methods development piece. And so basically the first step in this whole process is to identify potential indicators of socioeconomic resilience in this fishery. And fortunately, NOAA has a really impressive body of work led by Lisa Colburn and Michael Jepson that has evaluated some social indicators of resilience in fishing communities. And there's a link there that I'll put in the chat at the end. This work at NOAA uses census data, which is limited in its temporal and spatial sensitivity. And so we've also included the local expertise of DMR and Maine Center for Coastal Fisheries, among others, to also try to identify potential indicators that may be specific to the Maine lobster fishery and may exist at more fine scale, time and space measurements. And after we identified some potential indicators using those two resources, we developed an in-depth interview protocol to understand the importance of the indicators we came up with to industry. And so we developed 25 quantitative questions that use either Likert scale, strongly agree to strongly disagree, or on a 0 to 10 scale questions. And we also developed 15 qualitative open-ended questions. And those are a really important component of this work because they add context to the quantitative questions, as well as provide an opportunity for indicators that we failed to identify to come up from industry members. And as part of the interview protocol, we also identified a minimum number of interviews that we wanted to target in each of the 3 coastal regions that you see in the map on the right-hand side. And there's extensive literature about the appropriate number of interviews needed to gain perspective of spread in different experimental groups. And a review of that is listed here. And some of that work suggests that 6 interviews is enough to capture the majority of opinions within a group, and that in one case, 12 interviews was demonstrated to capture over 92% of all themes within a population. So we set our target for a minimum of 6 to 12 interviews per region, and then we were off. So moving on to the industry interviews, a step in our methods here, we were able to accomplish our goal and captured 13 interviews in the South Coast, 14 in the Mid Coast, and 11 down east. And we were also interested in gaining a wide perspective of people within the industry. So this donut chart here shows the different types of roles within the industry that our interviewees represented. And you can see that most of them are harvesters, but we also have representation from trade associations, support services, research, community leadership, and government. And many of our interviewees have multiple roles. And so the first thing we wanted to do was look at some of the questions we ask to verify that the regional differences we expect to see are in fact there. And I'm going to show you some results from this Likert scale, where on the left-hand side you see the possible responses from strongly agree to strongly disagree. And we put forth a statement to our interviewees. The lobster fishery in my community is important to our social and cultural identity. And you can see that here there's very strong agreement across the state, where this is a box and whisker plots where the black horizontal line is the median answer. And you can see that in all regions, there's strong agreement that the lobster fishery is indeed important to the social and cultural identity, with very little spread. But when we ask another question, the overall status of my status or well-being of my community as an indicator of how the lobster fishery is doing, you see the responses start to really diverge, where in the South Coast, the median answer is disagree. In the Mid Coast, we see it between not sure and agree, and down east maintains that really strong agreement with limited spread. And so this sort of validates what we were thinking, which is that the lobster fishery plays a different socioeconomic role in these 3 regions and is therefore useful to evaluate our indicators at this regional scale. And so whenever we were asking questions about the specific potential indicators we developed, we would put them forth in this way, where blank is an indicator of how the lobster fishery is doing. And so I'm going to show you just 4 examples of the 25 that we asked. And those are property values, petty crime, the number of student licenses, and the number of marine resource violations. So this is a broad range of things that we were asking about. And here you can see the average answer of all of our interviewees across the whole state. So statewide, there's agreement that property values are an indicator of how the fishery is doing, disagreement in terms of petty crime, and there's neutrality on student licenses and marine resource violations. But when you dig into the regional differences in those answers, you see some pretty interesting patterns. Property values, there's agreement overall, but at sort of differing levels. Whereas in petty crime, there's a really consistent disagreement across all regions. The student licenses is interesting because while it's neutral across the state, it begs further exploration as to whether that's because in each region, there's a wide spread of information where agree and disagree answers are being averaged to neutral. And then in terms of marine resource violations, though there's a neutral response statewide, we see 3 different answers across the state. And so this is a really clear indication that such an indicator might be important in some regions and not in others. And so the next step of our process is secondary data collection, where we connect these interviews or these indicators that have been identified as important to data to validate them and start to create candidate indicators. And so I'm going to show you sort of the beginning of that process with just one of these indicators, which is property values. And here we were able to get publicly available data at a really fine spatial and temporal scale from realtor.com, but I'll start at this sort of broader scale because we're still sort of in the beginning stages of this work. But here you can see that on the y-axis, we have average median sale price, and this is over time, and you can see our 3 coastal regions. And I was really excited to see this result because it matches up almost exactly with the responses that we're getting from our interviewees, where down east, the increase in property values is a little bit less dramatic, and that helps to validate why there might be a neutral response there as opposed to the agreement and strong agreement in the South Coast and the Mid Coast. And so this is step 1. And while we were looking through this realtor.com data, it's a wealth of information. And we also were able to look at how the proportional or how inventory of real estate has changed over time. And so this shows at the county level, with our colors representing our regions, how the number of active listings has changed through the years. And you can see that across the state, there has been a real decrease in the number of available properties, especially in 2021, with very few properties available across the entire state. And so for us, this paints this picture of increasing property values, decreasing property availability, and sort of this whole scenario of housing stress that has been identified as an important indicator of socioeconomic resilience in the fishery. But in order to sort of contextualize what that means for the fishery, we're now in the beginning of the candidate indicator development phase, where we need to incorporate what we're hearing from the qualitative portion of our interviews in order to understand the full sort of impact of this property value indicator. So for instance, property values is sort of the quantitative component of the indicator, but through our qualitative understanding, we're starting to gather information about how property values lead to a loss of governance of the working waterfront, because when fishermen are not able to live where they work, they then lose access to the governance bodies that make decisions about the waterfront. Similarly, increasing property values that encourage fishermen or other coastal workers to move away from the coast leads to a loss of community culture and identity, which is really important for resilience in the fishery. And an additional thing that we learned is that as property values increase, that also increases rental values, and the availability of sternmen and dock workers has been really dramatically decreased, and that is perceived to be related to these property values. So our goal is to take all of these things together to develop a candidate indicator that sort of represents this housing stress as an indicator of socioeconomic resilience for the fishery that can be monitored. And finally, our indicators will be validated by taking them back to industry and asking how well they match with the lived experience of our interviewees. It's really important to us that all of our work is co-developed and co-ownership is fostered. And so that is our way to encourage that. Our next steps are going to include finding and sourcing more secondary data sources. We have coding of the qualitative interview components to finish up, and we'll continue through the candidate indicator development piece of the project, along with the industry validation that I just mentioned. And with that, I would love to acknowledge Patrice McCarron at Maine Lobstermen's Association, as well as Dr. Lisa Colburn at NOAA for their key advisory roles, and all of the participants of our open meetings. And I'd like to end with an open invitation to all of you here to join us. We are approaching this project with a really collaborative and open approach that includes monthly meetings where anybody interested in commenting on or learning more about or participating in this work is really welcome to do that. And so I'm going to go ahead and put in the chat that link to a form where you can sign up to get an invitation if you'd like to learn more. And thank you so much.
Great, thanks, Teresa. So given the time to try and get a little bit back on track, we're going to jump right into our 5-minute speed talks. So again, we're going to have our 4 presenters do their presentations first and then have a Q&A period. So Eric Annis, if you want to share your screen first.
All right, can you see it?
I cannot see your screen, no.
No, that's not good. Let's try this again. No, really, when I click share, I want it to share. Survey says.
I see your desktop. Oh, there you go.
There we go. All right, we're in business. All right, thank you guys for tuning in this morning. I'm Eric Annis from Hood College in Frederick, Maryland. Today I'm going to present some preliminary results from our Sea Grant funded project that's led up by Heidi Henninger of the Atlantic Offshore Lobstermen's Association. And as you can see from the list of co-authors here, this is a collaborative effort that spans industry, state agencies, and academia. We have conducted a survey of the seasonal abundance of lobster larvae throughout the Gulf of Maine on a scale that really hasn't been done before. And we just finished sorting the samples, so this is the first cut at the data. The take-home message from what we can see so far is that the seasonal curve of lobster abundance varies depending on where you are in the Gulf, so it's not homogeneous. To illustrate that point, I'm going to show you a couple animations of the seasonal progression of abundance. And then a whole mess of graphs illustrating some of the differences that we see from one area to the next. So we sampled 4 transects along the coast from New Hampshire to eastern Maine and 1 station out on Georges Bank. We had weekly sampling with a Newston net pictured here from early June until late September. For this talk, I'm going to focus on the newly hatched stage 1 larvae, which should reflect hatching, and then the stage 4 larvae, which are competent to settle. So this animation shows the abundance of stage 1 larvae over the course of the season. The size of the circle reflects the size of, or the amount of larvae captured. And you can watch for the cross-shelf distribution in the west and a late spike in stage 1 larvae in the eastern portion of the state. We can look at kind of a similar progression with the stage 4 larvae. Some features to look for here is that there are typically higher stage 4 larval abundance in the western portion, and again, a late season spike offshore in the eastern portion of the state. And there are some larvae arriving out at Georges Bank, competent to settle. So let's step through a few of these seasonal curves in more detail, starting with our westernmost transect, which is off the New Hampshire coast. The yellow line indicates the abundance of stage 4 larvae. The blue line indicates the abundance of stage 1 larvae. What you can see here with our 3 stations is that we have stage 1 larvae and stage 4 larvae distributed across the whole transect, but arguably there may be more larvae actually offshore rather than inshore. If we move a little further east to the Booth Bay area off of Maine, we found, again, stage 4 across the entire transect all the way offshore, but stage 1 were only found at our station that was closest to shore. Looking at our stations in Penobscot Bay, and I would point out that these were actually up inside Penobscot Bay, they weren't on outside the bay. But this figure is representative of the 3 stations. We saw very little stage 1 or stage 4 abundance within Penobscot Bay. Our easternmost transect off of Millbridge, Maine showed something very different than what we saw in the western portion of the state. We see stage 1 arriving much later, so they're peaking probably 5 weeks after the western sites. And interestingly, the stage 1s, 2s, 3s, and 4s all arrive at the same time, simultaneously, which is consistent with the idea that they might be advected in from someplace else. We also see a spike in stage 4 abundance offshore. This is outside the eastern Maine coastal current, and this is actually something that we witnessed in a synoptic cruise way back in 1999. So this may be a persistent or reoccurring feature, and we don't really know where they're coming from. And finally, on Georges Bank, we definitely saw a moderate number of stage 4 larvae out over Georges Bank, but we probably didn't get out there quite early enough to pick up the stage 1 season. So what's it all mean? Quite simply, the seasonal patterns of abundance are not uniform. And the talks that we've heard today touch on some of the issues of advection temperature and adult movements and changes in temperature that might be contributing to that. We've got several other data streams that we are working on now to analyze with respect to larval distributions. And a big thanks to my team at Hood College, who just finished sorting these samples about 7 days ago. 5 seconds over time, mic drop. Thank you very much.
Thank you, Eric. That was fantastic. So moving on to our next speed talk is going to be from Kathleen Reardon, tracking lobster settlement signal through pre-recruit monitoring surveys. Kathleen, you're on top of things and it looks great. Go ahead.
Okay, can you hear me now?
Yes, we can hear you.
Okay, so thank you for the invitation to share our research efforts. The Maine Department of Marine Resources Lobster Research Program collects demographic data on the Gulf of Maine lobster population at different life stages through multiple long-term monitoring surveys. All of these data are used in the Atlantic States Marine Fisheries Commission stock assessment to inform the management of this valuable fishery. Today, I will be looking at tracking the settlement signal through pre-recruit monitoring surveys. So we start with the settlement survey, looking at newly settled lobsters. This survey was started in the Mid-Coast area near Booth Bay in 1989. In 2000, the DMR expanded the survey to 40 sites and has been sampling a minimum of 4 sites in each management zone or statistical area coastwide since then. Because the assessment looks at data by statistical area, 511, 512, and 513, I will present that spatial scale to provide information by region here. The blue lines represent an average number of young of year by meter squared annually, while the orange lines are the 20-year averages. Statistical area 513 is broken into west and east because of that longer time series near Booth Bay. As expected, there's been a fair amount of variability in these indices over time, and there was a period of higher than average settlement in the first 10 years of the 2000s. But since around 2012, observed lobster settlement in the survey has been below or at the 20-year average of the index of young of year in all areas. This is a concern if these sustained low densities of settlement represent a future decline in the broader population. So other research has explored the potential processes and population dynamics around this early life stage, but the department does have tools to look at trends in later life stages through other long-term monitoring surveys that target sublegals or pre-recruits and adult lobsters. So today I hope to explore the question, does the depressed settlement signal observed in the past 8 years show up in pre-recruit surveys? So to start, we have the Maine, New Hampshire trawl survey. These figures show the fall seasonal catch of sublegal lobsters. Over the first 15 years of the survey, that sublegal catch showed sustained increases in numbers, but the average sublegal catch was decreased in all regions coastwide in the past 5 years. Breaking the trawl survey data down by size, we can see consistent trends in the 3 size bins below legal size, with an increasing trend until the peak in 2015, and a downward trend since then. To compare with other surveys, I will concentrate on this size bin immediately before legal size, where we would expect these size classes or this size class to molt into the fishery within a year. The ventless trap survey has been monitoring coastwide since 2006. Focusing on the same size class, we observed an upward trend until 2016 and then a decline. These trends have more variability in the west and east, but are quite unambiguous in 512. To compare to another data stream, we can look at our fishery-dependent commercial sea sampling program. Looking at a similar time period and the size class, the catch per trap exhibited increases in all areas, but has shown declines in the past 5 years, especially in 512. In the east and west, the decline has been less steep. Ultimately, the reasons for monitoring are to help provide information for management and the commercial fishery, which depends on legal lobsters. Historically, the fishery experienced exponential growth with a sustained peak around 120 million pounds between 2012 and 2016, but a decreasing trend since then. To look at the trends regionally, we broke the landings roughly into statistical areas where you can see the highest magnitude of landings are caught in area 512. And this is also where the clearest trends have been observed, similar to the other surveys and driving overall landings. There's much more to dig into, but this is just a lightning talk to provide an overview of the available DMR data. But in summary, we have observed sustained low densities in settlement surveys since 2012. The catches of sublegal lobsters are still relatively high in the pre-recruit surveys, but exhibiting declining trends off the peaks. And interestingly, pre-recruit data from the surveys and commercial landings observed parallel declines without a lag. Some future research questions include, there are similar trends between the data streams. Is this a signal of true contraction of the population? Could it be environmental conditions driving catchability or a change in distribution and habitat use within the population? And the biggest question is, what will the future bring? Will the recent downward trend continue or will it plateau? And with that, I will finish up. And thank you so much for our funding sources and the people participating in these programs.
Great, thank you so much, Kathleen. Now we'll hear from Dr. Heather Koopman. Her presentation is Thermal Preferences and Movement Patterns of Female Lobsters in a Changing World, a Scientific Partnership with the Fishery.
Can you see that slide?
Yes.
Okay, great. So thanks very much for giving us a chance to share with you the tagging work we've been doing in the Bay of Fundy over the last little while. And you'll notice I have a number of co-authors here because that reflects the participation of the fishery, which is super important for what we're doing. So we know that temperature is really important for lobsters, especially for reproduction in females. And so we wanted to know what kinds of choices females are making, either buried ones, the eggers, or the V-notchers. And so we deployed 50, this is the first year of a 3-year study, 50 of these hobo loggers, and you have to get these tags back in order to get the data over the last year. And we were super impressed that we got data records from 18 lobsters. It vastly exceeded our proof of concept dreams. And so we're really happy about this. And these sample water temperature every 15 minutes. And I'll show you some of these examples of what these traces look like. And these data files are so big, which is why I have to show this to you in this mode. So here's an example of some thermal traces from some of these animals from December of last year up until June of this year. And each color is a different lobster. And you can see that water temperature on the y-axis here shows us seasonal declines as well as increases in the summer, and that each female kind of experienced a different thermal regime as well, which was different from the minimum bottom temperature that we collected from a trap that was set down at 200 meters. This is the one I just got back last week. I was super excited about this one. Caught on the first day of hauling in LFA 38 in Canadian waters. And we have a long track from this one. That tag's been on for over 300 days. And the cool thing about this one is that it's still collecting data. The way that you get data off these tags is you download via Bluetooth to your phone. And some of the fishers are super excited about it. And so they've got the apps on their phone. And if they catch one of these lobsters, They just download the data and text the file to me and then put her back, and she continues to collect data, which is fun. And you can also see sort of some oceanographic changes here. This is a 2-degree temperature rise that happened in March. We figure there must have been an influx of water because it wasn't unique to this lobster trace. We also saw it on the trap data as well as some of the other lobsters. So that's the thermal stuff. The movement study is basically a repeat of a tagging study that Alan Campbell did out of St. Andrews in Canada in the 1980s. And so our big question was, well, 40 years later, what kinds of movement patterns are we getting from buried females? Are they similar or not? And he tagged over 2,000 lobsters and had a pretty good return rate. The map on the right shows you generally where the lobsters went and sort of how long it took them to get there. So far in the 2 years we've been doing this is a pretty low tech project. These are plastic zip ties with my cell phone number and a number for the lobster on them. We've put out almost 3,000 of these, and we've gotten 860 reports, which is a pretty good return rate that we're happy about. But the best part of this is the number of boats that are reporting. I was really happy to see the number from Maine and Nova Scotia, in addition to the ones around the waters around Grand Manan. And so what are they doing? Well, so far our longest deployment is 500 days, which we're pretty impressed about. The greatest distance has been 300 kilometers. And these show you sort of where she was tagged in red and where she was caught in green, and then a number of other ones along the bottom. And we're still analyzing these data. So what have we learned? Female choices, female lobsters make very independent thermal choices. And that generally, overall, despite the fact that we're 4 decades later and we've heard of all these changes in the Gulf of Maine and Bay of Fundy, the movement patterns look fairly similar. What's kind of relevant here is that Gulf of Maine and Bay of Fundy lobsters don't see borders, right? They move across quite a lot, because I get reports from both sides. And we're also getting some tag data from the gray zone. These fishers are incredibly important for data collection because they're out there every day. And so we really rely on this. And why does this program work? I think a couple of reasons. First, it's really easy to report the data. I just need the lobster number and location. A lot of them find it easiest to just take pictures of these things and text them to my phone. And then I get right back to them with where the lobster was tagged and when, so they can see what these movement patterns look like in real time. And we also post our data on social media, Facebook page, so they can see all the thermal traces too. So I'll wrap that up quickly, thanking our funding sources, but mostly the fishers who are participating in this and texting me and asking me questions. It's just amazing to get such great buy-in from the fishery, both in Canada and in the US. With that, I'll stop sharing. Thank you.
Thank you very much. That was fantastic. So our final speed talk in this series here is going to be by Dr. Catherine Maltby. Her presentation is Social Resilience in the Southern New England Lobster Fishery. So take it away, Kat. Your screen looks great.
Thank you. Hi, everyone. So I'm Kat Maltby. I'm a postdoc at the Gulf of Maine Research Institute. And I'm going to be taking us a little bit south today to talk about social resilience in the Southern New England lobster fishery. And I just want to acknowledge our co-authors, Lisa Colburn at NOAA and Kathy Mills at GMRI. So things in Southern New England for the lobster haven't been looking so great since the late 90s, really. So abundances have been below critical threshold since around the early 2000s, and that's also reflected in fisheries landings, which peaked in around 98, 99, and then have since declined and plateaued up to the present day. And there's been multiple different hypotheses suggested, and which are also contested among different stakeholders. But these include things like pesticides, reduced natural mortality rates, but increasingly emphasized warming as well, and shell disease. And while there's been this kind of long-term decline, there's also been more punctuated mass mortality events, particularly in the late 90s and early 2000s, the most famous of which occurred in Long Island Sound in 1999 and was declared a federal disaster. So in this project, we're really interested in kind of how people responded to these declines over the years and what are the factors that have been influencing those responses. So to do this, we've been using a slightly different data source, that's being news articles. So because of the kind of importance of lobster to communities in the region, the story of the decline has been really well catalogued in both regional and national level news sources. So we've dug into news articles from across the region and nationally to start pulling out individual stories of how people have responded. So we got 90 articles in total, and which we were able to extract stories from around 82 individuals. So what are these response pathways that we've been identifying? So you can kind of split them into these three typologies. The first, viewing resilience as persistability. So people coping and trying to remain lobstering for as much as they can, doing that full time, in some cases changing their fishing practices, perhaps moving fishing grounds, or some of their business operations. For most of these people, though, they're often heavily reliant on financial buffers, such as incomes through their partners or savings and retirement funds. Another group we view kind of more as resilience as adaptability. So people adapting and diversifying their portfolios. So whilst part-time lobstering and altering some of the lobstering practices, also then doing other things. So either part-time fishing for other species such as sea bass or scallops, or working outside of the fishery, so into more construction and manual labor type jobs, or sticking on the water but doing something other than fishing. And then finally, we see another group of people that are really making much more substantial and significant changes and transforming out of the fishery. So leaving lobstering into a different fishery full-time, or moving into another job on land full-time. And for some, that means physically moving to a new area. And so in this case, viewing resilience as transformability. We can just see how these responses play out both kind of compared to each other. So generally we're seeing quite similar patterns in terms of numbers across those three typologies. But then when you split it out at the state level for where people were living at the time, you can see that, for example, in New York, we're seeing much more transformative responses, perhaps where the declines have been most significant and other options to diversify are more limited, compared to Massachusetts, where you're seeing more of these coping and remaining responses, perhaps because there's opportunity to go further offshore, for example. We're then interested in what are the factors shaping these responses. So I can't go into them all in detail here, but there's around 20 that we identified, which map onto 5 overarching dimensions of resilience. And these factors scale from the individual level through to household, community, and governance levels. So the whole kind of socio-ecological system. I just wanted to highlight the importance of emotions that has arisen kind of through our analysis, and which really highlights kind of that more personal and individualistic kind of decision-making process when people are responding to environmental change. So just finally to wrap up, kind of trying to think about how can we translate some of this Southern New England experience to the Gulf of Maine and lobster fishery when we're thinking about resilience for the future. So I think it's important, perhaps obvious to state, but still important nonetheless, that people will want to and will respond in multiple different ways to future potential declines. And so that means that there'll be different dimensions of resilience that are important for those different pathways, and therefore we need to be thinking about multiple different strategies to support people's resilience. Our work also indicated that there's a really important aspect of kind of the individual and personal nature of how people are responding and their individual kind of factors influencing them. And this connects really importantly to well-being considerations. So the declines quite substantially impacted people in terms of mental health and psychologically. And so it raises the question of whether increased resilience always leads to increased well-being outcomes. Thinking a bit more about sort of more specific and supportive mechanisms to promote resilience. So agency and collective actions were other key dimensions of resilience. So thinking about ways that we can support that, for example, through information and knowledge exchange to help people shape and plan their futures, and also strengthen connections, both people within the fishery as also kind of other stakeholders to help in those kind of more collective actions. And finally, I just wanted to pose a question, because we're reflecting on that in this project. Resilience in fisheries is kind of this goal of a lot of management and kind of policy perspectives, but we need to be careful about thinking about what we mean by resilience and at what scale. So for what might be resilient at one scale, for example, the individual, that might not always translate to that at another level within the fishery, so at the community, for example. So that's what we're hoping to explore next in our work. So yeah, thank you for listening. I'm happy to take questions now, or you can email or tweet me. Thank you.
Great. Thank you, Kat. And thank you all to all of our presenters so far. We do have time for one question for our speed talks. Again, anybody in the audience, attendees, please put your questions in the Q&A box if you have a chance. But Kathleen, this is a question for you. Is the question of environmental drivers altering catchability something that can be backed out by comparing trawl survey and trap data?
That's a good question, Ben. Environmental drivers altering catchability was a big topic of conversation in the last assessment. We found that it was likely that there were unrealistic increases in the observations for multiple surveys. For example, a long-lived population can't double in a year or two over time. So catchability must have been changing. But unfortunately, this impacted both trawl survey and trap surveys. So I don't know how we would find the interaction between the two. For this past assessment, we did develop catchability covariates based on temperature indices, but we all acknowledged, I think the whole technical committee and stock assessment committee acknowledged that this needed more work. So more research and thought needed.
Excellent, thank you. And again, thank you all for our oral presentations. We are going to switch gears now for our poster session. I hope you all can hang around for that, but we will again be recording everything and posting it on the RARGUM website. So Cassie, if you give me a hand promoting the first group of presenters to our panel to pin them. We're going to start off with Jay Kim, whose poster is titled Understanding Spatial Dynamics of Landings in the Inshore Maine Lobster Fishery. So Jay, if you want to share your screen, we'll get going.
Hi, can y'all hear me? Sorry
Yep.
about that. Okay, so this is my poster. So quick presentation of the work that I've been doing so far at the Chen Lab. If you don't know who the Chen Lab are, we've been sort of working with modeling dynamics within the Gulf of Maine, specifically working with the lobster fishery closely for a while. Although my advisor has left for Stony Brook University, we're still working a lot with the lobster industry. The project that I'm presenting to you right now today is understanding the impacts of climate-induced distributional changes on the fishery landings for the inshore Maine lobster fishery. It's a part of a larger grant work with the NOAA Coastal and Ocean Climate Applications, really looking to understand the adaptations to climate change within the fishery through a biological, economic, and social lens. The part that I'm really working on is the bioeconomic aspect of the understanding the adaptations to resilience, and this is serving as the groundwork, which will further inform the bioeconomic modeling aspect of the grant. And this is really just understanding the sort of, sorry, let me try that again. We really are blessed with the lobster fishery for having such a long-term time frame of data collection, and within the Gulf of Maine as well. And so we have historical landings by county all the way to the 1960s. We also have modeled environmental data all the way to 1970s. And we also have a lot of predicted future data for sort of how we expect the Gulf of Maine to change, as we've sort of heard throughout this entire session. Basically, what I've done so far with this project is we got historical landings data. We sort of split that up by the lobster zones and county throughout the coast based on the down east, mid coast, and southern coast regions. We then have this habitat suitability index model, which has been developed in the Chen Lab before, and we expanded upon that to include seasonal and, I'm sorry, the screen seems blank. Can you guys still see the poster?
Jay, I think you entered out of the.
Oh no.
There you go.
It's terrible. We'll just stick to this. Essentially, we predicted HSI based off of bottom temperature, bottom salinity, depth, latitude, and longitude for the current inshore Gulf of Maine. We hindcast that back to historical data and validated it with the sort of the lobster management zone spatial dynamics in mind. And then we forecasted that based off of the future IPCC climate scenarios. And then we put that we combine that data with our landings data to make a predictive model again of predicting proportional landings based off of environmental parameters. Basically, what that lets us know is that we can sort of predict proportionally how the landings are going to change within the coast based on these lobster management zones, which is important for the management spatial scale and shows us a distinct spatial area within the Gulf of Maine and how that's changing with what we think is going to change in climate. Keep in mind, I'm not trying to say this is the X number of lobsters that this zone is going to catch in the future, but more of like the distributional changes of how that might look proportionally within the entire Gulf of Maine. What we see is that despite varying changes in HSI, as things may get better or worse, we think that the proportional landings for the down east regions in the future, despite their warming or cooling circumstances, are going to decrease. That shows us that in itself can serve as a basis for the bioeconomic aspect of it, where we can put in cost and economic data along with the proportional changes in landings to see how the different regions along the coast are going to react economically. And so therefore we can have an economic or bioeconomic aspect of how the lobster fishery will change due to different environmental factors. Sorry, I think I ran over time.
Great. Thank you very much, Jay. So our next presenter is Crystal Boley, and I'll be sharing her poster. There's some tech issues earlier today. But Crystal, if you're able to unmute and let me know.
Hi, can you see me? Okay, there. Yep. Hi, so I'm just going to get right into the background for why we were asking this question. I'd like to talk about the sublethal effects of low-dose exposure to imidacloprid on the American lobster. So some European Atlantic salmon farms are using imidacloprid as a chemotherapeutic to remove sea lice, which are ectoparasites that may harm or kill fish at great cost to the industry. The treatment occurs on well boats and the water is processed to undetectable levels of imidacloprid before discharging. Their limit of detection using HPLC is the high concentration we chose for this experiment. Prior to being used in Maine, we wanted to determine possible impacts to the highly valuable non-target species American lobster. So I also want to point out that lobsters have important defensive behaviors they use to avoid predation and to compete for resources. So the question we asked was, if European practices are brought to Maine, how might this affect the behavior and physiology of nearby American lobsters? And I just want to clarify, there are no plans for bringing it here. We just wanted to proactively look at this ahead of any consideration since it's being used elsewhere. So to test this, we exposed subadult female lobsters to 0, 0.3, and 30 parts per billion imidacloprid concentrations for 120 minutes. Then upon removal from treatment baths, behavior was video recorded, and then hemolymph samples were taken. This was our measured acute response. All lobsters went into clean water for a 5-day recovery period. Then behaviors were recorded and hemolymph was sampled again to give an idea of persistent effects. This was our measured chronic response. Our behavioral endpoints included defensive behaviors used to avoid predation and used during social interactions and competitions for resources. These are ecologically significant as they increase survival and reproductive fitness. These are economically significant because active, healthy looking lobsters are chosen for live market sale at the highest profit. Our hemolymph endpoints were measured as indicators of stress to reveal physiological health. Today, I will just be focusing on 2 behaviors, tail flips and marrow spread, but the others listed here showed significant differences as well. I will also talk about 1 hemolymph endpoint, crustacean hyperglycemic hormone, but the others did not show significant differences. So marrow spread is a behavior used by lobsters and other crustaceans to indicate fighting ability or aggressive intent. It's the raising of both claws up and outward over their body. and marrow spread was significantly reduced in the 30 part per billion imidacloprid treatment. Less than half these lobsters marrow spread compared to the control. Tail flipping is an escape response that requires energy and coordination. Tail flipping was significantly reduced in the 30 parts per billion imidacloprid treatment, and this effect persisted 5 days later. 80% did not even have a tail flip response when removed from their treatment, and 25% did not have a tail flip response 5 days post-exposure. Crustacean hyperglycemic hormone plays a key role in regulating hemolymph glucose in crustaceans and is commonly measured as an indicator of stress response. So crustacean hyperglycemic hormone was significantly different in the 0.3 parts per billion treatment 5 days after exposure. This was the only significant hemolymph endpoint, so the physiological observations did not capture what was happening to behavior further downstream. So I guess I'll just conclude with this just emphasizes the value of observing effects as far downstream and close to responses of interest as possible. And thank you for listening. Contact me by email for any questions.
Thanks, Crystal. Up next, we have Evie Leyland, who will take us back to larval lobsters with her poster Ontogeny of Pursuit, Handling, and Ingestion of Planktonic Prey by Larval American Lobster, Homerus americanus. And we see your poster, Evie.
Awesome. So good afternoon, everybody. My name is Evie Leyland. I'm a master's student at the University of Maine, co-advised by Rick Wally and David Fields. My research focuses on the feeding behavior of early planktonic stages of the American lobster. So a little bit of background, the American lobster is an important player in the coastal Northwest Atlantic as a benthic consumer and the target of one of the most valuable single species fisheries in North America. In recent years, there's been a decrease in the number of juvenile lobsters settling on the benthos, despite increases in the number of reproductive adults in the population. These declines in settlement correlate strongly with the climate-driven shifts in the zooplankton assemblage in the Gulf of Maine. Zooplankton serve as the primary food source for young lobsters during their planktonic stages. However, there are still large gaps in our knowledge of the specific diets of larval lobsters during this crucial period. As they develop, lobsters undergo significant morphological and behavioral changes, nearly doubling in size between stage 1, shown here, and stage 4 before settling on the seafloor. The new hatch stage 1 larvae haven't yet developed any pleopods, and their feeding appendages are really small. Watching them swim, they're really slow and pretty uncoordinated. But by stage 4, they have fully functional claws and they glide through the water really fast. They swim kind of Superman style with their claws held out in front of them. As you can imagine, these changes translate into differences in their ability to capture and handle prey. Because of this, shifts in the available prey assemblage may have greater impacts on lobsters at different points in their development. So I addressed the following questions using feeding and behavioral experiments in the lab. 1, how does lobsters' prey selection change as they progress through these early life stages? And 2, what are the underlying mechanisms influencing these changes in prey selection? So to answer these questions, I conducted a series of feeding experiments using 7 different prey items of varying sizes and swimming abilities, categorized here as either slow or fast swimmers with slow or rapid escape responses. I also collected video observations of different larval and post-larval stages feeding on zooplankton using a Kreisel tank equipped with a video camera. I recorded free-swimming lobsters to measure swimming speeds and pursuit times and path lengths, as well as tethered lobsters to look at the more fine-scale handling behaviors. The results of my feeding experiments are shown here in figure 2. So the circles indicate faster swimming prey and the squares indicate slower prey. During the first 3 stages, we see higher ingestion of slower swimming prey. Stage 1 and 2 have very similar feeding patterns, followed by a drop in ingestion in stage 3, and then a rapid rise in stage 4. The stage 4 exhibit highest feeding rates on Artemia, and they also show an increase in feeding on Calanus phenmarchicus. Figure 3 down here shows the handling time in minutes for two different prey species, Artemia in blue and Calanus in red. At stage 1, it's taking about 7 minutes to consume an Artemia and 54 minutes for Calanus. At stage 4, we're down to it takes only a second to eat one Artemia, and it takes a little more than a minute to eat a Calanus. And then 3B shows the increase in swimming speeds over time. So as I wrap up, I'd like to share a video from my feeding experiments. The early larval stages exhibit a preference for smaller and slower swimming prey, which can be captured and consumed faster. Stage 4, significantly faster swimmers and can handle the same prey 40 to 50 times more efficiently than stage 1s. The benefits of reduced handling include increasing the amount of time a lobster can search for food, increasing the amount a lobster can consume in a given amount of time, and decreasing the time that it spends in a vulnerable feeding state. Benefits of increased swimming speed include the ability to search greater volumes of water, to increase their encounter rates, and capture faster prey items. In conclusion, understanding the early stage lobsters, how they interact with their prey and their environment helps to create a complete picture of how they interact with the ecosystem as a whole and how they might be impacted by altered prey availability associated with climate change. So thank you to all my collaborators, and thank you for this opportunity.
Thanks, Evie. As if you weren't hungry enough, let's talk about eating with larvae. So our final poster for this first group of individuals is Kristen Dinning, whose presentation is changes in juvenile American lobster density and habitat use over 30 years in the Bay of Fundy. We can see your screen. We can't hear you, Kristen, if you're talking.
Okay, hopefully sounds are coming out of my mouth now.
Yep, we can hear you.
Okay, perfect. Okay, so I'm Kristen Dinning, working with Peter Lawton and Remy Rochette at the University of New Brunswick in St. John, Canada. And I'm going to talk about changes in juvenile lobster density and habitat use over the past 30 years in Macy's Bay. So to start, I have a little graph here. We see that lobster landings by the fishery have increased over the past few years, the past 3 decades. And this suggests that perhaps lobster populations may also be increasing, possibly due to improved juvenile survival with declines in predatory groundfish. Now, this reduced predation may now allow juvenile lobsters to occupy cohesive sedimentary substrate, i.e. mud, although it does offer poorer shelter than the lobster's preferred structurally complex rocky bottom. If juveniles end up saturating rocky habitat, expansion into less densely occupied sedimentary habitat could remove constraints to further population growth. So we compared historical and contemporary juvenile lobster densities across a gradient of sedimentary substrate cover to look for changes in juvenile density and habitat use. So I'm just going to try and zoom in here. Okay, so in the Bay of Fundy, our study site is Macy's Bay, and it contains extensive patches of mixed mud and sand seafloor. So these are my unshaded blue areas, as well as rocky seafloor, the lightly gray shaded areas, which is comprised of various mixtures of bedrock, boulder, cobble, and gravels. Peter Lawton with Fisheries and Oceans Canada completed scuba transects in Macy's Bay in the 1990s. These are our orange lines here, and in the 2000s, the blue lines. We revisited several of his sites in 2019, 20, and 21, i.e. the 2020s, or our dashed lines in black. And scuba divers counted all lobsters and recorded seafloor composition in each of these transects. In this poster, however, I'm only going to present data on the juvenile lobsters. Okay. So for our results, on the y-axis is juvenile lobster density for each of our 3 decades. So the 1990s are the orange data, the slope on the lowest slope on this graph here. The 2000s are blue data and the blue slope at the top of the graph. and the 2020s are green data and the green slope, which is in the middle. On the x-axis is the percent of seafloor covered by sedimentary habitat. That's our muds and sands. And as expected, our juvenile lobster densities for all decades, i.e. all our slopes, dropped with increasing coverage by sedimentary seafloor. That is, as we move from the complex rocky habitat on the left side of the graph into more exposed, less protective muds and sands on the right of the graph, juvenile occupancy declines in these habitats. This decline is most pronounced in the 2000s, so the blue line at the top. The 2000s also showed the highest juvenile densities for any given habitat composition, i.e. at any percent cover. If we now look at the far right side of the graph, the data points in the red oval, we can look at just the data for 100% sedimentary habitat, which in Macy's Bay is largely comprised of cohesive or sticky mud seafloor that these lobsters can really manipulate and burrow into. So if we take that data from the red oval area there and popping that into this graph on the right, again, we have juvenile densities on the y-axis, 3 decades on the x-axis, and we see that in 100% sedimentary habitat, densities have increased over time from complete absence in the transects in the 90s, so our orange data, to significantly higher densities in the 2000s and 2020s. So in conclusion, the increased abundance of juvenile lobsters on sedimentary seafloor over the past 3 decades suggests that even more juveniles are exploiting sedimentary habitat than historically, possibly due to this reduced predation pressure and better survival in these sedimentary habitats than in the past. And as structurally complex rocky nursery habitat grounds are often sparsely distributed, this increased flexibility in habitat use could help more juvenile lobsters recruit to the population and eventually to enter the fishery. So with that, I will thank you all for listening.
Fantastic, thank you. And just a reminder to everybody, the posters are posted online on the RARGOM website. So obviously we haven't had enough time to look over all of the details, but I'd encourage you all to look over there. We do have one question for Evie. I know you answered it in the chat, but I think for everybody, it might be cool to address this here. Have you looked at these prey species in terms of carbon instead of the number ingested? Jess is wondering what scale, I wonder what items are preferred on that scale. And I don't know, Evie, if you or anybody else who's been working on the feeding experiments with you might also want to chime in there.
That is something that we're interested in looking at. And it's my next step in the process is going to be to look at carbon value of these organisms instead of just looking at number of individuals. But we haven't looked at it yet.
Great, thank you. I don't see any other questions in the chat. Do any of the panelists, I know the panelists have some issues with the Q&A function. If you have any questions, now is a good time. We actually caught up, we have a few minutes. So, oops, I lost my view. All right, well, in an effort to make sure that everybody is able to get to lunch and not too hangry by the time we get done, I'd suggest we move on to our second poster session. So we did have a change. Phoebe is unfortunately not going to be able to present, but Ben Gutzler is going to be sharing on her behalf. And so their poster is Effect of Warming Sea Temperatures on Post-Larval Lobster Swimming Performance and Energy Reserves. So Cassie, can you pin Ben instead of Phoebe? Sorry about that. And we see your screen, Ben.
Awesome. Yes, as Amalia noted, I am in fact not Phoebe, who is off in an airport at the moment, I believe. So you get me instead. This is a small component of some of the work that we've been doing with another one of these American Lobster Initiative funded grants through Sea Grant. And we have several other subcomponents that are not appearing today, but we are still excited to share what we have here. So this is, we are looking at how the swimming performance of lobster post larvae and their nutritional condition may change as we warm the water. And thankfully, since this is the Gulf of Maine centric thing, we don't need to worry about all the nice graphs from Andy Pershing at all, because it is, you know, very, we all know the background here. So basically what we ended up doing is we took lab reared post larvae and subjected them to 72-hour swimming trials in these chrysal tanks that you can see in the sort of the center of the screen here. During that time, we took video observations, so a minute of video every hour. And then at the end of the trial, the larvae were frozen and analyzed for dry weight and percent lipids. So we can get an idea of both behavioral activity as well as nutritional condition at the end of the trial. And that was, the nutritional condition was compared to a pre-trial reference sample. What we found was actually quite interesting. We found no obvious differences in dry weight or lipids between the animals we swam and the pre-trial controls. I will also note that our values are bang on with what Alex Asher and his people found and saw earlier, which was a very reassuring thing to see. And the fact that he didn't see really much mortality in starved animals made me feel a lot better about the fact that we were starving them for 72 hours during these trials. So it's not surprising we didn't see a ton of decline there. But what we did see is a rapid decline in swimming activity of larvae held at a higher temperature than our 15-degree controls. So this is a first order Loess slider fit to the data because we do see this strong diel component, which is reflected in some of the literature. So the larvae are swimming at a more during the day than the night. And we also, this is a significant decline between the 15 degrees and 22 degrees in terms of the proportion of larvae swimming. If you do a linear fit to it, but I think the display makes some of the other patterns clearer if you have this slider here. So larvae seem to run out of gas faster and revert to this sort of drifting catatonic phase where they're still alive if you poke them, but they just don't move much if you just watch them. So you can see in the video here in the 72-hour one, this is the 22-degree one on the left and the 15 on the right. So you can see a very strong difference in the number of swimming versus just actively drifting between the first hour of the trial and the last. So this suggests to us that this decrease in swimming behavior in warming water may both act as a way to mediate the nutritional consequences of warmer water and higher metabolisms, as well as potentially playing a role in this shifting distribution of larval recruitment patterns, because you might get larvae at these higher temperatures than historic, just not being as vigorous swimmers moving inshore. Some of the other work we're doing at the moment is trying to figure out how directional their swimming in the wild is, but we don't have that for you today. And we're also, this is just the first year of data, so we're very excited to see what we get when we start crunching the numbers for the next year. And that's what I have for you guys. Thanks for letting me tag in on Phoebe's behalf.
Great, thank you, Ben. All right. Up next, we have another presentation from Eric. He's just doing too many projects. So Eric, if you want to take it away for your presentation, American lobster larvae exhibit ontogenetic shifts in thermal tolerance and an overall greater tolerance to thermal stress when reared in the wild versus the laboratory. It's a mouthful, but take it away.
Yeah, when it rains, it pours. So good afternoon, and thank you for those of you who are still here for sticking around for the poster session. I'm going to present some results today that are part of a larger NSF-funded project in which we're attempting to link thermal physiology of individual larvae to patterns of settlement on an ecological scale. This is a collaboration with Bigelow Laboratories, Doug Rasher, Marcus Frederick from the University of New England, Jess Waller from Department of Marine Resources, and Aubrey Jane at the University of New England. The take-home message, one slide at a time. The take-home message is that we are seeing ontogenetic shifts in thermal tolerance through the larval stages, and wild larvae are more thermally tolerant than lab-reared larvae. So for these experiments, we use scope for activity as a general metabolic indicator of temperature stress. We used respirometry, microrespirometry, to measure oxygen consumption by individual larvae in all 5 developmental stages that were all reared at 18 degrees Celsius. Scope was defined as the difference between oxygen consumption when swimming actively, the difference between that and oxygen consumption at rest. When the difference is positive, it suggests that the larva still has some metabolic scope to engage in swimming activity. When scope is 0 or negative, it suggests that they've got nothing left metabolically and they're probably approaching a critical temperature. To illustrate the shifts that we're seeing in thermal tolerance, I'm going to show you a series of figures that depict scope for activity as a function of temperature for each developmental stage. So in this figure for stage 1, you can see that scope drops below 0 at 4 degrees and at 32 degrees, suggesting that these are the lower and upper critical temperatures respectively. Now, all of the respirometry work was done in an acute treatment that only lasted about 30 minutes. We also did chronic treatments, which you see to the right, where we looked at survivorship over, well, basically we put them in a temperature and waited until they either molted or died. So you can see that in a longer term treatment, the stage 1s are actually sensitive to temperatures at 26 degrees. We see our first ontogenetic shift in the molt from stage 1 to stage 2. And it's reflected in a decreased tolerance of cold temperatures. So you can see the lower critical temperature shifts from 4 degrees in stage 1 to 8 degrees in stage 2. And I should note that you have a corresponding decrease in survivorship in the 8-degree chronic treatments. And this pattern persists through stage 3 and into stage 4, where we see another ontogenetic shift, which is a reduction in heat tolerance in the stage 4 labrador larvae. If we move on from stage 4 to stage 5, we see yet another ontogenetic shift, which actually is a return of cold tolerance. So we see that lower critical temperature shifting to lower temperatures. And this tracks with, this is when the transition is occurring from them living up very close to the surface, actually, as stage 4 larvae, to settling on the bottom where temperatures are colder. Now, moving on to the difference between lab-reared and wild larvae, this is pretty straightforward. Wild larvae had substantially lower minimum critical temperature and substantially higher maximum critical temperatures than the lab-reared larvae. To reinforce the point, if you look at the chronic treatments, we had much lower survivorship at low temperature in lab-reared larvae, and well, nothing died that we brought in from the wild when exposed to chronic temperature treatments. But wait, you say, lobster larvae are ectotherms, and the temperature that you rear them at will affect their thermal tolerance. Well, we reared them at 14 degrees at a lower temperature just to see what that would do. And indeed, that does improve their tolerance of cold temperatures, but it also doesn't change their upper thermal tolerance. So the short of it here is that regardless of your rearing temperature, the laboratory reared larvae appear to have a narrower envelope of thermal tolerance than the wild larvae have. So our conclusions are pretty straightforward. There are ontogenetic shifts. They appear to correspond to the environmental conditions for each larval stage. And wild larvae appear to be more tolerant of thermal stress than lab-reared larvae. And this is a big deal because every paper that has been published on the thermal tolerance of lobster larvae in the past 100 years has used, wait for it, lab-reared larvae. So there's potentially some big things we need to consider here in terms of thermal niche modeling, understanding the linkage between temperature and settlement, and ultimately the impact of climate change on the species. Thank you very much. Oh, and a big shout out to Hannah and Alexis, who spent a lot of time raising thousands of larvae in individual jars in the environmental control room.
Thank you, Eric. All right. Up next, we have Jacob Calviti, whose presentation is Declining Maternal Size and Changes to Embryo Quality in the American Lobster Over a Decade. And we can see your screen, Jake. But I can't hear you if you're talking.
Okay, can you hear me now?
Yes, excellent.
All right, my name is Jake Calviti. I'm a master's student at the University of North Carolina, Wilmington. And today I'm going to be talking about maternal size effects on the egg quality of American lobsters. So there's evidence that recruitment in many marine fisheries is disproportionately dependent on the contributions of the largest females. While the positive relationship between fecundity and body size in American lobsters is pretty well known, there's a bit less attention's been paid to the effect of maternal size on quality of eggs, which could potentially also affect recruitment. So the characteristics of egg quality that we're interested in this study are diameter, energy content, and fatty acid profile. Some past work has found that larger eggs of higher energy content and higher omega-3 fatty acid content produce larger larvae that are more likely to survive. So the eggs I looked at in the study were collected in the fall months of 2008 through 2020, with some years in the middle without sampling, by my advisor, Dr. Heather Koopman, aboard commercial lobster fishing vessels in the Bay of Fundy. And about 200 to 700 ovigerous females were sampled every year. So moving on to some results here, you can see the pie charts on the left there are showing the size distribution of the ovigerous females that were sampled. The top pie chart there is showing the early years of 2008, 2009. So the largest size class, the 121 millimeter carapace length and over, in the first 2 years of sampling, that made up about 22% of the ovigerous females sampled. And moving down to the most recent years, only 3% of females were in that largest size class. So there's been a pretty abrupt shift according to our sampling, to smaller size classes. And if there is this effect on egg quality, that makes it all the more important. So in this middle column, we're showing the this top figure is the joules per egg, so the energy content in relation to maternal size, and there's a clear positive and significant trend between carapace length and energy content. And the figure below that showing diameter in relation to female size, and there's another clear positive trend. So larger female lobsters are producing larger eggs with higher energy content. For the fatty acid results, there was a significant effect of size on the fatty acid profile of the first 2 years of the study. So the early years, the fatty acid profile was affected by female size, but not in the most recent years. And that could possibly be explained by the lack of the very large females in the most recent years, as only the largest size class had significantly different fatty acid profile of their embryos from the smaller and medium sized classes. So this last figure in the top right-hand corner is showing the reproductive output, which was fecundity multiplied by the joules per egg. So this is the total energy investment into their brood. And the triangles there are showing the earlier years and the circles, the later years. And so as you can see, the larger animals of these early years are investing far more energy into their broods than these smaller animals in more recent years. So the big conclusions so far of this work are that larger maternal lobsters are producing higher quality eggs, and they're investing more energy into the brood than smaller lobsters. And we're seeing, based on our sampling, that there's been a shift in size distribution to smaller lobsters in the Bay of Fundy. That's all I have for you. Thanks for listening, and I hope you have some questions for me.
Great, thank you so much, Jake. And last but certainly not least, we have a presentation from Kristen Clemen. Her poster is titled "Science Industry Collaboration Builds Trust While Opening New Windows on Climate-Related American Lobster Range Shifts in the Gulf of Maine."
Everybody see that okay?
Yes, you're good to go.
Perfect. Good afternoon, everyone. My name is Kristen, and I'm a research associate at UMaine's Darling Marine Center. Today, I'll be talking about some results from a collaborative research project which began in 2016 involving myself, Rick Wally at the University of Maine, Kathleen Reardon, who we heard from a little bit earlier today, and our industry partners, Kurt Brown and Jordan Druin. What we refer to as the Deepwater Collector Project is a science industry partnership that was initiated to evaluate the relationship between bottom temperature gradients and the depth-wise and along-shore distribution of newly settled lobster and juvenile lobsters. The project tests the hypothesis that the recent surge in lobsters eastward and into deeper waters in the Gulf of Maine has been driven by warming temperatures. We accomplish this by deploying post-larval biocollectors, which, if you can see my mouse, are pictured right here, in coordination with the Maine DMR's ventless trap survey across 3 different depth strata in the west and east segments of Maine's coast. These 2 study areas, as you can see in this image down here, and the corresponding thermoclines, have oceanographically contrasting thermal regimes, allowing us to evaluate the influence of temperature gradients and other environmental factors on the nursery potential of the seabed for new settlers, as well as movements by older lobsters. This collaboration fills a data gap on depth-wise patterns of lobster settlement not quantified by the larger scale and long-standing American Lobster Settlement Index, or ALSI, which monitors the recruitment of young of year lobster in coastal nurseries from Rhode Island all the way up to Newfoundland, but only in shallow sites that are accessible to divers. This effort also enables us to follow cohorts through time by incorporating data on adolescent lobsters caught in the DMR's ventless traps. So I'm going to go over some of our lobster settlement related findings from the first 5 years of the project. All of these figures are displaying average lobster catch per trap or per collector by depth. The top 2 rows of figures are collector data from 2016 to 2020. And the bottom figure is VTS data in 2-year bins starting in 2006. In the top row figures, we see that young of year lobster have spread into deeper water over time, especially in our western study area in Casco Bay. And down east, we're seeing increases in young of year across all depths. Moving to the second row of figures, there's been some declines in older juvenile numbers in both study areas, which might be a reflection of some of the poor young of year numbers in 2016 and 2017. So we might expect the older juvenile numbers to increase in the coming years. And I can say that in the field this summer, we were getting what felt like more older juveniles than is typical. And then lastly, moving to the bottom row of figures, the VTS figures, adolescent lobster numbers have either been stable or maybe slightly increasing, which might be a reflection of the high older juvenile catch in 2016 and possibly preceding this project. So to conclude, the collective resources and experience of both commercial lobstermen and the DMR allows for more extensive and comprehensive research. As we continue to face many changes in the Gulf of Maine, collector-based sampling will continue to be a useful means to monitor recruitment patterns, changing recruitment patterns in lobster. Thank you, and I'm happy to answer any questions.
Thank you, Kristen. Well, we certainly jam-packed quite a bit of information in this almost coming up on 3-hour session. We do have, Eric, a couple of questions for you. So the first one is, how do you resolve age data within the 4th post-larval stage for wild-caught post-larvae? In other words, how do you account for the wide variability in condition and age of 4th stage post-larvae for your respirometry trials?
That's a really good question. This year, we didn't look at it at all. We caught them in the wild, brought them into the lab, used them within a day or two of having captured them. But I have used molt staging in previous work, and that's a little more difficult, but once you get a feel for it, you can slap them under a microscope and see where they are at where they are within the molt stage. So that's probably the best indicator, and that's probably something that we will be incorporating in future years.
Great. So we have another question for you. The lab larvae seem to tend to be smaller than the wild ones. Is some of the tolerance due to differences in thermal inertia or surface area to volume ratios between lab and wild larvae?
Oh, that's also a good question. We're going to look at that. I can't give you an answer right now, but what I will say about the differences between lab and field caught larvae is that this is historically the differences between these two have been stark. The lab reared larvae, in every lab that raises them, I think, come out half the size of wild larvae. They swim slower. They're not as aggressive. There are a lot of factors that could contribute to this. The area that they have to swim in while they're developing, the amount of food or the quality of the food that they're getting as they're developing. So I think then taking it to a physical aspect of thermal inertia and surface to volume ratios is also another thing to consider.
Great, thank you. So I don't see any other questions coming in. So I'll just, I think we'll end it here. I'll thank everyone again for sticking around to the end of the session. I think today was pretty obviously clear how much we needed a session to share all of this great work. So we crammed a lot in, and hopefully people will make some connections offline, and in the future we can have better discussions when we're in person. But Damien, I don't know if there's anything else you want to add before we take a break. I don't think he's on anymore, but so yeah, all right. Well, thank you everybody. And just a reminder that the offshore wind session will start back up at 1:30 today. So take a quick lunch and come on back if you can. And thank you again, everybody, for sharing all this really great work with everybody in the community. And we'll be posting the recording of the session in case people had to jump out on the website when it's all ready to go. So thanks again, everybody.
Hi, Libby. Were you trying to talk to us? Okay.
And not really. I just want to, I'm glad I'm kind of in this new, you've moved me over to the panelists. I was trying to figure out when people are presenting, I'm just wondering on Zoom, can I come back onto video and they'll see me? Because I think on my computer, maybe that's not going to happen.
So the way that we've been handling this is that when the current speaker, I think everybody has been sharing slides, right? So they will be sharing their slides from their device. And then we are going to be what's called spotlighting their video. So they will, basically what you'll see is you'll see their slides and then you'll see a little bar where their window of their video shows up, and everybody else's will basically like kind of disappear in a sense.
Right.
But
can you bring me up like at the 2-minute mark?
yeah, so what I was doing on Wednesday, I was helping with managing the spotlighting, and what seemed to work well is if you turn your video back on, and I'm watching to see that your video, because I can see in the list of participants that your video is active, and then I can re-spotlight you.
Okay.
And then you'll pop up right next, like underneath or on top of theirs. That seemed to work fairly well on Wednesday. I wasn't able to call in yesterday or this morning, but on Wednesday it seemed to be working that way, like as far as a like communication tool.
But do they, I know on my computer, if I'm on my laptop, which is where I am now, and I actually do have to present the slides for one of the people, when I put it, let's just try it.
Yeah.
So if I do share screen, I think what's happens is that I can't see anything except the slides when I'm presenting. So I'm wondering if they're going to actually see me. You know what I mean? When I pop up.
Why don't you go ahead and start sharing? Because people were seeing when
responding. Yeah. And if they have more than one,
they're back and I need to like wrap up. So, okay, for example, like Tom has his video going right now. So I'm going to spotlight him. If I can click on the right. Yeah.
So let's see. I'm like presenting now.
So do you see your video and Tom's video showing?
Oh,
yeah, I do. This is cool.
Yeah. So that's
okay.
That's basically what it's going to look like.
That
for,
person.
You know,
yeah. Okay. Perfect. Yeah.
So Liz, at the 2-minute mark, you're gonna spotlight yourself, so you pop up under there. Is that what you said you've been doing?
So what if Libby is like kind of moderating the session, then if she would like to turn her video back on when she wants to pop back up and give the kind of warning, right? Then I can just have, what I've been doing is having the participant list open so I can see the icon go from red to like white for her video being on. And then I just, right click or click on more and add to the spotlight. There is an option, there's two options. If there's already someone who's been spotlit, you can either add someone else to it, so then you'll have two people, or you can replace. Obviously you want to click the add so that, both of them are showing up.
Yep. Okay.
Yeah.
I think I got it.
And Tom, so if I was just as the other session was wrapping up, I was telling Cassie that I was happy to handle doing the spotlighting from 1:30 to 2:30 because I have a conflict at 3, so I have to get off early. But if you would like to like try it out and practice, you're welcome to do it. I'm going to be here anyway, and then you can get experience doing it if you would like to. I don't care either way.
Yeah, I might, if you're around, I might watch you do the like first few just to make sure I'm doing it right.
Yeah.
And then I can take over. And then I can take over when you have to go.
Okay, that sounds good.
Can you answer something for me? There was, it looked to me, because I've been participating for the last few days, that as a non-panelist, that I can post things in the Q&A, and I was kind of seeing back and forth. We as the panelists can see the Q&A, can't we? When or is there something weird that happens?
Yes. So the only difference with being a panelist is, well, I guess the key difference is that you cannot post a question
on the Q&A.
Yeah, but once there is a question there, you will see an option to reply, and you either answer live. So if somebody's going to
Got it.
respond, you as the moderator, if you say, hey, so-and-so, Carl, answer this question in the Q&A session, and he responds during the live, then you can just mark it as being responded live. Or the panelist can go in and respond via text to that specific question. And they can do that whether it's been answered live or not.
Right.
They just go to the answered section and yeah, so that's basically the key difference between being a panelist and being an attendee, is that as an attendee, you can submit questions, but you can't answer any, obviously. And then as a panelist, you can't submit any, but you can't answer all of them. So that makes sense.
And then the chat is just, we're trying not to use, bottom line. But some people do.
Yeah, I think some people do just, maybe not realize that there's a Q&A option and they're dropping things into the chat. The nice thing about Q&A is that it's a little bit easier to manage if there's a lot of questions, because once you mark it as being answered, it gets moved into the answered section. And so it's a little bit easier to not miss questions if they get buried in the chat, basically. That's kind of the advantage of using the Q&A.
So we can rate Carl is there, I see. So we could bring him in, the panelist. All right. Carl, did you want to, can we hear you?
Can you hear me?
I can hear you.
Great.
Do you want to, are you confident in your screen sharing capabilities, or would you like to try?
Let's give it a shot. I do not have an external monitor hooked up, which always seems to screw things up. So let's see if we can. All right. Do you see the draft version or the non?
Like, I see all the slides.
Okay.
Yeah.
So let's see if we can get to the power.
Yeah.
Full on, ready to go. Great.
Yeah.
Excellent. Okay, good. I'm good.
Good.
5 slides, Libby.
Wow. What? You have 10 minutes.
10 minutes to get in trouble. So I'm going to go with 5 slides.
Okay. Looks like you got a few backups there in case you want to cover more.
Oh, we've always got backups. That's right.
All right. Let me see if we have anyone else yet.
Elizabeth, good to see you. It's been a while.
Yeah, how are you doing?
Good. How's the Darling Center?
It's good. It's been very busy this year, which is actually like really nice.
Nice change, that's for
As
sure.
opposed to last year, yeah.
Yep. Are you in year 2 of your PhD? Year 3? Is that right?
I'm actually not a PhD student. I am just a technician.
That's not just a technician. I do remember that now. That's right.
Although I don't know. I am thinking about it. I'm like, I don't know. It could be fun. I don't know. I can't. But you're like, are you still considered a student?
I think that ship has sailed.
Okay. I'm just like that. Like, I'm interested in maybe pursuing a PhD, but I don't want to stop doing my job. Like, I don't want to be a full-time student. And so that's my kind of like.
Wait, Carl, are you not a PhD? I didn't know that.
No, I'm not. No, and I had made it through to the candidate stage, and then kids arrived.
All but dissertation, ABD.
That's right. Yep. Yep. Yeah, it is.
I almost introduced you as Dr. Carl Wilson.
No,
So
yeah,
good
no,
thing.
No, you got the wrong person here. Yeah.
I mean,
could have fooled me.
Introduce you as Dr. Wilson.
What's that?
I'm pretty sure Damien would introduce you that way. He has a tendency to do that to people.
There's plenty of other doctors in my family. You don't have to introduce me as a doctor.
Oh, wait, we have John here.
It
We
just
can bring
slapped.
him up.
Yep.
John? Are you there?
Almost. I was
Are
trying
you-
to figure out why I had no controls whatsoever, but it's because you had all the control, which makes sense.
Yeah, it's like we come down and we pull you out of the mess.
You're now in the fun crowd.
How do you feel about, do you want to practice sharing your screen? Any worries there?
Great.
Okay.
I forgot that this was on Zoom, so I'm using the Zoom browser.
Yeah,
Probably it's the same, right?
more or less.
John, are you at work?
Yes. The internet connections, because I'm plugged into the network, is a little bit more reliable here.
Yeah, good idea. Do you want to try sharing your screen?
I go to share my screen and then share.
All right. Looking good.
That work?
Yeah. Can you, John, I'm curious, can you see me also? Or
No.
Yeah, everything disappears for you.
Correct.
Yeah. So Elizabeth, there are probably going to be a few, especially if you do it from the browser where. Yeah, we were trying to figure out how I'm going to flag people, but for you, I'll just say, John.
Yeah, just shout.
I'll just
Libby,
shout.
I was able to see you when I tested my screen. So
Yeah, so I think if you're, I'm on the Zoom app thing now, but if I were on my actual computer, then I would be on my work computer. Yep, you see it. Well, good. That's a start. So, anyone else? Not yet.
So Liz,
How are you, Carl?
does
Oh,
everyone remain
Thomas.
a panelist during the entire talk?
Mhm.
You want us to shut our videos off while other people are talking?
Yeah, so my proposal is that the person who's speaking stay on video, and I'll go off, but for those for whom it's relevant, I'll come on at the 2-minute left mark. So that can be your cue to begin the wrap down. At the, John, you stay on because right after yours, we'll have a discussion period, a few, 10 minutes or so.
Yeah.
And then we move to Carl and 3 other speakers, and then we'll have another discussion section, and hopefully, John, if you're still around, you also come back on. So we'll just sort of
Okay.
accumulate people, and by the end, we'll have all the panelists up.
Can we end like around 4? Is that right? Or 3:30?
3:30. Yeah, it's kind of a tight.
Yeah.
Well, it's not as tight as some of the ones that have happened.
So I'm able to stay on for the whole thing.
Yeah, great.
So good.
Yeah, 2 hours. That's the advantage of it only being 2 hours.
Exactly.
And you should, although I don't know if you will, John, but have access to the Q&A. And the way the Q&A works for you, Carl, too, is that we can't, as panelists, pose questions in there. We can answer questions, but the audience can pose questions. If you have questions, you panelists, and we'll tell everyone this, then you can actually could do that in the chat, and I'll be trying to look at both, just because I think it will be sort of interesting to have the panelists also asking questions of each other, but we can do that, when we open up the mic and have the actual interactions also.
So the chat is only for panelists? Is that how
Well, I
this
think
works?
everyone can do chat, but they're trying to channel people into the Q&A thing, because then the audience asks the question, and then, if you're not speaking, but you see a question that's sort of relevant to what you had spoken about, you could answer in the Q&A.
Yeah, it just seems hard. Like, keeping track of the video and the chat is hard. keeping track of the video, the chat, and the Q&A is harder. I need to.
We'll do our best.
I suspect I will be just keeping track of the video during any time that I'm talking.
Yeah. Oh, yeah, yeah, yeah. I don't expect you. That's kind of my role, is to make sure that we're
I
keeping
guess they're just
tabs
trying
on
to
that.
get us even better at multitasking.
Yeah.
Putting additional tools out there that we need to learn how to
Right.
use and use at the same time as the other tools that they've given us.
One of our first COVID remote meetings with the fishing industry was on Teams, and there's a chat function there. And so all the DMR staff were kind of chatting back and forth. And finally, the commissioner just like exploded on us. And he's like, shut the hell up on these, chats back and forth. I can't keep track of them and I'm trying to talk, we're like, okay, we're going to drop you from the chat. That was a good lesson there.
Exactly. And a good solution.
That's right. Yeah. Don't want to task load people too much when they're talking on difficult subjects. Libby, are you in Maine?
No, I'm in DC right now, but I'm hoping to spend the month of January up there.
Oh, great. We had our first band of snow come through today, so it's official.
Wow. Yeah, it's chilly down here too.
Not on the coast, Elizabeth? Well, 20 miles away, you missed it.
Right, thanks. No, it's a beautiful blue sky right now.
Yeah, about an hour ago, it came pretty good.
Oh, we were like looking at the dark clouds going, what is that that's passing through? That makes sense.
Yep.
Oh God, that's horrible. We're in the same room.
Tom is also jealous about the snow.
So just remind people that all of the people who are signed up to participate in the Zoom can hear us now, apparently. So that's fine. I don't think we've said anything.
What's the participation, Ben? Oh, there's Emily.
I actually don't know. So Orla O'Brien is one of our panelists, so you can pull her in. Oh, and there's Emily. Hi.
Hello.
Great. And Annie is here. Awesome. And Annie, I'll share your slides, correct? Okay. Do you want to try, you're on mute, so.
Yeah, sorry about
Okay.
that. Yeah, that would be great. Thanks.
Okay. And Emily, did you want to try your slides?
Yeah, that'd be great. I'm curious if I can use presenter mode, or I'm going to try that.
Okay.
Let's see.
Hi, Julia. Hi, Orla. Yeah, I think we may actually have the, oh, wing. We're missing wing. Are you able to find the share screen thing?
Yep.
Okay. Oh, good.
Let's see how this works.
Hey, all, I'm here.
Hey.
You guys
Okay,
can see
that-
everything, right?
Yeah. The current and next slide, that whole thing. Oh, there you go. That looks good.
Okay. I'm going to try something slightly different because that puts it on a different monitor for me.
Yeah.
Give me 1 second.
Yeah, the trick is I'm hoping that I can just come on video as the 2-minute warning for everyone. For some people like John, I don't think he's going to see it, so I'm going to do a verbal thing for him. But for others, maybe you can try and see if you're still seeing it, any of the videos when you present.
All right. How is that?
Yeah, that's great.
Okay, perfect. I know what to do now.
Okay.
Good. Thank you.
And Emily, could you see the videos as well? Could
Yes.
you? Okay, so that's perfect. And Wing, did you want to try to present? Or are you good?
Let's see. How's that look?
So good. Perfect. Yep.
That's it.
And can you see us? Can you see your video too, or when you're presenting?
Oh, yeah. I see.
Okay. Okay. So I'll just, again, that'll work because then I can come on. And when you see me come on in the live version, you'll know that you're at the 2-minute mark.
Right. We have 10 minutes. Is that right?
You have 10 minutes and we have discussion sections. So don't feel like you have to end early for that. Also, okay, so Orla, did you want to try? And Julia?
Yep, I'll try.
Okay.
Let's see. Share. And
perfect.
Okay, so yep, that should be good.
Okay. And Julia, did you want to?
Yes, please. Let's try this.
Oh, you know, we're missing Everett.
Is that good?
Yeah, perfect. Oh yeah, we're trying the, that's like the spotlighting thing. Oh, we need Everett. We need to pull him up. Is he here? No. Elizabeth, did you want to bring Everett?
Yep, he should be already invited. He has to accept it though, so.
Okay. And Julia, the pronunciation of your last name is Stepanek?
It's Stepanek.
Stepanek.
But you are not the first, nor will you be the last to not get it perfect. So it doesn't really matter to me that much. But I appreciate you asking.
And I definitely need Everett's pronunciation. Brzezowski, maybe? Okay.
Hey,
I don't
Everett,
see the invitation.
I think you can hear me talking. So I am sending an invite that should be popping up somewhere on your Zoom screen that's inviting you to go to be a panelist. Send it a couple of times, so I'll keep kind of pinging it, and hopefully that'll be bringing it up. But I know your internet's not great if you're still out on the island.
I thought when you promote them,
They have to accept, yeah.
Yeah, and Everett, if you want one of us to do your slides, if that's going to be a bandwidth issue, then you could send them to one of us. You'll be near the end, so Liz wouldn't make sense, right? You could send it to me or Thomas.
So Libby, just so you know, I do have to leave a few minutes early from the end of the session, but Tom and is going to be here, and Cassie may be back on also during the session, so you will have support.
Okay.
How you pointed was really cool because Tom was right next to you the way you were pointing. That's just, I haven't seen the sort of coordinated video screen stuff yet. That was awesome.
She's also in the background.
Oh, that makes it easier.
Did you see Everett's note?
Everett, when I try to promote you to a panelist, it says that you'll be rejoining the webinar as a panelist. So maybe if you log out or like leave the session and then come back, you may automatically be bumped as a panelist, but I'm not sure.
And he's gone. Luckily, he's not on till the end, so hopefully we'll figure it out by then.
Okay, well, like you said, Libby, I think we have some time to work out that with Everett. So we'll keep an eye on that. Do y'all have any other questions? Everybody feels good?
Yeah, I think I'm
Thumbs up and head nods.
great.
Awesome.
How do you, is Damien gonna come back on and do a, like, we're back, or how do we?
So I know that he was had a family thing that was coming up this afternoon that he had to kind of like, he was going to be maybe calling in from his phone, but it was going to be a little sporadic and he wasn't sure about the timing. So I think if you're comfortable, I think you should totally feel free to take the reins at 1:30 and kick it off. And hopefully he's able to join us a little bit later on.
Sounds good. So I'll stay on video. And others, you can go off, stay around, but you can go off. And maybe, John, you can decide if you want to come on or off right now.
Yeah, so I went ahead and spotlit Libby. So you're going to be the primary video, and I'm going to turn my video off throughout the session, but I'm still going to be here. So yeah, we'll help manage it for you.
Sounds good. We'll wait until 1:30, which is a few minutes.
Sounds good.
I just tried to promote Everett, but it gave me the same warning again.
and I actually don't think I ever got an invite. I just like popped up. So. All right, it's 1:30. Time to get going. Hello, everyone. Welcome back to the final session of RARGM. And this is our. Look, we're getting John. You might want to mute because I think I'm getting some feedback from you. Okay. This is our final session focused on wind energy development in the Gulf of Maine. With the following sub questions. What are the potential science and society impacts if wind energy comes to the Gulf of Maine? And what might happen to fisheries, communities, or monitoring, and how might we adapt? My name is Dr. Libby Jewett. Although my day job is serving as director of NOAA's Ocean Acidification Program, and I actually had the pleasure of presenting at RARGM a number of years ago on ocean acidification, I'm currently serving a 4-month detail with NOAA Northeast Fisheries Science Center, assisting their offshore wind team with research priorities around wind for the Gulf of Maine. I'm looking forward to the presentations today, as I myself am still getting up to speed on the topic, and the people who are going to be presenting today are going to fill me in on many of the issues. I think we have a great lineup. Just a few housekeeping items. The speakers will come on video when they present their slides. I'm desperately going to try to keep people to their allotted times, and will come on video myself at the 2-minute mark. We have 3 discussion periods, 3 10-minute discussion periods. The first after our keynote presentation, 2 more. For the second 2 panel sessions, speakers will come back on video to answer questions, and our panelists will also be answering questions in the Q&A section of Zoom. And so we encourage you, our audience, to be asking questions in the Q&A. We'll do some answering as we can in real time there, but also may elevate some of those questions for all the panelists when we have our discussion. So we only have 2 hours and a lot of material to cover. So without further ado, let's move to our keynote presentation. So Dr. John Hare is the Science and Research Director of Northeast Fishery Science Center. He's activities related to NOAA Fisheries mission in the Northeast region, including fisheries, aquaculture, protected species, habitat, and ecosystem science. He also works to build regional science collaborations to address complex issues in the marine environment, including climate change, offshore wind development, and watershed science. So take it away, and we'll have some questions. You have the opportunity to ask John questions after his presentation.
Great, thank you very much, Libby, and thank you all for joining us today. I'm going to talk about some of the offshore wind, fisheries. Got the wrong button there. Okay, can you see my screen, Libby?
Yes.
Okay, great. Well, thank you again for being here. I'm going to talk about offshore wind, fisheries, and wildlife, and the science that we need to support coexistence among these different ocean uses. Outline, just to give everybody an idea of sort of where we're going, a little bit on the rapid expansion of offshore wind in the region, then discuss some of the interactions between offshore wind, fisheries, and wildlife, and then present some of the key challenges and opportunities from my perspective. So rapid expansion of offshore wind. This is a map from Bureau of Offshore Energy Management in August of the different areas that have been leased, and then the different projects which are in various stages of development. This project here, Vineyard Wind, has been approved through the permitting process, and that will begin construction in either 2022 or 2023. And then all of these other projects are in different stages of environmental review. So there's a lot going on in the Northeast region, but there's also a lot going on nationally. In October, the Department of Interior, Bureau of Offshore Energy and Management announced the intent to lease additional areas in the New York Bight, and additional areas in what they're calling the Central Atlantic, but which was really, I would call the Mid-Atlantic. They're talking about leasing areas in the Gulf of Maine, in the Southeast US, in the Gulf of Mexico, off of California, and off of Oregon. And the intent is to have those additional areas leased by 2025. So, what we have been thinking about as sort of a Northeast focused issue is now very clearly a national issue. The president announced in March the administration goal of 30 gigawatts of offshore wind in the United States by 2030, while protecting biodiversity and promoting ocean co-use. And this clause is very important. It often gets left out of the goal, but it's very important because this is really where NOAA's role comes in to offshore wind. It's offshore wind development while protecting biodiversity and promoting ocean co-use. And another important part of this goal is the idea is to unlock a pathway for 110 gigawatts by 2050. And so thinking about that goal, it's offshore wind and fisheries and wildlife. And the conversation here today, what we're going to hear a lot about is how do we use science to inform the coexistence of these different ocean uses. Much of what I'm going to talk about today is part of a special issue on understanding the effects of offshore wind energy development on fisheries. This was in the Oceanography Society Oceanography journal. The volume number and number are there. These are available complete PDFs, and so I encourage you to go check out this special issue if you want more details about many of the topics. So when we think about wind energy development, the steps of wind energy development, this is how we can begin to consider what are the interactions with fisheries and wildlife. There's the pre-construction phase, which is going on now. That is largely site assessments, geology and geophysic acoustic surveys, and developments of the infrastructure to support offshore wind. There's the construction phase, which, as I said earlier, the Vineyard Wind project has been approved, so the construction phase for that project begins soon. And then the construction for the other 17 projects in the Northeast area will also be beginning. And then there's construction for the additional lease areas at some point in the future. The operation of these facilities, of these developments, is typically 20 to 30 years. It would be a permit to operate for 20 to 25, 30 years. And then after that, these facilities are decommissioned. What decommissioning means at this point is premature, but one can imagine that the turbines are then themselves removed, and the areas are returned to sort of pre-development conditions. So when we think about these 4 steps of wind energy development, we can infer a number of impacts on fisheries and wildlife. There's the acoustic survey, the geology and geophysical survey, which emits noise to profile the bottom of the ocean. And then there's the construction noise, pounding very large pylons into the sediment to hold a wind turbine, which is as tall as the Eiffel Tower. That's a fair amount of construction noise. There's vessel traffic, the number of vessels to support the construction and the operation. So there's the vessel traffic impacts. There will be changes to the seafloor, to the water column, and to that sort of air-sea interface. And those changes will be because of the construction of these fixed turbines or floating turbines. That construction will create sediment suspension, resuspension, and deposition dynamics. It'll also impact the oceanography. We'll talk a little bit more about that in a couple of minutes. The cabling, each turbine is connected to the next turbine because they're generating electricity. They need to get that electricity to shore. So they're all connected. And then there's a cable that goes from the development to the shore. Those cables will have electromagnetic fields, and those electromagnetic fields will interact with marine life. There's the lighting issues and the navigation issues around these turbines. They need to be lit for maritime navigation. The lighting then creates sort of different habitats in the air-sea interface zone, which could either attract or repel different species depending on their behavior. And then we have this sort of habitat conversion. We're converting water column habitat and relatively smooth bottom habitat into what is in essence an artificial reef that extends from the bottom of the seafloor through the surface of the ocean. So we have those wind energy development effects, and then we have our trust resources, we have our fisheries, we have our wildlife, which we have legislative requirements to maintain sustainable fisheries and to protect and recover wildlife. Those requirements, those legal obligations have components of ecosystem-based management and habitat protection in them. So our trust resources are these fisheries, wildlife, ecosystem, and habitats, and these construction and operation activities will affect those. There'll be exclusion of some fishing activity. Again, we are altering habitat by building the turbines, but that is in essence creating new habitat. They're the biological effects of the noise and of the electromagnetic field, the biological effects of the increased vessel traffic. There'll be exclusion of some species and attraction of other species. There will be changes in behavior, changes in feeding behavior, feeding conditions, changes in socializing, changes in reproduction. Then there's those broader ecosystem changes, which we talked about momentarily a moment ago, is there will be changes in the oceanography, which will have changes in prey, which will also have changes in habitat. And then there is this impact on our ability to conduct surveys and assessments. Our traditional survey techniques will not work or may be impacted by the development of wind energy. So what I'm going to do next is just go through and sort of look at some of these interactions individually in a little more detail, sort of like vignettes. I'm certainly not going to go through all of them. But in terms of thinking about marine mammals, and there will be a couple of talks later on which will focus in more detail on marine mammals, there are the construction effects, which include construction noise, vessel traffic, and ecosystem changes. And these effects are going to be important. The slide shows the visual sighting locations of North Atlantic right whales from January 2010 to November 2019. And then you can see the orange inlays of the wind energy development areas. And there have, during these past 10 years, been a number of visual sightings in wind energy areas. So there is going to be an interaction between wind energy development and North Atlantic right whales. And that managing, understanding, mitigating that interaction is going to be very important for the protection and recovery of that endangered species. And that is but one example of many of the interactions with marine mammals. The sound, sound will have impacts on marine mammals, but the pre and during construction sounds will also impact a number of the fisheries resources. There has been experimental work which has been completed, which show that squid will exhibit a startle response to replicated pile driving noise. Similarly, black sea bass respond to replicated pile driving noise so they can detect the noise. There will also be effect of the geological and geophysical surveys on animals in those areas. So, there's evidence that animals hear and respond to the construction noise, one of our challenges is trying to understand what the population scale impacts of those responses will be. Similarly, some fishing activities are going to be excluded from these wind energy development areas. Mobile gear, towing a trawl behind a fishing vessel with wind energy turbines and electrical cables connecting them. So there'll be exclusion of some of gear types. There will also be exclusion of larger vessels. Smaller vessels may still be able to operate in these areas. And then the ability for vessels to transit through these areas will be impacted as they go from fishing grounds to port. So there will be a number of impacts on an exclusion of some fishing activities. Interestingly, in Scotland, the wind energy developer Equinor has started working with Marine Scotland, sort of the NOAA equivalent, to develop and sort of think about what fishing methods could be used in floating wind energy areas. And so this idea that some activity is going to be displaced, but what activities could be developed that would be permissible. And going to the habitat alteration example, I like to think of it, or I think it's useful to think of these turbines as essentially just very large artificial reefs. They will extend from the bottom all the way up through the surface. And some work which has been done by Elizabeth Mithrada, who works at the Science Center, sort of analyzing data from a number of studies, has shown that fin fish abundance increases in wind farms. And this increase is, one could think of it as sort of that artificial reef increase in fish abundance, whether it's an increase because it's pulling fish from other areas, or if that increase is an increase in productivity of the population, is a question which remains. But certainly the habitat alteration by putting these wind turbines into the ocean will cause changes in how fish, shellfish, and marine mammals, sea turtles, seals use these areas. From my perspective as an oceanographer, one of the sort of intriguing interactions is this interaction between the turbines and then the sort of the seafloor, the water column, and the atmosphere. And so these pictures show different just illustrations of those interactions. That top picture shows the sediment plumes behind an array of wind turbines in the North Sea. And when I look at that picture, it makes me think of an island mass effect. And so you have an array of 80 turbines. In essence, you have 80 small island mass effects in the wake of each turbine. That picture shows sediment, so that implies that there's sediment resuspension in the wake. Then the lower panel shows very similar sort of figure, but it's showing that same sort of increase in disturbance in the atmosphere. So these turbines are going to increase the turbulence in both the ocean and in the atmosphere, and that will have impacts on primary and secondary production, which will then link into higher trophic levels. And there will also be impacts on benthic habitats with this suspension and resuspension of sediments. The other piece which we've been very concerned about or working on hard at the Northeast Fisheries Science Center is this effect of wind energy development on our surveys and our assessments. The figure shows one of our survey strata. So we just completed our bottom trawl survey. We'd go into that strata and sample it, and then based on the statistical design, develop and combine information among strata and develop an index of the, say, the number of summer flounder in the Northeast US shelf in the fall of 2021. Now we're basically excluding our survey activities from 37% of that strata, so it's interrupting our survey design. It's also excluding our vessel from those areas. These areas will also likely exclude some of our aircraft surveys. There's the habitat effect, and so wind energy development, as we talked about, there's going to be an increase in finfish abundance, presumably an increase in finfish who are attracted to structure. How do we sample those new habitats when our current survey methods are designed to sample more sort of unconsolidated sediment habitats? So we have that habitat effect. And it's not just sort of interrupting our science. Our surveys are the first piece of our scientific enterprise, which provides scientific advice for the assessment and management of more than 80 fisheries species and marine mammal species or stocks in this region. So we're sort of working with the Bureau of Offshore Energy Management to develop a strategy to mitigate the impacts of wind energy development in the region on our surveys and on our assessments. The last piece, and this is sort of the even a larger scale than what we've been talking about before, is what are the cumulative impacts? A lot of effort on what are the environmental impacts of vineyard wind, which is down here, and then what are the environmental impacts of Mayflower wind, and the environmental impacts of Beacon wind, the environmental impacts of Vineyard Wind 1, the environmental impacts of South Fork wind, the environmental impacts of Revolution wind, the environmental impacts of Sunrise wind, the environmental impacts of Bay State wind, the environmental impacts of Vineyard Wind South. But as these developments continue to proceed, the question of what is the cumulative impact of all of this development on the marine ecosystem. These projects are going to be constructed over the next 7+ years. They will be in operation for the next 30+ years. It is likely that there will be more projects to come. So we can think about what are the project level impacts, but we need to think about what are the cumulative impacts of this development on our regional fisheries, on our regional wildlife, and on our regional marine ecosystems. And that's a real challenging question to get at. So I just quickly walk through some vignettes. And I think it's useful to sort of try to synthesize that information. We have pre-construction, construction, operation, and decommissioning. And each of those activities is going to have impacts on fisheries, on wildlife, and on ecosystems. And our job as scientists is going to be to understand those impacts, evaluate how they are affecting our ability to maintain sustainable fisheries, to protect and recover wildlife, and to have healthy ecosystems. And so the big question really is, what are the effects of wind energy development on fisheries, wildlife, and ecosystems, including human communities? And can these effects be avoided, minimized, or mitigated? And when I look at that question, it's a very simple question, but it really is a grand challenge. So how do we address that grand challenge? And it's challenging for several reasons. One is we're trying to address the challenge, and wind energy development is already progressing. So like many of our problems, we are working on it while it is ongoing. The pace and scale of development, the pace continues to quicken and the scale continues to grow. So that makes it a challenging problem to work on. And I work for NOAA Fisheries. Fisheries in the Northeast and the New England area are controversial. It's a very difficult topic to work in. Marine mammal protection, recovery of, say, North Atlantic right whales, a very controversial and difficult situation. Maintaining healthy ecosystems, developing ecosystem-based fisheries management, a complex, difficult problem. And now we have offshore wind and the interaction of offshore wind with these other complex problems. The scale of the problem is a challenge. And how do we address this big, grand challenge of a question of how to science to support coexistence? Also in the Gulf of Maine, the conversation is around floating turbines.
Hey, John.
Technology, yes.
Just another like 2 minutes or so?
Got it. Yeah.
Okay.
Floating technology, it's a new way there's no floating technology deployed in the US. So how do we transfer what knowledge we have around fixed technology to floating technology? And some of our opportunities, how do we do this? We are developing collaborative and cooperative relationships and agreements between NOAA, Bureau of Offshore Energy Management, Responsible Offshore Development Association, which is a fishing industry representative. We're going to hear from Annie Hawkins here later today. The other opportunity is we have built, the US, Rhode Island has built offshore wind farm, 5 turbines. And so there's a lot of lessons that we can learn about their success. Opportunities, offshore wind has been deployed in Europe for 20+ years. And we have worked with the International Council for the Exploration of the Sea to develop a working group to look at offshore wind development and fisheries across the Atlantic, working to learn lessons from the European experience, and then sort of translating lessons from the North American experience to Europe. And then the sort of our last opportunity really is innovation. You know, new fishing methods, new management approaches, new survey approaches, new science, scientific designs, ocean observing, and creating new partnerships. And I'll end with sort of I guess it's a hope. You know, we have all of these different developments, all of these different projects, all of these different topics, and the incentives for us will favor disaggregation, dispersion, and duplication of our science effort. Much of our science is reductionist. We like to look at small, definable problems. There are multiple funding sources which pull us in different directions. Much of our funding is project level, 2 to 3 year, write a paper or report, and then move on to the next project. And much of our science is disciplinary. And I think what we need to do is work against these incentives and really focus on working together to achieve that grand goal, that address that grand challenge of offshore wind and fisheries and wildlife. And I think there are structures which are developing in the region, Responsible Offshore Science Alliance and Regional Wildlife Science Entity, which we'll hear more about today, to really help us take all these different pieces and put them into a regional framework and consolidate our understanding to address this grand challenge. So thank you very much. I'm happy to answer any questions.
All right. Thank you, John. And I think ending on that note of coordination and thinking, you know, having sort of larger, more integrated approaches was a perfect way to end. There is one, I'm just gonna, so folks, feel free to go ahead and put your questions in the Q&A. This is actually a good one, although I'm not 100% sure you'll be able to answer it. Can you talk a little bit more about the 20 to 30 year lifespan for the turbines? After so much effort to get them out there, why would we decommission them so quickly? So I don't know if you have thoughts on that.
Yeah, I mean, that's a tough question because I'm not a wind energy, I'm not a wind energy developer, I'm not an engineer. You know, as I understand the situation, you know, the permit for operation has a duration. And those permits are, I think that what they're talking about is a 20, 25, 30 year duration. And so that's the federal permitting process. Presumably a company could either come back and say, we want to extend our permit, and then they're, you know, so then that extension would be there, or we want to decommission the development. So that is, you know, out at the edge of where we are really working and thinking now, is that decommissioning step.
Okay, looks like we got another question here. You stated that fishing, transit, and large vessels would be excluded from wind areas. Could you provide more detail on who or what is doing the exclusion? US Coast Guard has not taken this position yet. You're on mute. You're on mute.
I guess I got muted after every question. There's no formal exclusion, correct. So anyone can operate within wind energy areas. And I think it's going to come down to safety at sea. Many of us have been at sea. Boats are not as nimble as cars. And so working on a boat with a bunch of metal at 1 mile spacing would be daunting for any captain. And the larger the vessel, the more daunting the challenge. And the worse the weather, the more daunting the challenge. So there's the safety at sea piece. And then there's also this question, which I don't think I've heard an answer to, but regarding insurance. If an accident were to happen, who would be liable for it? And would an insurance carrier cover it? And so there's the liability piece of it, as well as the safety at sea piece of it. So yes, no one is excluded, but there are those practical concerns that I think everybody's going to have.
All right, so we have another wildlife question, or maybe not another, a first wildlife question. Could you go into more detail into the impacts of wind farms to marine predators? Will they benefit from artificial reef effects, or do you think there will be a decline in these species?
I mean, that's a tough question to answer without studies to understand. And I think it probably also depends on the specifics of the marine predators. One could imagine with an increase in sort of fish abundance around wind energy, around these turbines, that you could see an increase in predators. But if it, there could also, you could also imagine some of the larger predators avoiding the areas because there's just too much in the water. So I think it's an excellent question. I think it's something that we need to really start trying to address sooner rather than later. But presumably an increase in fish abundance around these areas would also lead to, in general, an increase in large marine predators that eat fish.
So we have one more question, and then we're going to move to our next speaker. And the good one, from our experience with putting oil rigs in the Gulf of Mexico, there must be some lessons learned on the impact of these structures on biological productivity. Are we tapping into those observations?
Yes, so that is work which is starting. So Northeast Fisheries Science Center, we work from North Carolina to Maine. We have our counterpart, Southeast Fisheries Science Center, that works from North Carolina to Texas. And they do have experience with the offshore oil industry. And so that is, we're learning from that. And then, as Celia says in the chat, there's also a lot of wind energy development that has gone on in Europe. And that's part of the purpose of that ICES working group, is to exchange knowledge from the Europeans to North America. And then, to the questioner's point, there is also an exchange of knowledge from the Northeast and the Southeast. Of course, wind energy development is also going to start in the Southeast, assuming those lease areas go through. So we've been working closely with our counterparts in the Southeast.
All right,
thank you very much.
Thanks, John. So you'll be back on later for our next discussion sessions, but we're going to move to our next speaker. So Carl Wilson is our next speaker, has worked with the Maine Department of Marine Resources for over 20 years. The majority of this time he served as the state's lead lobster biologist. 6 years ago, Carl was promoted to director of the Bureau of Marine Sciences, where he oversees coastal fisheries, monitoring and assessment programs. Carl was pulled into offshore wind discussions this past year as the department was asked to support the governor's energy office application to BOEM for the research array. Thanks, Carl. Take it away. You're on mute. Yeah.
Just a quick sound check. Libby, can you hear me okay?
Yep, you're great.
And see the screen?
Yes.
Okay, excellent. Thank you. It's a pleasure to be here today. Again, I work with the Department of Marine Resources. And last winter, we found ourselves in a very different role for us, where we were asked to help the governor's energy office move through siting considerations and make recommendations around a proposed research array that was eventually submitted to BOEM in the early fall of this year. The concept is approximately a 16 square mile research array with 10 to 12 floating turbines. John mentioned those at the end of his talk. This would be the largest floating farm if this is approved and moves forward, and would be building on earlier University of Maine development work off of Monhegan Island that I believe should be in the water around 2024. Some of the constraints that we are looking at, there were two anchors on the mainland as far as interconnection points at Maine, the former Maine Yankee site in Wiscasset, and the Wyman station off of Cousins Island. Those defined kind of how long the power cord could be, which was about 40 miles, which was the outer limit of our area of interest. And then the inner limit of our area of interest was approximately 20 miles based on siting considerations from shore. So with those broad constraints, we work through a number of different layers that helped us to try to characterize what the existing use was, what were additional constraints to potential siting in these areas, and really identify where the gaps were. And ultimately, our goal was to try to minimize the impact on the environment with a potential recommendation for a site, and minimize the impact on existing users such as fisheries. What we found, and John mentioned this as well in his talk, that we landed ourselves in an extremely contentious conversation. The fishing industry was united in their opposition to this work. And we, again, kind of had to work through some very difficult conversations and do some very intense listening. And I think we at the department, and I'd also say now with the governor's energy office, has a much deeper understanding of what some of the potential impacts and how this area has been used in the past. So again, just very briefly here, we touched on, the constraints: recreational fishing, herring fishing, ground fishing, lobster. You may have heard about this fishery in the Gulf of Maine. It's a pretty big one. Has very limited spatial data for the impact. And so we really needed to go deep into better our understanding in this area, 20 to 40 miles offshore, which is a little bit further offshore than we generally go for our sampling programs. Bathymetric maps provided the basis for all of our conversations, and they were the single most unifying tool to have conversations either with the fishing industry and or the governor's energy office and developers to try to understand and find an area that was suitable. One thing that we did that was slightly unique was we tried to provide additional information than what was just generally publicly available. And this was really to try to get a context what was out there. And so we did an interview process where we went out and gridded out the large area of interest, which is about 770 square miles, and tried to seek out interviews in a variety of fisheries to try to get an understanding of what the intensity of use was in this broad area of interest. We had tried to cover all areas, and ultimately we were able to have 26 constructive one-on-one interviews that I think greatly increased our knowledge and increased the quality of the product that we, our recommendation that we move forward to the energy office. Our narrowed area of interest is in the kind of right-hand corner, or the right-hand portion of the large area of interest. It takes 770 square miles down to about 50 square miles. And essentially what we found was between the existing constraints, whether it be Department of Defense layers or buffers around shipping lanes and existing fishing activity, is that there is no way to site even a 16 square mile area within this large area that does not impact somebody. And really by choosing an area that minimizes the impact, it actually targets, or the default is some of the lower activity targets a particular user group, such as some of the trawl gear or the trawling activity in this deep mud habitat that was recommended. And in the end, I think Wing is following me with this. I don't think he'll be talking much about the research array, but I think there was a very surprising and very interesting kind of synergy between the wildlife and fisheries issues, that areas that had, deep or high use for fisheries also were an area of high concern for the wildlife. And so there was a lot of synergy between the thoughts on different groups on what can be used and what should not be targeted. So a little perspectives on some of the research needs, just to finish up here. We had our goal was to try to eventually work towards a 16 square mile area, much smaller than 100 plus mile area. And so I think we were able to attempt to thread the needle as best we could in our recommendations. But some of these mitigation, you're just not going to be able to avoid large scale impacts on existing users if you are trying to cite a much larger site like we're seeing down in southern New England. So some perspectives on research needs. Totally agree with just about everything that John said in his keynote address. We felt that bathymetric and habitat maps focused our conversations when we were talking about site selections. There were some fisheries that had little or no data spatially, and even those that did have spatial data, you needed that context to understand truly what that meant by seeing a heat map on the map. And then finally, I think we knew and now understand even more so that there is an incredibly deep connection between the fishing industry and place. Location, location, location means everything in fishing. Every area has a story, and I think it's really important that before any conversation is happening around specific sites, that you try to understand what the heck is going on. And it's not just what's gone on in the last few years, it's what's gone on in the last 50 years. That's important. And I think it really deepened our understanding and improved the result again. And then I do think from a wildlife and a marine resource perspective, the design of existing surveys are kind of mismatched to providing really detailed and site-specific information that we were yearning for last winter. Finally, post-construction or pre and post-construction surveys. We've kind of come to the conclusion that a gradient-based approach of high-intensity surveys might offer the best reflection on the impacts or potential impacts of development. We're also very interested in assessing the impacts beyond the immediate footprint of the project. And that is, we've been listening to some of the discussions in southern New England, specifically around Block Island. And it's clear that the fishing industry has observed changes in that area that are beyond the level of detection that the mandated surveys were required to do. And so it'd be nice to match those observations with data as well. And then we're also looking that longer sampling timelines are really necessary beyond what is currently required by BOEM. And I think that's in this case with the research array and really trying to inform large scale development potentially in the future. I think if we can push anything, it's that we need more than just a couple of years to really understand what's going on in the site. And with that, I will end. Thank you.
All right, thank you, Carl. We're going to move to our next speaker, and after the next 3 speakers, we'll have a discussion section. So please put your questions in the Q&A. So our next speaker is Dr. Wing Goodale, who is a senior science director at Biodiversity Research Institute and adjunct research faculty at the University of Maine. His research is focused on the interaction of wildlife with offshore wind energy. Goodale has in-depth knowledge of the cumulative effects, offshore wind farm, wildlife literature, and environmental assessment. So I'm going to turn it over to you.
Thanks, Libby. Just a thumbs up that you can hear me and see my screen.
Both are good.
Excellent. Thank you. Thank you all. And it's great to just follow from Carl here because my presentation is going to step up to sort of thinking about the Gulf of Maine and siting for offshore wind. I am Wing Goodale, Senior Science Director with Biodiversity Research Institute. I'm also working as a co-chair of the Environment and Wildlife Working Group, which is a part of the Maine Offshore Wind Roadmap. So I'm going to talk a little bit about the roadmap and then a little bit about the working group and what it has accomplished so far. And of course, my slides are not, there we go, going. So the Maine Offshore Roadmap, for those of you that are not familiar, is an initiative by the Governor's Energy Office, and it is to develop an economic development plan for offshore wind industry in Maine. And the idea is to build from the state's history of planning and research, and then to ensure that offshore wind is sustainable and conscious of coastal heritage and existing uses and the environment. The structure of the roadmap process is led by the Governor's Energy Office, and there's an advisory committee, which is made up of the Governor's Energy Office, plus working group co-chairs, and then other public, private, and non-governmental leaders. So the working groups, there are 4 of them. We have supply chain workforce, ports and marine transportation, renewable energy strategies and markets, and then fisheries and environment and wildlife. And there's a really strong connection between the work in the fisheries group and the environment and wildlife side. So the overall timeline here is really a 4-phase process, and we're in phase 1 here. And this is to develop initial recommendations this fall that will go to the advisory committee and then eventually up to GEO's office. In phase 2, we'll be refining and consolidating recommendations based on the public and stakeholder feedback, and then finalize the roadmap content next fall, and then next winter into the following year, really start to get the results out to the public. Throughout this process is going to be ongoing stakeholder public engagement and communication through multiple different venues here. Okay, there we go. Sorry, I don't know. Sometimes it doesn't want to change screens for me. So anyhow, to the Environment and Wildlife Working Group. The scope here is to identify best practices, data gaps, and research needs to avoid and minimize and mitigate potential adverse effects of offshore wind on wildlife and the environment. So it is organized as follows. There's a facilitator, Laura Singer from CBI. There are 2 co-chairs, myself and John Perry from IF&W. There are 20 members representing state agencies, including DMR and IF&W, Fish and Wildlife Service, NOAA, NGOs, and universities. The deliverables are pretty big, and there's a lot for us to accomplish. First and foremost is working on siting criteria, again, on the scale of the Gulf of Maine, to identify areas where there might be greater conflict between offshore wind and wildlife and the environment. Then consider monitoring needs, and monitoring being considered both on what is needed to help support the siting criteria and also looking forward to pre-construction and then ultimately understanding potential impacts. We're also going to be looking at best management practices. So what can we do out of the gates to reduce potential impacts, review the policies of the state to see if there are any adjustments that need to be made in order to set up the state to succeed in offshore wind, and then also considerations about the research consortium. And obviously there's a lot to cover here, so we won't get to it all. So we'll be also considering what needs to be done in the future. So the phase 1 that we're currently in is working on the siting criteria and considering monitoring. So emphasize draft recommendations. So these are actively being worked on by the group right now. There's a Google Doc, and in fact, there's sort of emails going back and forth right now about this and adjustments being made. But this is where we stand, and these are not necessarily in order of priority. So one thing that's certainly come up is the importance of understanding federal consistency and the Coastal Zone Management Act, and how can the state of Maine be best situated in order to consider projects out of state waters in federal waters through the Coastal Zone Management Act. The second recommendation is a mapping exercise using existing data. And there's a level of urgency around this, as we've seen already discussed in this session, that BOEM has identified the Gulf of Maine as an area for wind energy areas. So the idea with a mapping exercise is using existing data while we recognize that there is data deficits in many different areas, but use the best available data that we have, aggregate that, and create some initial map products and initial maps. Have a workshop with fishermen and scientific experts in the Gulf of Maine to really take the knowledge here in the state and refine those maps, and then update those maps as we go forward, and hopefully with new data that's being collected. So the question is, as you start to develop a map like that, what are the criteria would you use to identify areas that have potential higher conflict areas? And so this is an ongoing list, and really at this moment, we're getting edits in right now. So please understand that this is very much in the draft form. But overall, you are looking to avoid important foraging habitat for marine birds, areas between coastal islands where migratory bird and bat pathways often occur, site projects as far from shore as possible. And just a note on this, distance from shore and wildlife relationships are very strong down in the mid-Atlantic because of the geography of the Gulf of Maine and its oceanography. These patterns don't hold up as strongly here in the Gulf of Maine. However, for terrestrial migrants, there is going to be some level of use of the coastal area that's going to, for some species, drop off as they go offshore. So moving offshore would potentially reduce some of those conflicts. Avoid areas used by listed species, areas that support aggregations, transport and aggregations of later stage Calanus copepods, areas in locations with high productivity. And this is a theme throughout, and just to the process went through with the research array that Carl talked about. These very basic oceanography pieces that are going to drive areas of high productivity are going to be really important. And then finally, known spawning and feeding aggregations of high priority managed species. And one conversation that's just constantly ongoing is that we need to prioritize. So how do we do that? And it's one thing to put these down as the sort of the key areas to consider criteria. It's going to be quite another to figure out thresholds of areas that have the greatest importance. The other remaining recommendations here, and I think I need to get going here, is to conduct coordinated below and above water regional surveys, bathymetry and sediment mapping, and conduct tracking studies. And one reason tracking will be important for some species is that surveys, by the nature of them, are going to be snapshots and will not capture wildlife, birds in particular that are flying at night. And for rarer species, it'll be more challenging. So listed species, tracking listed species and nocturnal migrants is potentially very important here. One last point here is that there's an overarching theme that the Gulf of Maine is changing due to climate, and that the data we have available to us represents what we know from the past, and new survey efforts will represent what is happening now. But we also need to consider how the Gulf is going to change in the future as we consider siting. And so with that, I will wrap up and hand it back to Libby.
All right, thank you, Wing. And there is a question in the Q&A for either Carl or Wing. So if you guys want to go in and answer that, feel free, or we'll deal with it when we get to the discussion section. So we're going to move on. Our next speaker is Annie Hawkins, who is Executive Director of the Responsible Offshore Development Alliance, a coalition of fishing industry associations and companies that are committed to improving the compatibility of new offshore development with their businesses. Prior to co-founding RODA, she provided government relations support to a variety of fisheries clients in Washington, D.C., and previously held positions in fisheries management and regulation. And I am going to be sharing my screen, so let's make sure I do that.
Libby, I apologize for the bad camera angle. I'm at the Pacific Marine Expo in Seattle, and it's kind of a weird hotel, so hopefully you can see me okay. Yes, so thank you so much for having me. I'm really happy. I haven't been to a RAWGRAM conference in years, so I think this is a great session, and it's great to be back. A little bit about RODA. So we are, as Libby said, a membership-based coalition of fishing industry associations and companies. All of our members are either harvesters, individual vessel owner operators, all the way up to big vertically integrated seafood companies and shoreside supply chain companies like marine electronics, stuff like that. We formed almost just about 3.5 years ago now in New England in the mid-Atlantic, specifically in response to the concern about the growth of offshore wind and its interactions with fisheries and how little attention that was being paid since really the outset of the offshore wind program. So within 3.5 years, we have about 200 members, where that makes us one of the biggest fishing associations in the country already, and spread to the West Coast and now into the Gulf of Mexico as well. So next slide. Oh. Thank you. So I bring this slide back once every few years just for fun. It's not fun though, because it doesn't ever get in. Here we are. It's this came from AFS back in 2018. And I think it's hard to talk about fisheries and offshore wind without understanding a little into these efforts, particularly the science efforts, but also obviously just overall planning. So every state that's active in offshore wind, even in the Gulf of Mexico, is very different in every state. We obviously just shared from the environmental side what the state of Maine's doing. There's a similar effort for fisheries in the state of Maine. Massachusetts has a working group. New Hampshire is doing sort of more individual outreach, but still quite a bit. And then of course the federal agency efforts, comment periods, regional association period, comments periods, conferences, all these things. And developers, prospective developers, leaseholding developers, all doing their own science and doing their own interactions with fishermen. And it is very, very overwhelming. And I think Dr. Hare spoke to this really well, that this is sort of incentivized to not be coordinated. And so we hear a lot, every day you can look and there's meetings about fishing and offshore wind, and it's in the newspaper almost every day, and it's something people are talking about, but it doesn't really feel like we're coalescing around any common principles or any common plan for how we're going to get better information and really plan more effectively and bring these two industries together more effectively. So next slide, please. So this is a hot mess slide. Yeah, it's a hot mess because there's so much of it. So, but really what this presentation is about today is what are fishermen asking, right? What are the hypotheses we're hearing from the fishing industry about the impacts to fishing and to the environment from offshore? Some early run results. We are in the middle with Rhoda of putting together a research priorities report that is specifically prioritized by fishing industry and the questions that they have to. This is not that report, but it's probably a little teaser of that report that'll come out hopefully in the next week or two. So, the environmental impacts are obviously a huge question. Whether it's fixed or floating wind, slightly different scenarios that we're looking at. Obviously, they're also all location specific, but with regional overlap. And I'm not going to read through all these lists. I think it makes sense just to hit on sort of the headings of what we really think about and what we're hearing about from the fishing industry. Obviously, the sort of pure environmental impacts, if you will, to fish stocks and fish life histories, socioeconomics, which is an incredibly complex field and bucket of things to look into here. And then I've got 2 more slides of categories. Yeah, so, and then fishing regulations and marine management. So how will this very, very different use of ocean space impact how influence, impact, to whichever word you want to use, how we're actually managing fisheries, right? There's all sorts of things that'll be affected. To my knowledge, no fishery management council in the country, nor NIMS, is looking yet at actual management regulatory changes for fisheries to address the offshore wind. That is certainly coming, and that's something that, as we know, is going to take a really, really long time to do at all, much less to do it right. Safety, huge issue. Probably the number 1 issue. We always say safety's first, right? There's a whole lot of ways that in a 10-minute presentation we can't get into about how safety comes up in these conversations about offshore wind, but we've written thousands of pages on this, so you can find it on our website too. Then questions about the technology itself. And I saw, some of these questions came up in the chat about decommissioning, for example. Fishermen are asking, what can we do with the technology here to try to design these things so that they are more accommodating to our existing practices? Can we put them in deeper water than we're looking at now? Is it true that the blades are going to fall off? Is it true the blades are going to ice up and throw ice? there's a lot of, is it true going on, right? Or if it is true, can we do better? Those conversations of all of these buckets, it's this technology one that I think is getting the least airtime with regard to fisheries, right? There's so much going on with technology, with offshore wind, but we really are not including fishing industry and fishing experts in those conversations pretty much at all. And then finally, of course, transmission. We're not only talking about turbines here, we're talking about a heck of a lot of cables, both floating in the water column, cables, mooring lines, as well as the ones to shore. And then, of course, the cumulative impacts issue, when you have this whole universe of different effects, how do you even begin to look at those cumulatively? So this is incredibly daunting. So next slide, please. What do we already know? Not as much as we would like from our perspective, certainly. A couple years ago, Lisa Mathrato, who I think is on this webinar, did a meta-analysis looking at peer-reviewed studies of finfish abundance at offshore wind farms in Europe. Found 11 peer-reviewed articles in 2019 in existence at the time. Obviously, there's been a few more that have come out since then, but it's not something that, you know, when you look at 15 to 20 years of experience with this in Europe, we don't have the peer-reviewed literature that you would think we might, or that you might ideally like to have after that long of a timeline. The European Parliament came out late last year and said there's a clear gap of economic and sociocultural impact assessments in Europe about, you know, specific to effects to fisheries. So we're really at square one here, trying to figure out what we can predict and how we can monitor, measure that, and how we can adapt to it when we do detect changes. I'm gonna focus on the what's below the arrow, because I see Libby popping up. So we do need to get as much information as we can as early as possible. I know Carl also touched on that, the need for sort of longer baselines and more time, longer time series. But most critically, we have to improve the transparency of information flow. There's a lot of places in which we don't have information, but there's a lot of places in which we do have information, and it's just not getting presented or going to the right people. The culture of science around offshore wind is fundamentally different than the way we've sort of traditionally approached science in fisheries management. It's a different culture, it's presented differently, its phenology is different. And I think this is where we're seeing a lot of disconnect, where we have offshore wind developers saying, oh, we're collecting so much information, and we have fisheries experts saying, well, but we still don't know anything. And why is that? In my opinion, it's largely a cultural difference. That we have this new era, what I would call science by press release. There's a lot of press releases going out with little bits of information. And of course, we don't do that in fisheries. You can't just put a video underwater and say, oh, there's a lot of scallops there, so we're good, right? Like you would never get away with that in fisheries management. But in offshore wind, it's, oh, now it's an artificial reef. That's science, it's good. And so actually, I think finding a common language between these things and really lining up the experts in both fields with some common standards, principles and practices is really necessary. And I think I'm out of time. I think there's a few more slides, but that's probably about all I need to say. Thank you.
Thank you so much, Annie, and definitely keep an eye on the Q&A. I actually wasn't able to look at it when I was showing the slides, so I don't know what's come in. But we have our last speaker of this round, Dr. Emily Shumchania, who has worked for the Northeast Regional Ocean Council since 2014, managing regional marine life data development and data product dissemination. She manages the Northeast Ocean data portal and is director of the Regional Wildlife Science Entity. So take it away, Emily.
Thanks very much, Libby. And thanks for having me today. Just as a point of clarification, given the sequencing of these talks, I just want to note the Regional Wildlife Science Entity, and I'll talk about this a bit more. The scope is wildlife megafauna and does not include fish and fisheries. And there's a separate organization that Dr. Hare referred to in his talk called ROSA, or the Responsible Offshore Science Alliance, whose sole scope is fish, fisheries, and habitat. And so we'll talk a little bit more about how we partner with them, but the purview of the RWSE, as we call it, is not fish and fisheries. So I just wanted to provide that context at the outset here. So to be very concise about what RWSE is, it's a research collaborative among 4 sectors: federal agencies, states, NGOs, and the offshore wind industry. These 4 sectors fund and support the operations and future research conducted by the RWSE. And the goal is to support this research and monitoring on wildlife and offshore wind by first identifying science and research priorities, then aligning funding with those priorities and ensuring that the appropriate data and standards are in place to support that science and research. Essentially, ensuring that data and research can inform decision-making as shown in the diagram on the right. So when we think about why you would build an entity like this from a scientific perspective, I think a lot of us as scientists understand intuitively that doing science at a regional scale and thinking about these bigger scales, we've heard lots about cumulative impacts already this afternoon, that's important. But I thought it would be also good just to touch on why each of the entities that came forward, each of the sectors that came forward to form RWSE, think it was important to do so, because there's some important differences there that will eventually, trickle down into the work products that RWSE produces and the research that it funds. So I've got 2 sectors per slide here, and this information, I don't speak for these sectors and these entities. They articulated these views to each other in a vision that they created for the RWSE. So I'm just reiterating those points here. So for federal agencies, we hear things about the trust resources. They have protected species that are legally obligated to protect, and they want coexistence with offshore wind, as there are other federal agencies who are advancing the renewable energy sector. Cumulative impact assessment, as I mentioned, they are the ones who conduct the leasing. So they want to be able to be informed about potential lease conditions that they could add or enforce. And they want to ensure unbiased data collection and analysis. State agencies, a little bit different, think about it like as a smaller scale. We heard from Maine today. There's states all over the Atlantic coast that are conducting processes very similar to what Maine is doing. So they want that autonomy to identify their own research priorities, but they also want to come together and learn from each other and not duplicate effort where they could potentially have research priorities be aligned. And this might result also in reduced procurement costs for them and their stakeholders, their residents, as they go out and purchase power from these offshore wind developments. From the industry itself, a really important participant in RWSE, they have an interest in identifying what some of those research priorities and needs are in how they characterize the interactions between wind and wildlife. They're really anxious to see harmonized monitoring protocols. They're looking for efficiency. They're also interested in cumulative impacts, and all of this might serve to reduce the risks that they have and the liabilities that they have in their developments offshore. And finally, NGOs as a sector, they're also very interested in cumulative impact assessment. They're interested in optimizing the funding of some of these things and leveraging resources. And we've seen that in other sectors too. Providing for independent scientific review and making sure that information is shared transparently. So just to dig into the structure and how those 4 sectors come together. So the structure of RWSE is based around these 4 sector caucuses. You see those in the middle of the slide now. We have an interim volunteer steering committee at the moment that we're in the process of finalizing. They're composed of representatives from each sector. RWSE also seeks to convene several expert subcommittees, which you see on the right side of the slide. They will really provide the scientific and technical underpinnings of the RWSE research agenda. The 4 sectors will have representation on these subcommittees, but most importantly, there will be a huge component from the research community as well. Moving down on the slide, I'll get into more detail on these in a minute. Moving down on the slide, the RWSE is hosted by the Northeast Regional Ocean Council and the Mid-Atlantic Regional Council on the Ocean. As Libby mentioned, I am also an employee of NROC as the Regional Ocean Data Portal Manager. We have a small staff that works on RWSE right now. Avalon also serves as a program director for Marco, and Nick Napoli also is an advisor for both NROC and Marco in various capacities. So for those that aren't super familiar with NROC and Marco, just a bit on them, they coordinate and convene ocean planning activities roughly in the same geographic area of interest as RWSE. We are, expanding a little bit further down the Atlantic coast in, engaging stakeholders and in the sectors in the South Atlantic, as well as offshore wind energy development expands down there. But back to NROC and MARCO, you might be most familiar with the regional ocean data portals operated by each of those entities. Each region and each portal also has really strong connections to the regional IUCE associations and the data and tools that those groups provide. The portals drive many of the initiatives operated by NROC and MARCO around developing and reviewing ocean data and ocean data products, both for ocean resources and uses. These are collaboratively developed and vetted by state and federal agencies, industry, and the research community, which sounds a lot like what we expect the RWSE to do. So we thought we were a great fit as hosts for this new entity. But moving on into where that bulk of that work will happen, the review and the identification of priorities in the subcommittees, these are really analogous to the NROC MARCO marine life work groups that are responsible for reviewing and guiding the development of all that marine life data that's on the portals. These groups have been meeting since 2014, and they focus mainly on the parts of the workflow that are shown on the lower right-hand corner of the screen, or I'm sorry, the upper left-hand corner of the screen, the synthesis and developing data products and disseminating data products. We think there's a great opportunity then to align with what the subcommittees through RWSE will be doing, which is mostly identifying those research needs, standardizing data collection, coordinating data collection and monitoring. And so we can complete this sort of cycle of ensuring that the data that are collected are integrated into existing and new data products and actually used to inform decision-making. So these subcommittees are the opportunity for academic and private sector researchers to participate in RWSE. They will be the core working bodies of RWSE and sort of the primary filtering mechanism for the steering committee to help the steering committee understand how to actually implement some of these research priorities. There are 6 subcommittees on tap, and I'll provide a little bit more information about the status of each in a second. But just to go over a little bit of process for how we envision this working, the subcommittees will meet over the course of a year or more, recommend research priorities to the steering committee. The steering committee will then take that back to each of their sector caucuses, consider them, review them together. There may be particular experts in each caucus, which can weigh in on a very substantial way on some of these research priorities. So we want to take the opportunity to do that. And then the steering committee will use all of that input along with potentially other considerations and broader stakeholder input to essentially develop science plans for each of those taxa-based subcommittees and coordinate funding for projects that implement those science plans. So when we take a step back and look at what this means for the next several years, especially in the context of that construction and operation sort of schedule that Dr. Hare laid out in the beginning, this year we've launched RWSE. We just launched in July. We've been formalizing the structure, enrolling members, building funding for operations. And next year, and maybe a little bit into 2023, we'll be developing these science plans. And in 2023 and beyond, implementing those science plans. So very quickly, a more near-term timeline, our steering committee has met several times since April. We've been convening the sector caucuses throughout this fall. And as I promised to show more detail about the subcommittees, we have some meetings coming up that maybe you all can participate in. We have a marine mammal subcommittee meeting on December 10th, a cross taxa subcommittee meeting on the 13th. And we just chatted with many of you about bird and bats earlier this week. And we'll be scheduling that first formal meeting very soon. Some quick research updates. RWSE has already been busy on this front. We're a member of a large team led by Duke University that was awarded a DOE grant about offshore wind and wildlife monitoring. We've been convening discussions and facilitating the development of a funding plan for continuing the aerial megafauna surveys in the Rhode Island, Massachusetts wind energy areas. And we've been supporting the development of a regional passive acoustic monitoring network, or at least the planning of one. And you see that picture on the slide here. That's involved data sharing among all sectors. They've all contributed, and using the regional data portals as planning tools for this. That's something we're really excited about advancing. wrapping up, I just wanted to provide a contact slide here with ways to get in touch with us. And you can follow sort of our progress. We post updates to this page on the Northeast Ocean Planning website for now, neoceanplanning.org/RWSE. Thanks very much.
All right, so we have, thank you very much, Emily, and we have about 7 minutes for this first or second discussion. So if all of the people who've spoken to date could come back on video so that we can ask you some questions. And Emily, I actually have a question. So you talk about creating like the end product being a research plan for the different taxa. Is the intention there that the feds or the states kind of pick pieces of that research plan to implement, or how does it work after you've actually created it?
Yeah, that's a great question. I think what you just described is one option. I think it sort of depends on the different priorities that are identified. There might be one that's of very high interest to a particular developer for some reason. They may say, oh, we want to put some funds behind this and maybe pool, developer money to address a particular research priority. And there may be, federal or state agencies that feel particularly strongly about some and decide to either go it alone or pool funds to do one or more. So there's a lot of different mechanisms that have been tossed around, and we'll kind of see how it ends up when we get the plans.
I guess would Carl or John or Wing or Annie, the rest of you like to weigh in on that question of how we're sort of cooperatively going to make this research happen? John.
I mean, I think many of us know that cooperatively funding something is challenging. It kind of goes back to the incentives within which we work are to disaggregate, disperse our activities. And so I think the regional wildlife science entity provides us an opportunity to do it right going forward. So yeah, I hope that we are able to sort of work putting together joint priorities and then work together to fund those joint priorities. It would be a shame, Emily sort of laid out the way it would be a shame is where each entity picks its own priority and then goes and does it. I think we really need to think about working together to address the grand challenge of offshore wind development, fisheries, and wildlife.
All right, seems like we have a few others who want to weigh in. So let's go with Wing and then Carl.
Yeah, just really quickly, NYSERDA in New York has been doing quite a bit of work over the last several years to identify research priorities and through the State of the Science workshops and also through the groups there. So there's several documents there, NYSERDA, I could share a link there that they've been working to coordinate. And also now here in the Gulf of Maine, there's the research consortium that was just created through legislation this year. It's a nascent group being formed, and that's going to certainly be a place where research can be coordinated here in the Gulf of Maine. But one discussion we've been having in our working group too is ensuring that anything happens here in the Gulf of Maine is closely tied to the work going on to the south. So I'm certain there'll be strong coordination between RWSE and the research consortium.
Yeah, maybe you can provide some of those links in the chat and or Q&A that you were talking about. Carl, and then Annie.
I had a separate question for John, so I'll let Annie go if she wants to address.
Annie, did you want to answer that question? There's also some more questions for you in the chat that I need to highlight. So
yeah, up to you. I can be really quick on that question. I mean, I think that in terms of talking about research priorities and regionalization of science, I think that it is never going to be effective unless we sort of address the elephant in the room here, which is that there are very, very few requirements, and that most of this research is being funded by developers and by states, and frankly now by the federal government, who are strongly incentivized economically toward wind development. And I don't think that means that money can't be used for good things, but I think that in talking about research priorities and where they're coming from and what's required and what we're going to put on the table, I think if we ignore that, we do ourselves a huge disservice and look at missing a huge part of the puzzle. Thanks.
So Carl, would you like to ask John the question, and then I'll ask Annie the questions that are in the Q&A?
Sure. Something that struck me, John, in your keynote was talking about the cumulative impacts. And when you were talking about that, you put up the entire East Coast. Do you think we have enough information about the noise of the system to pick up a single of offshore wind development if it was built out as you showed? And if we don't, what do we need to be doing now to understand that?
Yeah, I'm not qualified to answer that question. I'd want to call a friend and get back to you on that. But it kind of goes down to, what scale are we working at? Are we thinking about Gulf of Maine? Are we thinking about Northeast US? Or are we thinking about whole East Coast? And I think it's really what scale do the different parts of the ecosystem use? For North Atlantic right whales, it's Florida to Gulf of St. Lawrence. For some fishing entities, it's both the Southeast and Northeast US. And so I think we need to be sort of flexible on what scale we're addressing the problem and not get fixed at working at just one scale.
All right, we're going to ask one more question, then we got to move to the next group, but then these guys will all be back. So, Annie, there's a question. I would love to hear you build on how you think there can be movement towards common language, standards, practices between the wind industry and the fisheries.
Yeah, so this is a really, really complicated question. I've been sitting here this whole time since I read it, thinking about how best to answer it. But honestly, I probably would have given a different answer a few years ago. But where we are now, I think that there needs to be neutral oversight. I mean, I really do think that we need more kind of adults in the room, if you will. Not that we're not all adults, but we probably need an adult that's standing a little bit aside from the others. And I see that being a role for fishery scientists, for people that do have a long-standing relationship with the fishing industry, that have come to understand the value of cooperative research, of participatory governance, of the inclusion of fishermen's knowledge and co-development of regulations in a way that works for people. I just think we need to integrate that more into offshore wind. I think it's going to be, frankly, a while at this point until a lot of fishermen are really stepping up and saying, yeah, great, we really, really want to work closely with developers. Honestly, in most places, that's not actually where we're at right now, for better or for worse. There are some, right? There are some fishermen, and there are, particularly engaged in project monitoring. But I'm not talking about monitoring when we're talking about research priorities and information we want to get. Monitoring's monitoring, right? But hypothesis testing and actually getting results and adapting to things is a different issue altogether. And I do think we just need more collaboration and more oversight, but also more incentives, carrots, if not sticks, to force that to happen and to get people to really start listening to each other better.
All right, well, all of you can go off video again. We're going to move to our last round of speakers. So our next speaker is Orla O'Brien, who has a background in marine mammal surveying and has been participating in large whale aerial survey since 2012. She has been on the aerial team at New England Aquarium for 4 years, where her current work involves monitoring long-term trends in whale abundance and distribution. So take it away.
Okay, thanks for the introduction and thanks for having me as part of this session. So I'm going to talk about an increased abundance of right whales in southern New England. And I just want to acknowledge all of my co-authors listed on this slide and just point out that this is actually a manuscript that is currently in review. So just to start off pretty basic with some background, right whales have come up in presentations on previous days, but just so we're all on the same page, the North Atlantic right whale is a large, long-lived ram-feeding whale. Their main prey are calanoid copepods, but they also feed on other species of zooplankton, such as centropodes and pseudocalanus. They're coastal whales, and their distribution used to be across the North Atlantic as part of the Eastern North Atlantic as well, but as a result of whaling, only the North American part of the population survives. They're currently critically endangered, and the most recent population estimate is about 336 animals. So over the past few decades, right whale distribution has changed dramatically. They used to follow a pretty predictable movement pattern from their calving grounds off the southeast United States up the coast to feeding grounds in the Gulf of Maine and in the Bay of Fundy. But around 2010, there was a change in bottom water temperatures in the Gulf of Maine, and this change coincided with a change in right whale distribution patterns. And this change in distribution patterns has resulted in large declines in once important habitats for right whales, such as the Bay of Fundy and the Great South Channel, an increase in the importance of Cape Cod Bay as a feeding habitat, and the emergence of two new habitats, the Gulf of St. Lawrence in Canada and the Southern New England shelf waters, which are the focus of my talk today. And these changes in right whale distribution have complicated the conservation and management of the population as we struggle to keep up with where and when protections should be placed. So one of the areas that right whales have moved to is Southern New England, but this isn't actually a new habitat for right whales. Early whalers hunted these whales off Nantucket and Martha's Vineyard. Since the beginning of modern survey effort in the late 1970s, small numbers of right whales have been seen periodically. However, until 2011, survey effort in Southern New England was pretty sparse. Also, there are plans, as we know, to develop the first US large-scale commercial wind farm in this area. But we don't know a lot about how this will affect large whale species, including right whales. And I just want to point out here that we also see humpback, fin, and minke whales regularly in the area, as well as occasionally sei and sperm whales. So our existing knowledge of the effects of wind farms on marine mammals relies a lot on European studies. And in those areas, the main marine mammals are pinnipeds, such as gray and harbor seals, and harbor porpoise, not large whales. And findings from these studies are fairly mixed, and some point to displacement, and there are variable levels of recovery in wind energy areas post-construction. In terms of effects, we can divide these into short-term effects and long-term effects. Short-term effects could be temporary displacement during construction, probably as a result of the impulsive noise of construction, but long-term effects are more difficult to sort out, whether or not whales are permanently displaced by wind farms and why, and if they aren't displaced, are there downstream effects, maybe as a result of increased interaction with vessel traffic? So there's a lot to sort out. So as a result of the interest in developing wind energy in Southern New England, the New England Aquarium began conducting systematic aerial surveys in 2011 to collect baseline data in this area. Previous analysis of the data through 2015 showed small numbers of right whales, and more recent work has suggested that there's a potential change in right whale use of the area. However, trends in right whale abundance have not been quantitatively assessed. So to assess trends in right whale abundance, we use New England Aquarium aerial survey data. There are 3 eras of survey effort. In 2011 to 2012, surveys focused south of Martha's Vineyard in Nantucket. In 2013 to 2015, survey lines were added over the Rhode Island Basin. There were no surveys in 2016, but beginning in 2017, surveys focused on a slightly smaller area covering the wind energy lease zones pictured in white on the map. And we chose to use this survey area size to calculate right whale abundance. So using our data set, we fit a detection function to our right whale sightings. This detection function was used to calculate seasonal density using the standard line transect equation. We also calculated relative abundance, and we did this to have another metric to look at right whale use of the area. And the metric of relative abundance we used was IPUE. After this, we analyzed trends in abundance and relative abundance. We excluded summer and fall from the trend analyses because we didn't have enough years in those seasons. We used a GLM to assess the trend of abundance, and a linear model to assess the trend in relative abundance. During the time period analyzed, the team flew over 40,000 kilometers of broad-scale survey effort and cited over 350 right whales. So here are the results of our abundance calculations, and there are two main takeaways from this table. The first takeaway is that estimates are higher in the second half of the survey period than the first half. And the other main takeaway here is that whales are present in the second half in summer and fall, but completely absent during summer and fall in the first half of surveys. And for the second metric of abundance, IPUE, we have the same general patterns here, which is to say that estimates are generally higher in the second half of surveys in the winter and spring, and we see an absence of right whales in summer and fall in the first half of surveys, but presence of whales during summer and fall in the second half of surveys. So the GLM showed a significant increasing trend in right whale abundance. Although the confidence intervals around specific abundance estimates can be kind of large, the trend estimation is actually weighted by the variance of the abundance. So the significance of the trend is robust to the confidence intervals. And we also found an increasing trend in the linear model of relative abundance. And I just want to point out that we did remove the winter 2013 data point because it was overly influential on the trend, but removal of this outlier didn't affect the significance or the direction of the trend. It just resulted in a better fit of the remaining data. So some takeaways here are that although right whale occurrence in southern New England has been documented since the whaling era, right whale use of southern New England has fluctuated. And in our analyses, we found a significant increasing trend in winter and spring right whale abundance from 2013 to 2019. And we also found that right whales are now present here year-round. And the novel occurrence of right whales in this area during summer and fall began in our data set in 2017. And some conclusions here are that right whales are returning to this historically important area, and this repatriation is probably part of the larger scale distribution shifts in right whales that are driven by climate change. And southern New England currently represents an important habitat for what is a declining right whale population. And wind energy development is scheduled to begin in 2023. And as it progresses, we believe it's critically important to document the effects it's going to have on right whales and other large whale species. And one way to do this is to continue to leverage data sets with long time series, which are extremely valuable in ecological sciences. And I just want to end by saying that the importance of the opportunity to understand how wind energy affects these animals cannot be understated. And I just want to acknowledge and thank all the people that contributed to the work and to the funders of the surveys used in the analyses. And I'm happy to take any questions during the Q&A time.
Thank you, Orla. Now we're going to move to our next speaker, Julia Stepanek from Stony Brook. Oops, sorry. Julia is a fifth-year PhD candidate in Dr. Leslie Thorne's lab at Stony Brook University. Her research is focused on assessing anthropogenic risk to large marine mammals, and she will present today on the potential use of novel climate forecasts to anticipate whale distributions in the Gulf of Maine. Take it away.
Okay, great. Everyone's seeing my screen correctly and everything.
Yeah, it looks great.
Okay, awesome. Thank you all so much for being here today. I'm excited to present some of my dissertation work to you on forecasting highly mobile species. And advance. There we go. So effective management of marine species, as we've been hearing about during this session, is vital for risk mitigation from anthropogenic activities. Within this, it's important to figure out who is impacted, when they're impacted, why, and where that is. And monitoring and management practices are especially difficult to suss out for these highly mobile marine species, including the pinnipeds, whales, and birds that we see here. So the classic approach for management is sort of the static spatial management. This is adopted primarily from terrestrial practices, And this involves sort of area closures. They can be determined for maybe certain times of year or they're year round. But here on the left, you can see this is the southern coast of California, and you can see all of the red and colored blobs are various marine reserves or closures. And if we think about this scale from the perspective of something like a migratory gray whale, it's clear that they're using this area in a very different way than this sort of static spatial management approach. So this might not work for highly migratory species or might not work well for them. And it also could, these regions could be rendered less effective or even ineffective in the face of climate change that could affect distribution shifts or habitat use changes in species of concern. So for today, I'm primarily going to focus on marine mammals as a case study. Marine mammals are impacted by many different anthropogenic activities, including things like vessel strikes, harassment, entanglement in fishing gear, acoustic disturbances, and oil and gas exploration. And there's a couple of different management practices that are in place to sort of deal with some of this and mitigate some of this. The one I'm going to talk about today is predictive modeling. So this is a way to take sightings of a marine mammal or animal in general and create a distribution map of them to look at high use or high risk areas for them. So an example of this is from Dr. Elliot Hazen, who modeled blue whale habitat and distribution on the west coast of the US, where there's lots of vessel activity. And this is a product called Whale Map. It's actually available online, and every month a likelihood of occurrence of blue whales is released for the west coast of the US. So I pulled this map the other day, and you can see the dates for it are from October 1st to November 1st of this year. Well, we're in the middle of November now. So these maps were actually built using satellite data that was recorded from the previous month. But it's not really giving us any big inference into the future. And this is sort of the crux of the issue here, is that a lot of the distribution mapping involves inferring potential future management problems that are based on recent conditions. But what happens if we shift that narrative from a reactionary management framework to a more anticipatory one? So the goal here would be to use something like a climatological forecast to anticipate where and when conservation issues are likely to arise. So we used sub-X models. These are a suite of models that are released by NOAA's MAP program. And these provide subseasonal predictions of surface and atmospheric environmental covariates. We specifically focused on the GEFS model, even though there are a bunch that you can choose from, and we focused on sea surface temperature. This is a product that's available more or less from 1999 onwards, minus a few years. And it's unique because they release this forecast every 7 days, and it forecasts out to the next 35 days in the future. And I just want to note we chose GEFS specifically because it had the best skill for our study region, and I'll get into that in a little more detail. Our ultimate goal was to use these sub-X models to forecast whale density in regions of wind energy interest in the Gulf of Maine. So for today, I'm just going to focus on humpback whales because they're primarily what I'm looking at in my dissertation, and that's what I modeled, but this could apply to many different highly migratory species. So humpback whales grow up to about 18 meters in length, and they usually migrate into the Gulf of Maine in spring and summer months to feed. In the Northeast US, they primarily occupy continental shelf waters. And they face a number of anthropogenic threats. Mortality recently is thought to be primarily due to entanglements in fishing gear and vessel strikes. And since 2015, there have been enough mortalities documented along the East Coast of the United States that there's currently a federal investigation or unusual mortality event in place. So while this population of humpback whales is not ESA listed, there are other major anthropogenic threats facing this population that are important to consider in the cumulative impact framework of various anthropogenic impacts on these species. So my process was to build a humpback whale density model. Then I wanted to assess how good the forecast, the sub-X forecasts were. And then I basically combined those two things together and forecasted a humpback whale model. Lastly, I wanted to assess that humpback forecast in the offshore wind areas of interest in the Gulf of Maine. So to build the marine mammal model, I used standardized line transect survey data collected along the East Coast from 1998 to 2016, and it is still ongoing. This involves an observer on the left and right side of a vessel or plane. They're counting the number of animals that they're seeing, and then post-hoc statistical analyses are conducted to provide an estimate of abundance along the track line, as we just heard about in the previous talk. I then took those data and I related them to relevant environmental variables at the location and time of a sighting. So the variables that I included in a generalized additive model, or GAM, were depth, distance to shore, and bottom slope. The only dynamic variable I included was sea surface temperature and derived products. So while other people who are modeling this data could incorporate things like chlorophyll A or sea surface height, we don't have forecast products for those, so I wasn't able to include those in the model. I was limited to just SST and the derivatives. So next after this, I wanted to say, okay, I have my SST forecast data. How good is it? So this is the forecast skill. This is a correlation between a forecast and the satellite SST that was recorded for a given historical time period. A higher value here, indicated in dark blue, represents a better agreement between the forecast and the real data. And so if we look here, we can see forecasts 1, 2, and 3 weeks in advance from left to right. You can see the week 3 forecast is not very good. So we primarily focused on forecasts up to 2 weeks in advance to be able, because they demonstrated high skill. But you can also see this is a pretty granular product. It's at 1 decimal degree. And so we used a statistical downscaling method that incorporates the forecast with satellite SST measurements to resolve a product that was actually at 0.2 decimal degrees, which has way more ecological relevance for the kinds of questions that we're asking. So once I have this forecast product, I incorporate it with my distribution model and create a humpback whale forecast. This forecast actually performs similarly well when compared to traditional satellite-based prediction surfaces. And from that, I was interested in seeing what this looks like for the main areas of interest. So here, as we saw earlier, this is the broadest area of interest. I focused on this because it was the shapefile I could find publicly available online. And these values and the colors represent the number of humpback whales per 100 kilometers squared. I have 2018 and 2020 represented here, and those are the rows, and then the columns are April 10th through May 1st in their weekly forecasts. And it's important to note that the April 10th forecast was actually released on April 3rd. So that gives about a week of leeway. And you can see differences both within and between the years. So there was a drastic change in the forecasted arrival in 2018, right on April 24th there. And we don't see that in the same way in 2020. So this shows some of the variation between the 2 years here. This gives us information about when we could conduct or avoid various disruptive activities and maybe determine high-risk areas and weeks for highly migratory species. And it gives you that information a week in advance. It's important to note this is not a replacement for monitoring or anything, but it would be a tool for decision-making. So in conclusion, this is a first step that shows how subseasonal forecasts could be used to predict distributions of highly mobile species. This is actually very generalizable to a variety of different species, different ecological models. You're not restricted to just using GAMs, different management scenarios and different regions. And in the future, I'd be interested in looking at some sort of iterative model that could improve as real-time data are added, sort of like what happens with weather forecasting. And also the SubX folks have created a model that has a greater forecast skill into the future using multi-models, where we might be able to look 2, 3, or even 4 weeks in advance. So with that, I'd like to thank you all for listening. I'd like to thank collaborators and co-authors, marine mammal observers, and the agencies that support SubX. And I want to mention that I plan on finishing my dissertation, hopefully by next summer. And I'm sort of looking where to go next. So if anyone's interested in chatting, my information is in the upper right-hand corner. With that, thank you very much for listening.
Awesome. Thank you, Julia. And now we're to our last but not least, and I believe we do have Everett now, right? We were having a little trouble getting him on. Everett Rosowski from the University of Maine. It recently graduated from Bowdoin College and began his graduate studies at University of Maine's Darling Marine Center. he's working with Dr. Damian Brady, who is in charge of all of this program, on fisheries monitoring for the University of Maine Monhegan wind test project, and has been staying on the island the past 6 weeks. So I'm so glad, Everett, that you were able to get on and present today. So take it away.
I'm going to be talking to you today about the methods and motivations behind the UMaine Commercial Trapping Survey, which has been undertaken over the past 6 weeks on Monhegan at the future site of the New England Aqua Venice turbine. Before we dive into what I've been doing on Monhegan during the sampling period, I thought I would start with a brief overview of the NEAV project. So on the right, we have a cartoon schematic of the turbine. It'll stand about 732 feet from the water line, with another 66 feet submerged, making it the largest structure in Maine. Dwarfs apartment complex is in Portland and is also larger than the 200 Clarendon Tower in Boston, the largest building in all New England. This project's been in development for over a decade. So it began with site selection pre-2010 and baseline monitoring in 2009, 2010 through 2014. Then the installation of a scaled down 1/8 to 1/8 scale turbine as proof of concept, the Volturnus in Castine during 2013, 2014, and into our present state of preparing / beginning monitoring of fish, wildlife, and other resources in preparation for the 2024 installation. So the turbine itself has been designed by UMaine's Advanced Structure Composite Center and will be moored with a semi-taut mooring system. So generally there are 2 mooring types that are under consideration for a structure this scale: the catenary chain hybrid or semi-taut. Catenary chain hybrid links sort of fall away from the structure very quickly and then lay across the seafloor, which has potential to impact the benthos, whereas the semi-taut mooring system keeps the line in the water column, has a little bit of chain at the beginning and then onto the synthetic rope, and then onto more chain at the bottom anchoring the mooring. This comes with a significantly reduced scope, which sort of decreases some of the spatial scale issues in siting a turbine. Installing a structure this size, though, does have the potential to spark unplanned changes to the surrounding resource environment. So therefore, fish, wildlife, and resource monitoring is required by BOEM to install offshore wind power. And as the first of its kind in the Western Hemisphere, this project and its required monitoring aspects offer a really unique research opportunity to discern some of the impacts on the surrounding environment, or if there are measurable impacts on the surrounding environment from this turbine. So an important portion of this monitoring is determining whether or not there are measurable impacts on commercial fisheries. The first portion of this fishery monitoring has begun in the form of the UMaine Commercial Trapping Survey. So I arrived on Monhegan October 8th and have been fishing with the help of 4 Monhegan captains, 2 36 trap arrays, with the primary goal in the long-term survey of determining whether or not there's an impact or a measurable impact on commercial lobster catch or landings post-installation due to installation and operation. We also have a secondary goal of modeling lobster population during each haul at each site using mark recapture analysis. So how are we actually doing this? We designed a control impact. We designed a before-after control impact study. The study will survey the test area in yellow on the right, and then also a control site that was sited with the help of Monhegan lobstermen to control for bottom type, bottom depth, and fishing pressure. Surveys will take place each fall and spring for 2 years pre-construction and 2 years post-deployment, making this the first survey or first sampling period. And in this phase, the before phase, we're really just trying to establish baseline size distributions, sex ratios, population estimates, catch per unit effort, and landing per unit efforts indices between both the different trap types that we're using, the different arrays, and the seasons. So we're actually getting this data by sampling the testing control sites with paired vented and ventless traps, and this allows us to address multiple size classes. The traps are located with random stratification over a grid at each site. for 50% site coverage. And this random grid stratification sampling is similar to that of the Vineyard Wind ventless trap survey. Additionally, we tag the lobsters, as mentioned before, to perform mark recapture analysis using a method developed by the Wally Lab for lobster population analysis in Penobscot Bay. So what do we actually take from the lobsters? We don't take anything, take biological data, including carapace length, sex, number of claws, molt status, V-notch, and egg status for females. Each lobster is then tagged with a unique ID and then released. We've tagged about 7,000 lobsters to date and have another 2 hauls this weekend. So hopefully we'll deploy about 1,000 more tags. We've received over 150 commercial recapture reports so far, and then have another 175 recaptures from our traps. On this slide, we have the haul schedule so far. Here, day 0 is when we set the traps. We've been aiming to maintain a 4-night soak, but over the course of the study, we've been pushed to 6 nights twice by weather issues, and then also 3 nights once. So what have we actually seen so far? On the right, we have the box plot of all the entered and aggregated data for carapace length for each site from October 14th to November 13th. We'll ultimately have data through November 21st for this sampling period. And so far, it seems that the control site is a good analog for our test site, at least from this standpoint. Size distribution is similar between the sites, and there's no significant difference between means for carapace length or for sex ratio. Not shown here, but we have seen the mean increase over the course of the study from about 76 millimeters in the first haul up to 78 millimeters a couple weeks in, and then up to 80 millimeters this last week. But what's really interesting is that at no point have the means between the sites separated. So they're actually showing similar dynamics in size distribution from what we can tell so far. Here we have the commercial recapture and release information overlaying over the study site map from earlier. I should mention that the depth data here is from Tom Weber at UNH and was taken previously at Monhegan. But we've received over 125 commercial recaptures with locations, and the recaptures have largely been reported north of the release locations in deep 100+ meter water. Our furthest traveling lobster to date has been 5 kilometers, which we've seen 3 times. When I get back to shore, we'll be doubling down on outreach to harbors outside of Monhegan to make sure that there's no reporting bias, as most of our reports have come from within the conservation area so far. So what have we talked about so far? A brief overview of the NEAV project, as well as this phase of monitoring, and then some super preliminary data from this phase of monitoring. And our next steps will include finishing the sampling for this period this weekend, preparing for the spring 2022 sample session through outreach, and then continuing the catch and mark recapture analysis. Ultimately, we really couldn't do this without the commercial partners. Chris, Lucas, Brandon, and Nick have been, I mean, they do the actual fishing, so we really just couldn't be out here without them. And then additionally, thank you to Chris, Cash, Rick, and Damien for all their support through this.
All right. Well, thank you very much, Everett. So glad you were able to make it back on from the island. Hoping that the rest of our panelists will come back on. We have another 9 minutes or so. There are a few questions have come in, and I think I'm going to start with those and then go. You guys may have questions for each other. Definitely keep asking questions, audience. I have some questions as well. So there are a few questions for Orla, and maybe you've seen those in the Q&A. One, I'm just going to put them all out there and you can answer them. So we have one from Rick Wally. Interesting talk. Is there any evidence that there are concentrations of Calanus in the southern New England right whale aggregation area? If so, is that surprising, as Calanus is a cold water copepod whose distributions we are told is shifting north? And then from Chris Orphanides, a few questions. Also, can you talk about the seasonal distribution differences seen in your surveys? Also, do you have a sense of what is driving the distribution of these whales and how offshore wind development may or may not impact those factors?
Yeah, so those are all great questions. So thanks for asking them. So they're kind of both intertwined. So I'll start with Calanus. So we do have evidence that there is Calanus in this area. It varies seasonally, and it also varies between years, which is interesting. As for whether or not it's surprising, I'm not a Calanus scientist, so I guess I wouldn't know specifically, but I will say that my understanding is that in the Western Gulf of Maine, maybe the distribution shifting north isn't as prevalent. For instance, there is quite a lot of Calanus in Cape Cod Bay, for instance, as a result of this regime shift. So maybe it's part of that, I'm not really sure. And then for Chris's questions, the seasonal distribution differences on the surveys are actually incredibly interesting. So in the summer and fall, which, as I mentioned in the talk, is a new time of occurrence for right whales, we've started to see that they're only present during those seasons over the Nantucket shoals. And in the winter and spring, they're more likely to move west into what are going to be the wind energy areas. And that also changes between years again. So some years you have a really strong concentration of whales south of Muskega Channel or south of Martha's Vineyard, for instance. And some years in winter and spring, you don't have as strong a concentration, or that aggregation has shifted east to the Nantucket shoals. And I think what's driving that is prey. And so if there's more high value prey to the west, which tends to be Calanus, then they move there and we see huge aggregations. But if there is a lack of high value prey in that area, then we're still seeing right whales in southern New England. They're just concentrated more to the east. And I guess as far as speculating on how development will impact that, I can only say that, if there's Calanus south of Muskegon or Martha's Vineyard, then whales are more likely to interact with it.
Thank you. Oh, it looks like we may have another question. Hold on. Actually, I'm going to ask a question, then I'll go to that. So, Julia, I was curious and interested in your climate modeling related to whales, and you may have mentioned this and maybe I missed it, but have you tried to do some ground truthing of whether the whales are in fact in the places where you are predicting that they will be? And that leads me to, that's a question for you, but then for the broader group is, how do we provide the information that we need to provide for offshore wind development in a changing climate? So I think Wing and others had noted, we're providing this information that's sort of the historical information, but that's also dynamic and changing, and we're going to need models like what you're talking about, Julia. So I don't know, first, if you want to answer that and then talk about the how you've ground truthed it or plan to, that would be great.
Yeah, so it was surprisingly or unsurprisingly pretty difficult to find a good metric to be able to compare what's essentially a rare sightings event in the grand scheme of the data that we have to what's essentially a relationship between a species and its environment. So what we've come down to is basically that we're modeling where whales could be, but we're not necessarily modeling where they are. We ended up using a similar metric to what some Becker studies on the West Coast have done, of looking at the observed abundance in a region that was observed for, say, the Gulf of Maine, to the predicted abundance for that region, and using a ratio of that. And we found at our ratios, you want it to be close to 1 because you want it to be approximately the same, but we found that the ratios when we built those distribution maps with the forecasts compared to building them with the satellite were both reasonably close to 1. There was a slight difference, but it wasn't enough to give us concern. So that's the extent of the ground truth thing that we've done to this point. That's where I'm interested. If I could do my dissertation over again, I'd want to do it with a Bayesian model, and maybe come up with some sort of iterative update or iterative real-time test of the data as flights are going and as observations are being made. Because I think that that would be a really interesting way to sort of refine those species environment relationships. And that sort of starts to answer your second question of like the continuity of the relationship between like where a humpback whale is and a temperature of 9 degrees Celsius through time or something like that. At the end of the day, the modeling I'm doing, I'm trying to kind of capture their food prey distribution via SST. And we know that those are not constant through time. We also know that these whales are generalists, so they're not following the same sort of like Calanus dedicated shifts that right whales are. And so this is actually another part of my dissertation I'm interested in getting into a little more is if we're relying on a relationship between a whale and its environment in 1998, can we still rely on that moving forward after this? And I know that some groups have dealt with that by splitting up their models pre and post 2010 or 2012. But yeah, that's something I've been thinking about a lot in terms of building models using just the past 5 years and iteratively sort of testing and training with that. And yeah, that's where I've gotten to, but it's something I've thought a lot about.
So would others of you like to weigh in on this, providing information products in a dynamic environment, particularly dynamic environment? Wing.
I'll just say briefly, I think something we need to keep in mind is that we're trying to predict distribution of animals when we know we're putting new structure in the water, and that new structure in the water itself is going to change the ecosystem right around where the turbines are put, but are more broadly going to change the ecosystem. So I think that's something that we will struggle with, is that these predictions of distribution abundance now are going to change because of climate change, but they're also going to change due to putting structure in the water. And I think we have to be ready for that and aware that, you know, the best predictions now are only that. And put something new in the water, there's going to be changes in the microclimate around the turbines themselves and then more broadly.
So along those lines, Everett, wondering what your predictions are for what this ecosystem is going to look like and what the lobster abundances might look like once the turbine goes in.
Well, hopefully there would be no measurable impact or no change. So far, what we've seen is that our traps that we fish on mud, we have both of our sites are sort of half hard bottom and half mud, seem to be doing really well. I think that the Montana Lobster Conservation Area provides a sort of interest, it provides a really unique study site in the Gulf of Maine in general because of the season. But ultimately, just hopefully to see no measurable impact. The sites are very similar. They're both successfully fished by the license holders on Wauhegan. So hopefully that would be the continuation.
All right, well, I'm just noticing that we just went over time. Thank you to all. I think we're over time, right? Damien, Cassie, Elizabeth, someone in charge?
I'm here right now, but it's only 1 minute over time, so.
Okay, good. I didn't know if there was a plan to close out. Otherwise, I suppose we could keep going, but I'm sure that people probably have other plans for the weekend. So you tell me.
Yep, I think it's probably, time to end it. Yeah, I just wanted to say from Damien on behalf of the Ragon board, thank you everyone for attending this year, and we look forward to seeing you all again next year. Yeah, thanks everybody.
Thank you very much. Happy Thanksgiving.
Thanks, everyone.
Take care, everybody. Bye.